Y'all let me have sound coming through here. Okay, so we start in 15 minutes. Just letting people come in. Make sure and send this to anybody who you know doing biology unit 1. We reach.
Let me tell you something about CXC. Nah, not this cinema CXC. The Caribbean Examination Council. CXC is amazing. Oh my god, you don't need nothing else when you have CXC. First, you have your paper ones. Amazing. Multiple choice exams. You shade little dark circles and come closer. Come closer. Let me tell you a secret. When in doubt, choose C. Better yet, all you have to do is a bunch of paper ones from before, and you're guaranteed to have done at least 50 out of 60 questions by the time you write the exam. Thank God for Google Drive. Incredible. And you get questions like somebody get paid to draw this, yes? But then you have your paper twos. They call these the structured questions. You just answer the questions. Simple as that. Explain to Sunil the advantage of his testes being carried outside body. What, you stupid? Just answer the question, dummy. And then you have your paper trees, labs, SBAs. See, the trick is to not do your labs and SBAs, and then at the last minute let pressure build up and then steal somebody else's book and copy all of their labs. You're guaranteed to do real good. CXC is gangster. See, what you do is you pay them to write exams that you don't want to do, while they just do whatever they want. It have hurricane, exam, volcano, exam, pandemic, exam. Even when things go wrong and a bunch of people receive the wrong grades, like in 2020, they denied for years because they can never be anomalies. Oh my god, what a beast! CXC is the best. 10 out of 10. Would recommend for everybody.
What going on all here? Biology paper 2 broad topics review. Here we go. But first, a message from our sponsor.
Let's play
Raid Shadow Legends.
Start.
You're raiding thing. You're doing thing. You're doing thing. You're downloading thing. You're raiding thing. In the shadows. You become a legend. Raid Shadow Legends. Oh yeah. There we go. Hopefully like the intro. I spend a little too much time on that than I should have spent. I could have been doing Cape Physics. Alright, so let me get started. So let me give already rundown here. We already have a whole set of past papers done on some videos. And wait, first, let me know audio and music commentary, right? Okay, so it look like it coming through. Alright. So what we're gonna start with sorry. I'm gonna load this down a little bit. So what I feel we'll do is that we'll start reviewing work first, right? And then we'll go through some past papers after as we're doing it. Because as we're doing the past papers, well, you know, we don't have to do all the questions because it's just the broad topic ones. So let me get started here. But first thing I want to go through it all here is to show you what come last year. And it's likely that the things that come last year not gonna come this year. Don't take my word for it, but you know, we'll keep that in mind so we wouldn't spend too much time on it. And let me see if I can get this here. Alright, that's now So this is July 2021. Let's see if we get this here. Get this. So this is what come last year. And the thing is, some of the broad topics from last year come this year. So I don't know, I have a feeling like they won't repeat it, but we still will just go through what come, right? Now this live stream going into the night, all you know how it is, all you was here last year for CXC. Well, CSEC. But let me go through this here. I'm not going through like all the different answers and things, but just showing you. This first question here had like a annotated, to draw annotated diagram of an E. coli bacterium. This was the micrograph, by the way. I didn't redraw it, all I do is label this. But as you can see, it's a very bad micrograph. And you can't really tell anything from it. As I say, it looked like a kiss goodie. So I don't know. But they say label, put 4 annotations, however. So I have a feeling like a drawing of a cell that not coming. What might come instead would be like a die-cut root or a die-cut stem. So what else come here was similarities between plant and animal cells. To me, that's like a CSEC question. And same thing, differences between animal and plant cells. Now, what didn't come was endosymbiotic theory and anything to do with plant tissues, roots and stems. So you never know if that'll be it here. Well, there's a biochem question, a little biochem. For module 2, sickle cell anemia did come. And usually sickle cell anemia might fall under mutations, cell variation and things like that. So that mightn't come. So we had a whole thing about sickle cell anemia. And in module 3, we had stuff with hormones, FSH and LH. So again, that mightn't come. If it come, it might come for like a little bit of maths. Role of FSH and LH. Implantation. Then we had stuff with fertilization of a plant, of a flower. And we had a whole 15-mark question here on asexual reproduction. Now, I don't know if you all know, but the syllabus changed in the last 2 years, as far as I know, to exclude asexual reproduction in terms of all the mechanisms involved in it, like when you have spore formation and things, those things not there again. Your teacher might have still go through it with you. All you have to know now for asexual reproduction is the advantages, disadvantages, limitations, things like that. You don't really have to go into how it is done so much. You just have to know how to name a few mechanisms, but not to go in depth with it. So what I think we'll be going through most here is the plant and animal reproduction, because that is the entire module, basically. And because they didn't really break that down into anything else. The other parts of it, which is what was like I don't think it's really hard. Enzymes, cell division, mitosis and meiosis, that might be the kind of tough one. But also mitosis and meiosis involves mutations and natural selection, which I find is easy. I find all they get away this time. All you have to know triglycerides and phospholipids and all kind of thing. And nucleic acids, all you have to know, that's all they kind of get away, but that could be a good thing and a bad thing, for all you know. So what we're going to start with is we're going to go through some of the work first, and then we'll do some past papers just to make sure that we understand the thing now. But here we're gonna go for it from scratch, because you never know, huh? You never know. I have a little bit of a cough for you. It's not COVID, but just letting you know. So first thing here, let me see how this showing. It's supposed to be, I know the text a little small here, but I could zoom up a little bit. Let me zoom out. This don't really come for I never really see it come for any paper 2. You might get a little your odd paper 1 question with the difference between light and electron microscopes. What you use in school is light microscopes, of course, which has much lower magnification than an electron microscope. You don't need to memorize the numbers, but it's much lower. And the light microscope uses glass lenses, right? And the electron microscope uses electromagnets, because that basically uses electron beam radiation to form the images. And well, light microscopes, you just have the light bouncing off and going into your retina, and you're seeing that. So with light microscopes, you're seeing the whole visual spectrum. You're seeing Roy G. Biv, basically. You're seeing all the colors, because is visible light being used to form the image. Where electron microscopes more depend on higher frequency radiation. So you don't have it's kind of like ultrasound, but not the same as ultrasound. But you know, with an ultrasound you don't get any color being formed. So electron microscope, you get a lot of clarity, but you don't get the color. So any colored electromicrograph you might see, that was done post-production, like somebody would have colored that in computer software. Using a light microscope, you have living cells and tissues are used. Typically you take like a little transverse section through something, or you could even use whole mounts. Like the other time I find a little flea on my cat and my pet cat, not that kind of cat, right? And what happened there is that you could actually put that under a slide and you could view that, and it was actually still alive. You don't have to actually dehydrate or kill the specimen. Electron microscopes, on the other hand, you have to dehydrate the cells. You have to cut them real thin because the electrons had to penetrate those samples. Of course, those types of samples are very expensive to make, and only really professionals could do it. Not me in my house with my microscope putting a flea, under a microscope. So the main advantages is, well, light microscope is much more affordable. That's why you could use it in school. The slides can last a very long time, and there's little risk of distortion when viewing it because, again, you're not using electron beams, it's just light. And the main disadvantages of the electron microscope is basically the opposite of this. It's expensive, the specimen deteriorates during viewing because the electrons actually bombard against it, and there's a higher risk of distortion. But the main advantage of using an electron microscope is the higher resolution and the higher magnification. But you won't really do this in school. You might even do this in university only when you get to a really high level. So, I'm going to show you all what an electron micrograph looks like. So here we have one. This is the same PowerPoint that I create for the let me zoom this out. Actually, no, that's fine. So this here is the typical animal cell. We have some parts here that are not labeled. So if we look here, which is kind of a porous material, or like a porous barrier, it basically allows stuff like messenger RNA to exit. It's kind of like the nucleus has its own plasma membrane. So you'll see wrapped around this here and is usually adjacent to the nucleus because these contain ribosomes. So this is what you call the RER, or the rough endoplasmic reticulum. It's quite a mouthful, but basically this is like, think of it like a little conveyor belt for the synthesis of proteins. They house ribosomes, so you have the rough one which has the ribosomes. Think of it like little sandy grains on it, that's why I call it rough ER. And then you have the smooth ER. The smooth ER is not really for protein synthesis because it doesn't have ribosomes on it. You might use those more to synthesize lipids and things like that. So here, although you can't really see it, but they look more elongated, these are mitochondria. We'll get more into that just now. And this here, it look like nothing, but they're usually adjacent to the endoplasmic reticulum, but there's a Golgi body. I know you really can't see anything here because it is really just look like a bunch of blobs. This is the plasma membrane here. And here we have some secretory vesicles, which are usually used for endocytosis and exocytosis, basically to transport materials around the cell or to get them to the extracellular matrix, basically to get them out of the cell. And these are usually wrapped around with phospholipids. We won't really get into those. So here we have the labels again. And these are the ones that you kind of have to take note of for the animal cell ultrastructure. Ribosomes, which are located on the rough endoplasmic reticulum. The nucleus with a nucleolus, the nuclear envelope. And this is the nice thing. If all you get a cell to label with, and you see in the nucleus clearly, and you see in the nucleolus, that's 3 different labels, right? That count as 3 labels: nucleus, nucleolus, and nuclear envelope. You're real good to go if you get a nucleus in the diagram. Nucleus, nucleolus, nuclear envelope. If you're not seeing a nucleolus, don't label some random thing and call it that, eh? So here we have the mitochondria, the plasma membrane, the Golgi body or Golgi apparatus, and the vesicles. So what do each of these do? I might have to zoom this out a little bit. Yeah. So other things, if you ever have to label one of these, again, a lot of this stuff is C-sec things. They just have a few added things here. We have the cytoplasm. Wait, I see just now. I'm not reading comments here. What is the review to break rate? We are breaking here. We'll break when I get tired. If all they get tired, all they could just go and come back. I just be good to have plenty stamina. So the cytoplasm, which is, just the jelly-like substance that is found in the intracellular part of the cell, keeps the shape, cytochemical reactions, all you know the thing. Basically, here is where you'd have a bunch of vacuoles and vesicles and things traveling back and forth. The mitochondria is the site of ATP production. Is basically the site of energy release. If all you do in Unit 2, you'll get a lot more into that. May I could spend too much time on that, but you have this scary thing that's occurring there called the Krebs cycle, where basically, so I'll go real fast, say you have a 4-carbon compound called oxaloacetate, and that combines with a 2-carbon compound called acetyl coenzyme A to make a 6-carbon compound of citrate, and then through a series of reactions with dehydrogenase enzymes and other things to catch those enzymes called nicotinamide adenine dinucleotide, or NAD, that reduces to form NADH, and that there forms a whole cycle that would eventually convert that 6-carbon compound back into a 4-carbon compound after it loses carbons as carbon dioxide, and restarts the whole cycle. But as that happens, there it enters an electron transport chain where oxygen is the final receptor, where it passes through protein complexes, and those jump as it goes, and that there is actually what forms ATP, as those hydrogen ions passes through ATP synthesis. You don't have to worry about that right now though, because that's something you're doing in Unit 2. So take your time. So then around here we have the plasma membrane, which is also called the cell membrane, but we're gonna call it plasma membrane for now. And that regulates this is supposed to be exit. This regulates the entry and exit of materials. So small non-polar materials basically mostly could pass through there. Could pass through the bilayer. We don't have to go through that so much, but if it's a large polar material, it would need a protein carrier or protein channel. Water would need an aquaporin in order to pass through, but stuff like gases could pass through just easily because they're small enough. So we have the nucleus, the rough ER, the smooth ER, we went through that. Right, so the Golgi body is something I didn't really get into. What that does, and you could see it here on the diagram, what that does, right, is that is kind of like a packaging system. So that would receive material from the endoplasmic reticulum, package it into vesicles, and then that would transport them to the cell membrane, basically exocytosis. You don't have to go through too much with that, I guess, but it's a packaging system. It's not the reason why it's very difficult to detect even with an electron microscope is because it's literally blobs that break off little vesicles to go to the cell membrane to let out things like, if it's a white blood cell, it might let out some antibodies, it might package some waste, some enzymes, different things. Lysosomes contain enzymes. Lysosomes is basically the garbage men of the cell. These would contain digestive enzymes to kind of clean up these things don't live forever. So like a mitochondrion might dead, right? You'd be like, well, what are you doing? You have a dead mitochondrion in the cell. What are you going to do? The lysosomes would basically kind of engulf it, and it would have digestive enzymes in there. And, it's basically the Das funeral home of the cell. It will clear it up, it will take it, send it out, and then it would excrete it. That's what lysosomes do. Centrioles are only found in animal cells. Centrioles produce microtubules to pull chromosomes to the ends of the cell, basically to the poles of the cell during cell division. So plants don't have centrioles. Plants have something called a MTOC, microtubule organization center. It sounds like a place you could go, but it's just a different system of doing mitosis. So you don't really see the centrioles in plant cells because only animal cells have them. Little fact here, the centrioles is actually what does extend the sperm cell to form the flagellum, the tail. So when it extends, it extends all the way to the tip of the tail. Nice. So the nucleus, we went through this already. As I said, the nuclear membrane is porous because this allows material to exit, such as messenger RNA. The pores are rather big, but you have to remember that it is not an impermeable barrier. It could let stuff pass in and out of the nucleus. We have DNA and RNA in here. There are 3 types of RNA, which we don't really have to go into, but it's messenger RNA, transfer RNA, and rRNA, ribosomal RNA. The nucleolus contains mainly ribosomal RNA, which is of course used to form ribosomes, which helps with protein synthesis. Not bad. I say if you get a nucleus to label at least nuclear membrane or nuclear envelope and nucleus. You could even label nuclear pore if you're seeing it. If you're seeing it, don't label it. And you could label nucleolus if you're seeing it. This is an electron micrograph of a nucleus, so you could see, you could see the comparison here. Kellogg-Jones, you would call NADPH, but as unit 2 thing, NADPH is for photosynthesis. So NAD and NADH is for respiration. So you'll find that in the chloroplast. Again, all you don't have to worry about that, I'll study tomorrow. Alright. So here we have a plant cell, and I'm going to zoom this up a little bit. Electron micrograph of a plant cell. And let me give all you a little minute here to see if all you could figure out what is what here. We already have the plasma membrane. Remember, we have an outer membrane here. We have the Golgi body. As I say, it don't usually ask it to label that because it is literally look like a bunch of blobs. We have this thing here. We have a nuclear envelope, so you could guess what that is. We have the mitochondria. Now you might be confused why sometimes the mitochondria does look a little more elongated with the little folds inside, and sometimes it does look like, I don't know, it's look like the inside of a pomegranate. But that's because, sometimes you could cut the mitochondria different ways. It depends on how you have the cell cut, whether it's a transverse section or it's a longitudinal section. So if you cut it longitudinally, it would look more like folds. Transverse, it would look more like pores. I fully understand what I mean. But basically, if you look for anything oval-shaped in there, small and oval-shaped, that would be a mitochondrion. So here we have a tonoplast, and if you remember what a tonoplast is, the tonoplast is the membrane that encompasses the large central vacuole here. Right? So here we have the labels. We have the large central vacuole. Remember, there's a permanent deposit of cell sap. We have chloroplasts. Right? These are chloroplasts. How you can actually tell the difference between the chloroplasts and the mitochondria is that the chloroplasts have, they look a little more striped. They have these little stacks of membranes called grana. These kind of look like green pancakes. And they have these thylakoid membranes in there too. And they look more striped, whereas the mitochondria looks more, it looks a little more porous, so it looks a little more folded inside. Also, chloroplasts are bigger. We have the cellulose cell wall, which is meant to resist any internal hydrostatic pressure, basically prevents the cell from bursting. What is not, well, you could kind of see it here. I don't know if you look at my cursor here, but you can see like little gaps in the cellulose cell wall. Those are called plasmodesmata. The singular for that is plasmodesma. You'll learn more about that in Unit 2, but basically this allows water to flow from cell to cell in something called the Simplast pathway, but you'll learn about that later on. Of course, this is the nucleus. So in this case here, you cannot see the nucleolus of this nucleus, right? You cannot see one central collection of ribosomal RNA. This is because this nucleus is probably in the midst of undergoing cell division. So it's not in the interphase stage, basically. The interphase or the prophase stage, I should say. The nucleolus has broken down, basically. We'll get more into that later. This come last year, differences between plant and animal cells. But I don't think it will come this year because it come last year. So I'll just go through it quickly. chloroplasts for animal cells. Of course, they don't have any chloroplasts or any plastids, anything containing any chlorophyll or anything like that. And in plant cells, they are present in photosynthetic cells. They're not present in all plant cells because you have some like some parasitic vines like Cassita filiformis, which is like an orange vine that doesn't have any chloroplasts. That's why it's a parasite that wrap around other plants to suck out the nutrients. All you know, I mean, like you try to suck out Johnny Depp's soul from you. So the cell wall is called the love vine, by the way. Animal cells don't have cell wall and plasmodesmata. Plant cells have them. So they usually have cellulose, pectin, or lignin. Animal cells are small temporary vacuoles, usually distributed in the cytoplasm. Plant cells have a large permanent central vacuole surrounded by a tonoplast. Animal cells have centrioles, plant cells don't. They have a different system for mitosis. Waste removal for animal cells, as said, lysosomes, the last funeral home of the cell. They clear up any dead organelles, any dead material, and move them out of the cell, ship them off. And plant cells, usually you have vacuoles to you have parts of the vacuole that deals with this. They don't have lysosomes. I'm not sure about all plant cells, but most of them don't. Animal cells usually have glycogen granules distributed in their cytoplasm to store any sugars and energy reserves. Starch grains are found in plant cells, which are found in amyloplasts. If you remember, starch consists of two polymers called amylose and amylopectin, and that makes up starch. The difference between starch and glycogen, by the way, glycogen is more highly branched. So there's a lot more energy reserves in them when they break the bonds. And animal cells, some of them have cilia and flagella. So for example, sperm cells have flagella. That's little tails. And some of them have tiny little hairs like goblet cells in your trachea. Cells in the fallopian tubes, those have cilia in them. Plant cells typically don't have cilia and flagella. Of course, bacterial cells would also have that. Let's see what they say in his comments here. Oh, shucks. Sorry about all this. Wait, hey, well, I'm watching the chat every once in a while now, but I was watching the table too, hey. Well, at least I was explaining something real simple, hey. If all you need to watch a table, hey, all you go on through, you know. All you go on through, you know. If all you need to watch a table, when I explain the difference between plant and animal cells, live, we live. You just get a little technical errors. All you feel like is do this as a job, or all you feel I have whole Instagram and thing dedicated to this, or let me see if I could disconnect this chat just now. Pop it out. Let me pop out the chat and just have it in the corner there. I should have done that in the first place. I'm going to pop out the chat there. Yeah, I'm doing one for Unit 2. That will come in a little while. Alright, so Prokaryotic says, again, I feel like this is common because it came last year. But just to go through it here, you have prokaryotic cells and you have eukaryotic cells. If you know the prefix, pro means first or beginning, and you could think of them as like the first types of cells that there was. It's very simple, and typically they have their own kingdom Monera, kingdom Prokaryota, and typically there's bacteria, cyanobacteria, or archaea. And these cells here, they don't have a nucleus. Instead, what they have is a nucleoid region. And they have plasmids, which is like circular DNA. The nucleoid region would have more linear DNA or more like strings of DNA. but not a nucleus. So they don't have a nuclear envelope, they don't have a nucleolus. If you had to label one, again, this are common because they come last year, but you never know. They have small 70S ribosomes. The S stands for Svedberg unit, and basically that relates to, is a kind of thing that for density, it tells you like how fast the ribosomes are sinking something. So they're small now, they're not as dense. That's all. That's what the S means. So they are small 70S ribosomes, and they don't have any membrane-bound organelles. So they don't have mitochondria, they don't have chloroplast, they don't have endoplasmic reticulum. They're simple, there's the first set of cells, right? So they don't have these things. They're now learning now. Well, not that they're now learning, but there's the OG cells. So they may need them things. They're surviving without them things. They're typically very small, usually 5 to 10 micrometers, and they have cell walls. Animal cells will have cell walls. Plant cells have cell walls. But these are cell walls. But these cell walls are usually made up of peptidoglycan or murin. So, example, bacteria would have that. Like penicillin does actually break down the cell walls of bacteria, and that's actually what's caused them to, to get destroyed. That type of antibiotic. That's usually mode of action of it. Right. So, eukaryotic cells, again, this are common. This are common, right? Because it come last year. But it might come for your multiple choice. We went through this. This is your typical animal and plant cell, your kingdom Animalia, kingdom Plantae, kingdom Fungi. And these here, they have a nucleus. They have DNA in long helical strands connected to histones. Of course, they have a nuclear envelope, nuclear pores, nucleolus, if it's visible. They have larger ribosomes, which is 80S. And plants have cellulose cell walls, whereas the bacteria have peptidoglycan cell walls. They are much larger, as much as 100 micrometers. If all you know anything, you know like the largest cell in the human body typically is the ovum. Well, not the ovum now, the secondary oocyte. And, well, when that fuses, it forms an ovum and then a zygote. The repeat last year for chem was a good thing. We're going through everything. Carry over the left side. I'll just make this a little bigger. No, but all you need to see organelle. We now went through this though. And some of them have flood. We now went through this. Rewind the video. The picture they just for flavor. The picture they just for don't take on the picture. Take on the text. The picture they for flavor. Alright. If all you will talk about things that will likely come for a year, is this. With my stupid self, that's why I predict. Because I think the last time that this come is, I think, 3 or 4 years ago. I think. And this usually comes in the long, the long part of the question. They usually have the, the little structured parts, and then you have the kind of long semi-essay part that worked a very depressing 12 and 15 marks. So this could come. Typically what they ask you to do is to define what is endosymbiosis. And if you know endo means in or inside, it better know. Okay, good. Endo means inside. And symbiosis relates to a relationship between organisms where at least one organism benefits. You just go, all you do that in C-sec, mutualism and parasitism and commensalism, you exactly go through all of that in Unit 2 again. But in this case here, what this theory states, and all you have to remember that a theory is something that is proven. This theory is not just I have a theory about the sinner. I have a theory that the Illuminati exists. Now, a theory in science is something that's proven. So this is proven. So what it says, right, is that long time ago, long time, prehistoric days or before prehistoric, when the earth was just like a primordial soup with no real large living organisms. It had nothing. All it had was Pangaea and Panthalassa, big land and big ocean. They say in that ocean, eventually some of them some of them molecules, some of them carbon and hydrogen and oxygen, they basically would have kind of come together and they would have formed a kind of blob. A blob that became one of the first living things. And that they would have resembled cytoplasm. That would have be like, it would have cytoplasm, it would have like some nucleic acids in it, and it would have resembled something living, nothing sentient, by the way, just something that you would call life. I don't know what's life, but if it are life, if you could define life, that was the first life. That set of molecules and carbon and nucleic acids and nitrogen and whatever, all of them slopping together into a kind of cytoplasmic soup. And this thing here now, what happened is that you had a bunch of them, and then you also had ones that resemble mitochondria. And you had some of them that resemble chloroplasts. Now, mind you, it's not the same as mitochondria and chloroplasts, right? We could call them early mitochondria and early chloroplasts. But basically, you had some of them that could respire. They had the ability to take in oxygen and form ATP and release carbon dioxide. You had some of them who's doing that, and they could swim around the ocean. You had some of them who could use sunlight and convert that solar energy into chemical energy. So you had some of them who could do that too. And eventually, this cytoplasmic blob, which you could call the host cell, what that do is that basically engulf the mitochondrion, and it engulf the chloroplast, and you could think of it like it get the powers from it. It get powers. So once it engulfed the mitochondrion, it was able to undergo respiration. Basically, it had the ability to break down sugars and use oxygen in order to form energy. When it absorbed the chloroplast, the early chloroplast, it was able to convert that sunlight into energy and ATP and sugars and things like that. So it formed a self-sustained system. So I don't know if I kind of went about that in a roundabout way, but basically it was like a vampire, if you know what I mean. It take this, it take that, suck it up, and they get the powers from it. Alright, that's now I feel like me and seeing the chat moving. Unless everybody just kind of stop talking. So I'm gonna pop this back in. And pop it, so I don't know how to use a computer. Oh, okay. I guess people were just listening. I wasn't sure. I thought something happened. All they know me, I don't do these things. This is literally not even a side hustle. This is just repaying society. As I say, because I know how hard it was for me. Try to make things a little easier for everybody. Okay. So just to go back through this again. And I say I'm going through this again because this might come. Know how to define endosymbiosis. That is when one organism could live within another organism and at least one of the organism benefits. Typically, it's a mutual relationship. An example of this would be rhizobium bacteria in leguminous root nodules, like in pea plants, so that they could fix nitrogen into the roots. That's like an endosymbiotic thing. But this, what we're talking about here, right, is how the first eukaryotic cell was formed, right, where you had this large cytoplasmic blob taking a mitochondrion, early mitochondrion could respire, early chloroplasts respire, photosynthesize, easy. The next part of this question that has come here is, well, how we know this. As is a theory is something not proven, right? So what's the evidence for it? You need to have evidence if you're claiming something. So typically they'll ask for 3 or 4 lines of evidence, right? And this is I have 5 here. Any 3 or 4 of these would work. So how mitochondria and chloroplasts, as we know them today, because we know mitochondria and chloroplasts only found in eukaryotic cells and not found in prokaryotic cells. So remember, they would have kind of evolved to become the forms that we have today. Those mitochondria and chloroplasts were once prokaryotic organisms that kind of remain there to today. It's kind of freaky in my opinion, but that's how the thing does work. So they each have their own kind of DNA. They have their own circular plasmid DNA, just like prokaryotes. They don't have a nucleus, of course they don't have a nucleus, but they have their own type of DNA. And they have their own ribosomes, and the ribosomes are actually 70S. I should put that here. The ribosomes are 70S. Similar in size to those in prokaryotes. In fact, the mitochondria and the chloroplasts are actually similar in size to many prokaryotes. If you was to put a mitochondrion side by side with E. coli bacteria, they actually might be the same length or very close. So how prokaryotes divide? They don't divide by mitosis, they divide by binary fission, which is like a shorter form of mitosis. But mitochondria and chloroplasts actually have their own version of binary fission, whereas prokaryotes do as well. Eukaryotic cells divide by mitosis, so it's where I have this thing that's operate differently inside this thing. Lastly, if you was to observe the inner membranes of the mitochondria and chloroplasts, they have a more prokaryotic structure, the way in which they are folded, while the outer ones have a more eukaryotic in structure. So I would say these first 3 or these first 4 is the easiest ones to memorize. I know about all here. But if you could know, define was endosymbiosis, explain the endosymbiotic theory. I'll go through past papers with them in a timing and give evidence, at least 3 or 4 lines of evidence. You're good to go. That would be like a good 12 to 15 marks right there. Right. Now what else could come is this. This plant root diagram and plant stem diagram. All you know, every year you're likely to draw some kind of diagram, and if it's not a diagram, it will be a if it's not illustration, is a plant diagram. Sorry. And the plant diagram, I think this year might be the dicot root, the dicot stem, or the anther. The anther for the in the flower. So we're getting to that timing. So how do we, I'll make sure everything here is showing good. Yeah. So we have 4 things labeled here, and I could zoom up on this a little bit. I go forget to zoom back. Actually, nah. Let me just make this a little bigger. And I'll go put it back the size. So here we have a light micrograph of a ranunculus root. And it's really only this part we have to pay attention to in the middle, and you could see it here magnified. All you know, when you're drawing a plant diagram, you need to label it, you need to title it, per annotations, per magnification. All of that is musts, right? All of that is your marks. You might think drawing the thing is your marks, but the magnification and the titling and thing, that's actually go for like half the marks right there. Especially if you have to show the magnification, the calculations. Anyway, so what we have here is this middle part here is where we have the vascular bundles. We have the xylem and the phloem. How I always remember this, by the way? Well, the thing looking like a croissant, it looking like an X. So the X part here, there's the xylem, and there's actually continuous tubes that transport water. The phloem, what you could say they're on the corners of the xylem. The huddled in the corners here. So these are the phloems here. 1, 2, 3, 4, and there's X here, there's the xylem. Easy way to remember it. Now surrounding it, you have the endodermis. So the endodermis is what surrounds this We also have we don't have the pericycle label here, but oh yeah, look at here. That's not. So we have the pericycle here too, which is right adjacent to the endodermis. Now, what the how you calculate? I'll go through magnification when we're doing some past papers. What the endodermis is basically is learn more about this in Unit 2, but they have these things called Casparian strips. Casparian strips are impermeable to water, right? And they're actually made up of a material called suberin. Suberin is actually the same material that you find in wine corks. And that, you know, it mostly impermeable. It has some porous spaces, but it's mostly impermeable to water. And it basically limits how much water could flow. It's kind of like a waxy cuticle, but it have like, you know, little barriers here. So you have some of them without the strips, some of them with the strips. The ones with the strips, they prevent water from flowing out or water from flowing in. The ones without the strips allow free flow of water. And that's basically your root. Now on the outside here, in case you have to draw this, there's not much to talk about. You have the epidermis, and you might have some root hairs branching out here. You have the cortex or the parenchyma, and the cortex has intracellular air spaces, not much to talk about, and this allows like what they call the apoplast and symplast pathways for water to flow. You don't have to know that, but basically it allows water to flow cell to cell through the cell walls or via the cytoplasm in the cells. So you remember the root hair cells absorbing the water, so they had to be able to flow through to reach the middle here. This is where the party happening. No, I'm not a teacher. So if you look at the annotations that I can make here, epidermis, root hairs allow water absorption. There's a large surface area to do that. The cortex moves water to the center of the root through cells or cell walls. The cortex has a lot of intracellular air spaces, as I said. The endodermis is a waterproof layer. It contains Casparian strips, as I said, that contain that impermeable substance. And vascular bundles contain xylem and phloem for water and transport of sucrose. Translocation and transpiration, basically. Easy. Easy. You just have to know how to draw the thing. Alright. So here we have of a plant stem, and to me this it looks like a dahlia stem. Yeah, I teach, but I'm not a teacher. I don't get paid for it, and I don't want any payment for it. As just do these exam things when they come close to exams. But for me to teach throughout the year, nah, have no time. Anyway, so you have a transverse section of a plant stem, and it has all the other stuff that is located in the root, except the Casparian strips and thing, but these are the ones that we have to take note of. Of course, you have the xylem, and let me make this a little bigger. Right? Yeah, I used to teach lessons there, but I don't teach again. So these are the vascular bundles. So each little thing here, what it looking at? It looking like a little like a tooth. Each one looking looking like a mouth with I don't know if all you see Dune part 1, and you had the sandworm. It looked like the mouth, you know, when you open up the mouth. You know, it coming for you. But of course that was much scarier. Well, there's no mouth. There's a transverse section of a dicot stem. But anyway, so the outermost part of this, right? This is the there's the phloem, there's the sclerenchyma, sorry, and all of the sclerenchyma, collenchyma, and parenchyma. You don't need to bother too much about that. What you want to focus mostly on is the vascular bundles, and the inner part here is the xylem. Then this middle part here is the phloem, and right in the middle is the cambium. You need to know much about the cambium. The cambium is just a site of cell division and cell specialization. That is just basically form other parts of the cell. You need to worry too much about that. But together, the three of these things form a vascular bundle, right? Now, there's usually a little band of tissue connecting these vascular bundles. It's called the medullary ray. It's not labeled here, but you need to label that. And on the outer part here, we have the epidermis, then we have the cortex, and then we have the inner endodermis here. Of course, epi means on top or outer, endo means below or more inner. So, of course, the epi one is more outer, the endo one is more inside. And inside here, we have the pith. The pith, in biology, pith just means innermost or inner layer. It's synonymous to medulla. The opposite of pith, by the way, is cortex. So usually when you hear about the pith in something, is usually the inner part. When you hear about the cortex, is the outer part. It could be anything. And the sclerenchyma and collenchyma, all these things here are just used for support. They are just support tissues. It's kind of like, how we have collagen to hold the skin together. The plants have these sclerenchyma, collenchyma, and parenchyma to hold their cells together and their tissues together. Nothing to memorize. All of them just used for mechanical support. Okay. And that's actually it for cell structure. So let me bounce right into enzymes. Because I don't think membranes not the end. It's just enzymes. Membranes itself not harder, but the osmosis part could be kind of tricky. So it's a good thing all you don't have it there. So, to me, enzymes pretty easy. A lot of it is the same things that you know from before. But you have a few things added in, like inhibitors, So first of all, let me get into what is an enzyme. I want to make sure everything. Right, so after we do this, after we go through all the work, we could do past paper questions. But I feel like past paper questions are really the thing all you want to go through so much now, because I don't know. Sometimes they repeat, sometimes they don't, who knows, but I'll show you all how to answer the questions and get all the marks. Anyway, so enzymes are biological catalysts. Yeah, amoeba sisters, they are very, very good at explaining these things. Yeah, shout out to them. Go on the YouTube channel if you wanna get into anything biology. I think chemistry too. Anyway, enzymes are biological catalysts. What they do is that they speed up reactions. So how they do that is that they turn reactants into products, but they allow that to occur at a much faster rate. There's usually a fixed amount of energy to turn these reactants into products. Well, if I had a credit all you do, hey, amoeba sisters, I credit it. Anyway, so where I was, reactants and products. There's a fixed amount of energy needed to turn certain reactants into products. You could think of it as climbing a hill. I need enough energy to climb this hill to allow this reaction to occur. Of course, it's not climbing a hill, it's using an analogy. What the enzyme does is that it lowers the amount of activation energy needed, so that that mountain or that hill becomes a lot less steep. So you require a lot less energy to climb that hill. So it could occur at a faster rate. Imagine if you're climbing this red one and this green one. If you climb the green one faster, that's what enzyme does. Module 1 is go more into proteins and things, but enzymes are globular proteins, which means that they're not formed of repeating sequences. They have very specific sequences of amino acid chains. And so, like collagen is formed from like repeating amino acids, like you have 3 or 4 that repeat continuously. But enzymes have a very specific sequence, and that specificity of the enzyme is what's coming very important when to answer these questions, is to address that. when to talk about its mode of action, of its mechanism of action. So enzymes have a tertiary structure, tertiary protein structure. Basically, they are polypeptide chains linked together with alpha helices and beta pleated sheets. Those are secondary structures, and those are usually connected by different types of bonds. You have disulfide bonds, you have hydrogen bonds, you have hydrophobic interactions, you have ionic bonds. You have all these different type of bonds that hold the enzyme together. But it's a very, very specific shape, and it's a three-dimensional shape. So even though this illustration here, you're seeing it as a two-dimensional shape, is really a 3D shape, and they actually look kind of weird. And let me see if I could show you the most popular enzyme is something called Rubisco. When I say most popular, I should say the one that is most found on Earth is one called Rubisco that is found in chloroplasts. And this enzyme is seen here, which has a quaternary structure, is actually made up of multiple tertiary structures. And don't worry, it's looking like nothing, we'll only see it there. But it have a four-parter here, and this it has some prosthetic groups on it. And you could see I have these twirly things. These are alpha helices, each one is an alpha helix. And you have the ones that look a little more flat, these are beta pleated sheets. This enzyme is Rubisco, and this is needed for non-cyclic photophosphorylation in the chloroplast. Basically, it allows electrons to pass through chloroplast protein complexes and allows that electron to move like a ping pong ball, pling, 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 and it initiates something called the Calvin cycle, which allows this compound called ribulose biphosphate to break down into glyceraldehyde 3-phosphate or G3P, which then goes through a cycle to basically form ATP, right? And this enzyme is found in all green plants. Don't worry about that too much. But yeah, without these enzymes, these reactions would not occur. And they are very, very specific in structure, like that. As I say, that structure show you there, that's not no random structure. That is highly, highly, highly specific to catalyze that reaction. If it even had like a few folds at outer place, or a few clefts, or a few depressions that there where it shouldn't be, that reaction wouldn't occur, I tell you, for photosynthesis. So they're very, very specific. Of course, what we know about enzymes mostly is in terms of catabolic enzymes, which are those that break down molecules. They break down large molecules into smaller ones. This would be amylase, pepsin, trypsin, lipases. Those would be catabolic. Anabolic ones are the ones that combine small molecules to form larger ones. This would be like renin, which is found in babies to curdle milk. So a good example of another popular enzyme is lactase. And if you know, lactase breaks down lactose. So as you get older, you develop less and less lactase. So you can't really eat ice cream without going to the toilet and things. Some people born without it, or born with very low amounts, and they say that they're lactose intolerant. Anyway, so how this works is that they contain a cleft called an active site, and the substrate is the molecule that would be the reactant. Whether you want to break it down or make it into a bigger molecule, the substrate is going to fit into this depression called the active site, and is going to bind to that to form an enzyme substrate complex, right? We'll get into that just now. But you see this active site here? What makes this special is that this is actually, how the amino acids have these residue groups, or what you call R groups. You have your leucine and glycine and arginine, all these different types. And those are some of them could be very simple. Some of them could have disulfide bridges, some of them could have aromatic rings, but basically each type of amino acid is different because of those R groups. This active site usually the R groups would be sticking out. Think of it like sticking out like little teeth here. And they would be very, very, very specific. Like this gap here would be very specifically shaped. What age group develops enzymes? You would have them since you're born, honestly. But as you get older, you tend to develop less and less of them. I'll give you an example. Like your livers produce an enzyme called catalase that does break down hydrogen peroxide into hydrogen and oxygen. They build up a hydrogen peroxide in your body, which is, it has free radicals in it. That actually is what turns your hair gray. But, when you're young, you tend to break down the hydrogen peroxide very easily. But as you get older, your liver produces less and less catalase. That's why your hair is turning gray. And people who don't produce much of it, their hair is turning gray before the time, So it depends on the person. Right. So the enzyme mode of action. So enzymes bind substrate molecules to form an enzyme substrate complex, and they convert them into products. So this substrate would fit into this enzyme here, and they would fit to form one whole complex called an enzyme substrate complex. It will then either break down or build up the substrate into something bigger or break it down into something smaller. Now in the end, the enzyme is break down of alcohol. I actually have to get into that just now when I talk about inhibitors. Anyway, so the enzyme itself doesn't change, it remains unaltered. The substrate will change because that would either build up with other molecules to make something bigger, or it will break down to make something smaller. But the enzyme remains unchanged during this reaction, right? It's unaltered. And this is what is called a lock and key mechanism. Lock and key meaning that the enzyme is like the... I've actually mixed this up, I think the enzyme is like the key, and the substrate is like the lock. Me, I know which one is lock and key now. It don't matter. But the enzyme is like the lock. No, the enzyme is like the key. The substrate is like the lock. Because I guess the key don't change, but the lock does not open. Yeah, that makes sense. So anyway, sometimes it's not like that. Sometimes what has happened is that the enzyme actually has to slightly change shape to accommodate the substrate. So this substrate here, you can see that it don't fit exactly into the active site. But what the enzyme could do is that it could actually fold a little bit. It could fold a little bit to accommodate the substrate. So it don't have to be exact like a lock and a key. So this is called an induced fit. The enzyme is the lock, but how that makes sense? How the enzyme is the lock? And the lock does open? And the key does open the lock? So and the enzyme would be like the key to allow the lock to open, the lock being the... I don't know. I know them thing, All I know is called it the lock and key mechanism, but in reality, that's not really how it does work. In reality, the thing does be more like induced fit. Because the enzyme is the lock, the substrate. Okay. It don't really make sense to me, but that is what the scientists and them say. Who's me? I just a man live streaming. Anyway, so I'm gonna show you here what I mean by this, right? Hold on, So hold on, I'm trying to find something here. Give me a little thing there. Give me a little thing. Okay, so if all you watch this here, Right. So there's the active site, right? And there's the substrate. Of course, they're going to fit into the active site, and that's going to make enzyme substrate complex. I said the active site is highly specific because it's usually the exposed part of the amino acids is the R groups that usually exposed here. The very specific R groups. There's a specific set of sequences. I said the keyword here is specific. Now sometimes they could fit like a lock and a key. But sometimes they could have something called an induced fit. So induced fit means that the enzyme will fold slightly, and the enzyme active site will actually change slightly to accommodate the substrate. Some people call this the hand in glove model, because when you put on a glove, the glove just kind of fall over your hand, and it might fit. It might fit slightly, change shape slightly to fit your hand. So that's called the induced fit model. Alright, we'll come back here. Well, it's the same thing in CSEC. I say all get away, all get away with easy thing. This year I find. But anyway, now we come into the parts that are a little more ugly. Right. Yeah, so the active site is complementary to the substrate. That's correct. And but as I say, it have two modes of action. You have the lock and key, in which they fit complementary, but you also have the induced fit, where the enzyme, it bend a little bit to accommodate the substrate. It'll give it a little bly, you know what I mean? It'll be like, nah man, you're not shaped exactly like, but that's alright, I'll give you a little bly. Anyhow, so let me look here. I say this is where the topic starts to get a little ugly. So rate of enzyme reaction. So this is what the graph would look like with the enzyme, and this is what it'll look like without the enzyme. So at A, so here we have on the y-axis the total amount of product formed, and this is the time that passing. So we know that as you convert substrates, you get more products. Kind of like if you're breaking down starch into maltose. Starch converts to maltose, so of course the more maltose that you have is the less starch, right? Because you already convert the starch into maltose. So look what happening here. Denaturing for real. Alright, so at A now, where you have a lot of substrate, let me say that you have plenty substrate, a lot of that is going to bind rapidly to the enzyme. It's going to fit into the active site, it's going to accommodate with induced fit. Well, basically you have a whole bunch of them binding to the enzymes here. A whole set of starch binding to amylase. So at B, so this is what happening here. I will let you imagine this. Imagine an enzyme is a toilet, and a substrate is somebody who wants to use the toilet, right? So if you only have a limited amount of enzymes, right, the substrates are going to occupy those enzymes. So what they're going to do is, the ones who some people had to wait to use the toilet. So if the enzyme is the toilet, and people who want to use the toilet is the substrates, well, you're going to have a line forming. Basically, all the toilets will get occupied at a point, right? Everybody had to do the business, and people had to line up to wait. So this is why the rate is going to slow down here at B, because at B, all the enzyme toilets occupied. So the substrates have to wait now for active site is supposed to be B, supposed to be free. As a result now, You're still getting product form in it, but the rate of product formation is going to decrease. Now at C, you'll notice here it start to plateau. This is because most of the substrate now has already been converted. You have very few substrate molecules left at this point. Basically, everybody done use the toilet, if you know what I mean. Everybody done do the business. So product formation rate is very low. It plateaus when there's no more substrate left. This reaction would most likely happen without the enzyme, by the way, but it would be a directly proportional, usually it would be a directly proportional relationship without the enzyme. Yeah, as Mark said, if it are no toilet, right? You have to get it out somehow. You might do it on the ground. You might go outside and do it. But it still had to happen. So without the enzyme, it just wouldn't happen at the optimal rate. But with the enzyme, you have all these nice toilets here. You have people going to use the toilet, and people don't use it. So by the time C reach you and you have a few people, them is people who's like, that line too long, man. Yeah. This is what it's called enzyme turnover. And you have an enzyme turnover number. That's correct. Jaden, why does the line run straight through? Well, it's a directly proportional relationship. So it will occur it will occur regardless, because those molecules have kinetic energy and they will break down regardless. It depends on the molecules, I should say. This graph here is dependent, but most of them would just break down just so it just wouldn't happen at the optimal rate. Of enzyme will do it. So both of them would eventually reach the same point. The reactions will occur. It's just sometimes the reaction will take so long to occur, you're dead by the time it occurs. In that case, it wouldn't occur. Let me give you an example, right? You need an enzyme called succinate dehydrogenase, right? Or let me just say dehydrogenase. You need that enzyme in order to allow respiration to occur. And without that enzyme, it acts as like it helps to turn certain sugar compounds into others. Without that enzyme, it will still work. It will still happen. But by the time it does actually work, that dehydrogenase enzyme works. Sorry, I'm mixing up myself. Without the dehydrogenase enzyme, that process will still happen. But by the time it happening, you're probably on the verge of death. So obviously that's not what you want to happen, right? Same thing with food. If you drink milk and you don't have any lactase in your body will still try to break down some of the lactose. It obviously cannot break down all. So eventually what will happen is that you will just end up getting diarrhea before it could happen. It's not the optimal thing, because you could be writing a CAPE Unit 1 paper 2 and you drink a whole set of milk and thing and you have diarrhea. It's not the optimal thing. So typically without the enzyme it will still occur, but you might go into a coma by the time all the product form, or you might get diarrhea. It depends. But with the enzyme, it occurs at a fast enough rate where you can actually break down a lot of that substrate in time. Anyway, toilets. Okay. So what happens if you was to increase substrate concentration? Well, using my analogy with the toilets here, right? It's kind of like having the same amount of toilets, but having more people who want to use the toilet. Well, what's gonna happen? Well, the rate, the reaction rate the enzymes are gonna get occupied very quickly, and you're gonna have long lines for people waiting to use the enzyme toilet, for substrate people waiting to use the enzyme toilet. So the reaction rate is going to slow down because you have extra substrate that has to be broken down, but you only have the same amount of enzymes. You only have the same amount of toilets. So what will happen is that it will take a much longer time to reach its maximum velocity, or its Vmax value. So at that point there, that might actually be lower than usually is, because you only have a limited number of enzymes. So what you can actually do is you can just increase the number of enzymes to counteract that increasing amount of substrate, because you have more toilets for more people. It's like if you're having a fet, plenty people, you need plenty toilets. So here again, we have increasing enzyme increasing enzyme concentration. You have the initial rate of reaction, and let me just tell you the difference here. The initial rate of reaction is only accounting for like the very beginning, right? Because as the reaction occurs, those enzymes are gonna get occupied and gonna plateau. Just imagine this as like the beginning of the graphs that you see just now, right? So using our next example here, if you think of enzymes as, you go to the bank, you have tellers in the bank. Tellers deal with customers. You have first citizens is only have like one teller sometimes with a whole set of customers. That's like having one enzyme to deal with a whole set of substrates that ain't gonna work. So how you could make that line shorter, how you could make that line move faster, how you could convert all that substrates to products, how you could ensure that all these customers in the bank get taken care of. You need more tellers. Just like you need more toilets. You need more tellers in the world, you need more toilets in the world. So what that does is that adding more enzymes means that the substrates can then occupy those enzymes, just like lock and key mechanism or induced fit, and is actually a directly proportional relationship. So for example, if you have 0.2% enzyme concentration, right? This refers to enzyme concentration. And I was to double that, I would actually get double the rate of initial reaction. If I was to double the 0.4, if I was to double the enzyme concentration here, it would be twice as fast. Is the same thing. Imagine you have one toilet for 10 people. But imagine like each people, I guess each person just take the same amount of time in the toilet. That's not true, but yeah. But now you put two toilets. Well, the line could be half as short, half as long. You put 4 toilets. You put 10 toilets for 10 people. You understand? The thing directly proportional. You have 10 people and 10 toilets. Watch now. Don't start now. This thing here. Hey. Right. Enzyme activity versus temperature. This C-secting. All you should know this already. These molecules substrate molecules have internal energy, right? And internal energy is kind of like energy stored within the molecule that allows it to move. Basically kinetic energy. So this kinetic energy is in the substrate molecules, and they zipping around kind of like gas molecules in a little container. They're just bouncing off the place. Eventually now they're going to bounce and they're going to go into an active site, right? Basically, if you have more kinetic energy, it's more likely that those molecules are going to bind with the enzyme, those substrate molecules. Low energy causes slow amount of movement. So here the enzyme rate is very low, below the optimum temperature. The rate of reaction would increase if you were to increase the temperature. Now, this will occur until you have an optimum temperature. This is the point where the molecules could move fast enough, but it also doesn't have too much energy to break the bonds between the proteins. So remember I said here that enzymes have tertiary structures. And those are typically held together by ionic bonds, hydrogen bonds. They have hydrophobic interactions, and some of them might have disulfide bonds, right? But let me focus here on hydrogen bonds. Hydrogen bonds real weak. Unless you have real plenty of them, like in cellulose, they're very, very weak. And you just get a little ching and it break, right? A little bit of heat and it go on through. So this is why even let me tell you how weak this thing is. When you go out in the hot sun and you have ultraviolet radiation from the sun, no, the sun hot, but that UV radiation could rip through your nucleic acids and could possibly give you a melanoma because you have hydrogen bonds and little phosphodiester bonds holding the nucleic acids together. So they're weak, right? They're weak. And in the enzymes, you have plenty of them, but they're weak. So having a little bit of heat there, they will say above 40 degrees Celsius in this case here, you notice that there's a steep decline past this point. This here is when the enzyme is starting to denature, undergo denaturation. This is because this tertiary structure, which is highly specific, because it's folded in such a way that the active site has a very specific shape, the enzyme itself has a very specific shape, so that it could be complementary to the substrate. Once that begins to warp a little bit, that active site begins to change, and some of these bonds begin to break, it unravels to form its secondary structures instead. I don't know, but that don't make sense, or what's going on? Hopefully that makes sense. I mean, it's kind of the same thing in CSEC. It just have this added thing with the protein structures here. That's all you have to explain in addition to that. In terms of pH though, in terms of pH, you have to remember that, as I said, ionic bonds holds this together. Yeah? Ionic bonds hold these together. And ionic bonds, the word ionic of course comes from ion. And pH, which kind of stands for like potential hydrogen ions. You can think of it like that. But basically what pH is a suppression of hydrogen ions. So the lower the pH, basically the more acidic something is the higher the number of hydrogen ions. So differences in pH, what it could interact with the ionic bonds that holds the enzyme together, and it could end up breaking those bonds, right? So to come back here, just to explain it to once more. In terms of temperature, it is the heat energy that breaks the hydrogen bonds because they're very weak. When it breaks the hydrogen bonds, the tertiary structure unfolds into its secondary structures and primary structures, which is just polypeptide chains. At this point here, it doesn't really have the same shape. It doesn't have the same specific shape or the same shape or active site. So it's no longer complementary to the substrate. So of course it don't work. The thing don't work. pH, on the other hand, breaks ionic bonds. If there's a shift in pH that the enzyme is not really accustomed to or cannot accommodate. The two common ones that we have here is pepsin and trypsin. And of course, the tips of these graphs here are the optimum parts. Pepsin has an optimum pH of about between 2 and 3 because this is usually found in the stomach where there's loads of hydrochloric acid. And trypsin, which is usually found in the duodenum, in the small intestine. So this is slightly alkaline. So typically the optimum pH for this would be about between 8 and 9, around 9. So it's not different. It's just you have to understand, you just have to note which bonds break for which. Temperature, hydrogen bonds, pH, ionic bonds. Easy. Okay. So, enzyme inhibitors. What's going on here? Let me give all the example of an enzyme inhibitor first, right? And then I will explain what it is. So me know how much all it is drink real rum and get like real drunk. Well, they shouldn't do that, eh? But if you just drink real rum and you eat and you drink no water, you're liable to wake up the next day with a hangover, right? And if you know anything about rum, if you know anything about ethanol, you know that is broken down by your liver, your hepatocytes, that's your liver cells. And your liver has an enzyme in it called ethanol dehydrogenase. Basically what that does is that it breaks down the ethanol into acetyl coenzyme A, which is a kind of harmless thing, right? So without that enzyme, without that ethanol dehydrogenase, you cannot break down the alcohol. So if you just drink real plenty and you don't have that enzyme, you're gonna get a hangover like immediately. So when you get old, you produce less and less of that. And as a result, you can't really, as people they say, hold a liquor, right? So it actually have a type of, not medicine, I would say, it have a substance you could take called ant abuse. I'm going to type it here. It have a substance called ant abuse. Basically anti-abuse. And what it does is that it inhibits that enzyme, ethanol dehydrogenase, from breaking down alcohol. So what has happened is that when you drink a whole set of rum and you take your anti-abuse, that ethanol dehydrogenase don't work because this compound here, anti-abuse, sometimes it is a little pill or sometimes it's like a little dose you could take. And what is happening here is that stops ethanol dehydrogenase from working, so you just get a hangover like immediately. So I don't even know if all you know that exists, but it's for people who have like alcohol addiction and they want to deter themselves from drinking. What they do is that they take doses of this anti-abuse. So the moment 5 minutes after they drink a shot of puncheon or they drink a beer, they get headache, they get sick, all kind of thing, because that alcohol actually not breaking down in the body. It will eventually break down because I say eventually things that happen. It's just the enzyme not working because the enzyme is actually getting inhibited. So the anti-abuse, right? This compound is the inhibitor. The enzyme is the ethanol dehydrogenase. There's the enzyme that's break down alcohol, and the substrate is the alcohol. So we boil anti-abuse here, fitting into the active site and preventing the ethanol from binding to it. As a result, now the ethanol can't break down because it's kind of like somebody take your seat, be like, hey, that's my seat now. Or you move from here now. So somebody is asking me about reversible and non-reversible. Well, think of reversible like this. Reversible is like, imagine you have an inhibitor. You have somebody sit down in your seat and you be like, hey boy, that is my seat. And the man say, alright, I'll go on. That's reversible. And somebody sit down in his seat and you say, hey boy, that's my seat. And the man say, but you going from here now? That's irreversible. Right? That's irreversible. Because the inhibitor might get permanently bound to the active site. They're moving from the seat basically. You have to go and find a next seat. You have to go and find a next enzyme. And well, if all the enzymes occupied, hard luck there. You have to do without. The reaction had to occur without the enzyme, you might dead. But anyway, this is a good example of an inhibitor because the enzyme ethanol dehydrogenase, which is supposed to break down ethanol, cannot break down the ethanol when people take the dose of this. Instead, it lodges into the active site. I actually think this is an irreversible reaction because you need to form more enzymes. So for a good few days, people can't drink. You would form more, but for a good few days, they can't drink. Giovanni, most competitive inhibition is reversible, but not all. Not all. Most of it, but not all. Anyhow, so they prevent or limit the binding of substrates to active sites or enzymes. Basically, the competitive inhibitor is similarly shaped or similarly complementary and bind to the active site instead. And this is competitive inhibition. Now, what could also happen is that they may bind to another site. This is called an allosteric site, and that would disrupt the shape of the enzyme. That's called non-competitive inhibition. So I'm going to show you that there. Right, so this is how inhibitors work. See, the inhibitor, which is seen as like the pink or the purple molecule here, pink, purple, whatever, violet, is similarly shaped to the substrate. And what could happen here is that this would bind, basically takes up the seat, it binds to the active site, and limits or prevents the substrate from binding. most competitive inhibitors are reversible, which means that they can unbind as easily as they bind. Some of them are irreversible. So I'll give you an example. One that's irreversible. It's a one called, if I can remember it, malathion. Yeah, malathion, which is used in insecticides. And what it does is, I think it's classed as an organophosphate. And what it does is that it binds to receptors in the insects that produce a neurotransmitter called acetylcholine. And to break down acetylcholine, that neurotransmitter, you need an enzyme called acetylcholine esterase. And malathion, which is found in insecticides, it binds to that. Those enzymes that break down the neurotransmitters so they can be reused. As a result now, basically the insect's nervous system gets totally destroyed. That's why when you spray bop and you spray fish and you spray death on some insects, not all, because some of them, they do that just so. You see, they start to beat up, they start to. That's because literally you mash up the nervous system. The chemic after that. That neurotransmitter, that enzyme has breakdown that go on, because so that's actually a competitive one that is irreversible. So some of them reversible, some of them irreversible. Anyhow, so competitive, similarly shaped inhibitor, substrate carbine as a result. Non-competitive is this. Alright, so I imagine non-competitive like this, right? Imagine this is the girl you like. Right, fellas? Imagine this is the girl you like. This is you. There's you. The substrate is you. There's the girl you like. And the green is some fella named Kyle, right? And look at what happened here. You think that you and the girl compatible. But Kyle come now. Kyle come and mess up the whole scene. Because you actually have two sites on the enzyme. You have the active site, where the substrate would fit into, and you have other depressions in the enzyme called allosteric sites. The term allo means other, so it literally just means other sites on the enzyme. So Kyle now, look at what happened here. Kyle come and bind to the girl. And look, the whole active site change because the whole enzyme now underwent induced fit. And now you can't get through. You can't get through again. You cannot get through. A good example of a non-competitive inhibitor is cyanide. Cyanide binds to cells and binds to enzymes and prevents a lot of respiratory compounds from converting to other compounds. Basically, that is a non-competitive inhibitor. Most of them are irreversible, which means that they bind there and they stay there, and the substrates cannot break down into products because the active site is no more. It have no way you could actually do this. Who is this? Akisha, you telling me to skip and go to module 3, Gil? Just come back later now. I doing the thing how I doing to do. We doing module 3, what happened? Go and run around and come back. Right. So, coming back here. So if you look here now, competitive and non-competitive inhibition. So same thing I explained there. In competitive inhibition, the inhibitor temporarily binds to the active site. Oh yeah, Anaya, to answer your question, you should know at least 2 examples of each. I would say like competitive, you could use the insecticide one, and you could use the ant-abuse one. The non-competitive, that's the one with the allosteric sites. You could use cyanide is a good example. And what's our next example? Next example is it have something called succinate dehydrogenase. I don't think you want to use that one though. But sure, if you Google it, you're gonna find one. You also have other examples like digitalis, which is used to treat heart rhythm problems. Alpha amanitin, I think that is a non-competitive one. That's a poison that's found in some mushrooms. Heavy metals, yeah. You Google the thing now. Learn 2 examples. Learn to be. I don't think you have to explain the examples. You just have to be able to list them. I think. But it's good. The ant-abuse one is a real good one, I find. Anyway, so non-competitive, the inhibitor binds to the allosteric site. And this can be irreversible if it permanently alters the 3D shape of the enzyme. So we boy Kyle here permanently chain up a girl so you can't get through again. You can't get through. So they married and think you can't get through, just go and find a next enzyme and done. So it permanently alters the 3D shape of the enzyme and active site, stopping product formation altogether. The competitive one is just it could be somebody sit down in your seat, be like, one boy, get up from a seat, alright, so and you go and sit down. But if it's irreversible, that man not moving. Get up from a seat now, but get out of here. That's irreversible. Right? Easy. And using with toilet analogy again, think a competitive is just somebody they go on in the toilet and they're taking real long. You'd be like, one boy, hey, they're taking real long in the toilet, but I still have to. But once that person done with the toilet is still a toilet. You could still go and use the toilet. Non-competitive is like a man going in and mash up the whole toilet. So now nobody could use the toilet. I'm sure that has happened in some other schools. Some person they be nah man, or they spoil the thing for everybody, boy. A non-competitive inhibition, especially if you go and mash up the toilet, nobody could use it, or the real wicked boy. But anyway, but that's non-competitive inhibition, especially if it's irreversible, because at this point here, all your substrate cannot convert to products. That enzyme basically useless, just like this mash up toilet. Nairobi, right? Can non-competitive inhibition and induced fit hypothesis happen? Nairobi, they typically go hand in hand, because what happens is that the allosteric, the non-competitive inhibitor binds to the allosteric site, and as a result, the enzyme undergoes an induced fit to accommodate Kyle with non-competitive inhibitor. And as a result now, the actual active site changes shape and cannot accommodate the substrate. Yeah, boy, hacker specials are mash up the toilet, boy. Mash up the thing, it can't work again. Anyhow. So, I think this is the last part of enzymes here. And they love to bring this. They love to bring this graph. If I had to predict what will come in enzymes, it will be this graph. Maybe all you had to draw this graph. Maybe you had to sketch it. Maybe you just had to explain it. But this graph, they love this graph. They're in love with it. And enzyme specificity, whatever that word is, specificness, and mode of action, that's the things you want to look out for. Non-competitive and competitive inhibitions. Anyway, so let me look at the graph here. Let me think. Alright, give me. I'll type out some examples for it. Just give me a second. Alright. So let me go through this here. Malathion insecticides is organophosphates. If you see that word, it refers to insecticides. Alright, so you go through these graphs here. So this here describes the initial rate of reaction, which in the beginning, we're looking at the beginning here first, right? And here we have the substrate concentration. Cram, jam, we cramming and jamming all the info and we had before tomorrow. That's why I call cram jam. Anyhow, so look at what's happening when we have no inhibitor present. You see that the initial rate of reaction is much higher than the other two. With a competitive, remember not all, most competitive inhibitors are reversible, which means that you just tell a man to get up from his able to get up from a seat and you could go and sit down in the seat. Substrate, see a inhibitor, a inhibitor, competitive inhibitor, get up from my enzyme now. Alright, yeah boy, alright, and you sit down there. So, non-competitive inhibitors, most of them, not all of them, but most of them, because they go hand in hand with the induced fit hypothesis, most of them are irreversible. They're like we boy Kyle. And they fit into the enzyme, changes the whole three-dimensional shape of the enzyme. It loses its specific shape, so it can't work again. You mash up the toilet. So with this now, a lot of these enzymes, depends on the amount of inhibitors, by the way. But these here, you'll see that the non-competitive inhibitors doesn't reach the maximum velocity, the Vmax value of having no inhibitor and having a competitive inhibitor. This is because a lot of the toilets get destroyed. With the non-competitive inhibitor, we have less toilets, we have less enzymes that viable. Now, the difference here with the competitive inhibitor and no inhibitor, you'll see that with no inhibitor, it has a much faster rate of reaction, a typical reaction rate, and it reaches a Vmax value faster than with a competitive inhibitor. Competitive inhibitor, it also accomplishes the same Vmax value, just takes a longer time to get there, because they constantly have to tell people, hey, get up from a seat to get up from a seat. And let me type here. Right, remember this here is just a generic graph. This graph will differ depending on which actual inhibitors and enzymes you're talking about. This is just a generic graph. So, how do I... Weird. Somebody said in but say, or anyhow, the main thing to realize here is that with non-competitive inhibition, it doesn't reach the same max velocity of reaction as the other two. Easy. And as enzymes, so that's module 1. Okay. I'll go along with what I think is Akisha or Akila was saying. Which one we should go, module 2 or module 3? Or tell me. I feel we should just go in order, you know. Ball, tell me. Anaya, the Vmax value is the point where you have the max rate of reaction. So that would be basically on the ends here, where it plateaus. So you see that the Vmax value is a lot lower for the non-competitive. Okay, module 3. As in 3 and 2 and 3 and 2 and 3 and 2. Alright. We are one man spamming today. I'll do 2. Ole who are 3, Ole could sit down and Ole could wait. Let me do the thing in order now. Let me do the thing in order before we chain up ourselves. The reason why I want to do 3 last, so I could take my time now. I want to take my time with 3. Because it's basically the whole module we have to learn. Right, so we're going with module 2, sorry. Yeah. No, you see, if I do module 3 now, I might rush it. And because I would want to wrap up now. So I rather take my time and do module 3 last, especially if you do module 3 last, it will be closer to your exam. So you might remember it a little more. Because the module 2 part's easy, you might as well just get it done with and done. So all you who good with module 2, or they go and take a run and come back, or they could come back for module 3, depends. Anyhow. Okay. Did we take a 5 minutes break? Did we take a 5 minutes break before we start module 2? So we'll come back just now.
Hello, good day, sir. We are doing a survey. We want to know why you just come on YouTube and what's spewing.
Because after I
What's spewing live stream?
and I kiss my babes, it sends shivers up her spine.
Ooh, it can't get better than that.
Because it freshens your breath.
Brother, tell me why you just what's spewing.
Because no matter where I go, no matter what I do, I must always do my best.
Because it's refreshing.
For me, it's the only thing that can relieve my upset stomach.
I's a smoker.
That's spewing live stream.
It does help freshen my breath and take away all the smokiness.
It is very good. It is very useful.
It keeps my breath fresh. Full stop.
Need to keep it fresh all the time.
So you're from Tobago?
Yes, I'm from Tobago. Yes, I love spewing. Original.
Spewing.
God, you're real spit in there. All right, stop.
Jesus, exam thing.
Hey,
Who's that, me?
yes you. Fix that face now. What happened?
Another red man. Not the red man I'm looking for.
The rain is way harder and the wind is...
We wish all our candidates the very best in making the final preparation for your
This is what you're reading?
Jesus, exam thing.
Just right.
In Trinidad, this going on?
That is real ginger beer.
You want to tell the secret to being successful at CSAC?
Yeah, tell me now.
Balancing a top one sideways.
Really? That is your big secret?
Yes. This man real good.
Real ginger beer.
I went from alright to real good.
The letter of the day is J. J is for
Jesus exempting.
Plenty people dying, but we can't do nothing about that. If it's dead, they go dead. Correct. Correct. Correct.
B is for bias, or body bias.
And the answer.
Or body bias.
Alright, I ain't that.
Correct.
Wait, you wanna help me study later?
Study. Study the Guardian. She said practice that.
Correct.
Jesus exempting.
You will. All right. I think that was 5 minutes. So let me start back there. Hey. What I just see there was the prep for this module. Overproduction of offspring, which helps with natural selection. We'll come back full circle just now, man. Alright. So, cell division and variation. Yeah, man, that was 5 minutes. That was 2 minutes. 2 minutes, 5 minutes, who cares? Let me start. We have all this work to get through, and then the past papers too. We have to make sure and get it once tomorrow, no? Imagine not getting a one, no? Imagine not getting a one. Imagine not getting a one in biology. You have to be real stupid. You have to like not do your labs or nothing. No, I'm not doing that. All your copy no labs, and then you must get an A for labs, A for IA, and imagine not getting a one. There we go. Alright, so cell division. Most of the cell division we're going to be looking at in terms of the individual stages is going to be in animal cells. Because remember, in plant cells, they have a slightly different way of doing it because they don't have centrioles. It occurs more or less the same way. The stages are the same, but it occurs without centrioles. Instead, it has a microtubule organization center, or MTOC. So it still has the microtubules and all kind of thing in it, but most of the examples we're going to look at here are with animals. And we're going to look at some electromicrographs that involve plants, typically onion root or allium, right? So first, let me get into mitosis and meiosis. Basics. So we know mitosis, let me talk about the daughter cells. There's two daughter cells, right? Two daughter cells are formed here. And these two daughter cells are genetically identical, if you don't have any mutations, to the parent cell. So first, the first stage of cell division, we can say cell division has two main parts. 95% of cell division is the interphase. Interphase is basically where you're going to have DNA replication. And the amount of DNA or genetic material in the nucleus is going to double up, right, during the interphase. At this point here, you can't actually see the chromosomes though, but 95% of the cell cycle is doubling up of DNA replication, right? Mitosis is the other 5% of the cell cycle. Mitosis is where after the DNA has already replicated, the cell splits into two identical daughter cells. There's only one cell division, two daughter cells are formed, and they have a diploid or diploid chromosome number. Basically what that means is that they have two sets of chromosomes, or 2N, right? And so this is the replication phase. So basically this is doubling up the amount of DNA. So imagine that we have a certain amount of DNA in this parent cell. It doubles, sounding good? Anyway, so it doubles, and then it splits into two identical cells. So here we'd have, like say in a human, we would have 46 chromosomes. That doubles up into 92 chromosomes, which then splits into 46 and 46. Don't worry about it too much, because chromosomes and chromatids, that is give you a little chain up. But that's mitosis. And mitosis is used for asexual reproduction, is used for cell growth, cell repair. All those things are involved with mitosis. Meiosis, on the other hand, is only used to form gametes. It's a very specific process that is used to form either sperm cells, a spermatozoa, or secondary oocytes. Secondary oocytes are what we know as egg cells. What C-Sec lied to you and tell you that the female gamete is an ovum, but really the female gamete is a secondary oocyte. That's module 3 thing. But meiosis is used to produce those two cells in humans. For in a plant, it would produce actually multiple types of cells. It would produce microsporangial cells, it would produce the megasporangial cells, it would produce synergids, antipodal cells, polar nuclei. All of those there would actually be haploid as well, which means that they only have one set of chromosomes. We'll get into that when we reach module 3. So, what the hell is this way? Where them bots come from? Hold on, Mahindra, seeing you posting plenty, use a moderator. Akisha, use a moderator. I don't know, or let's just get rid of that. We're trying to keep this as PG or PG-13 as, as possible. Anyhow, so meiosis was the difference. So we can see the difference here is you have cell replication, you have DNA replication, and we have two sets of divisions. There's the first set of division, there's the second set of divisions. So the first time it divides, that's called meiosis 1. The second time it divides, it calls meiosis 2. So what we have here is 4 daughter cells, and each of these is varied. 2 cell divisions, 4 daughter cells, each one is genetically varied, and each one has half the chromosome number. So they will say that we start off with a parent cell of 46 chromosomes. That's going to double up, and eventually you're going to get 23 chromosomes, 23, 23, and 23, basically half the amount. Yeah, and that's the basic differences between it. So what is the importance of mitosis? As I said, this is something that could come. They like to bring this question here, what is the importance of it? And sometimes they had to go up to 3 different things. So the first thing here is asexual reproduction, which is you only need one parent, no gametes are involved, no sex cells involved, no fertilization. You have one parent cell and it produces identical offspring rapidly. So example would be with not bacteria, because that would be binary fission, but protists such as amoeba, or slightly more complex organisms such as hydra, or even in vegetative propagation with budding and fragmentation and things like that, basically, like with bryophyllum leaves, where it forms little leaflets. You don't have any fertilization of gametes, but you have one parent and it could have multiple offspring as a result. This is highly recommended, or basically its survival rate is very high if this is in an environment that is not changing, if it's not prone to environmental changes or changes in populations where you could have a invasive species come in, or some kind of pathogen come in, some kind of disease. So if it's done in a controlled environment, it you could get through real good with it. Next we have growth. So for example, if you have, let me say, phototropism in a plant, the plant have to grow towards sunlight, and you have auxins, which is a type of hormone that encourages or facilitates cell growth, that bends the shoots towards light. That there would be mitosis, because there will be one cell divided over and over and over. Can a water bear be an example of an amoeba? A water bear would be, I don't think specifically amoeba, but it would be a type of protist. It will be in kingdom Protista. You can look it up and make sure. Assuming the water bear talking about is a tardigrade and not actually a bear in the water. Anyway, so our next example with growth would be, next thing in module 3 would be when the zygote is formed from a fertilized egg. With cuttings, cuttings would be an example, Anissa, because you would have adventitious roots and things growing out of a cutting like sugarcane. Yeah, coming back here, the zygote would have to divide continuously to form a ball of cells called a morula, and eventually into a blastocyst. So, and eventually an embryo. So, whereas meiosis would form the gametes, mitosis would encourage the growth of one cell into 2, and 2 into 4, and 4 into 8, and 8 into 16, and so on. Also, tissue repair. So let's say that you get a cut on your skin. Mitosis would help they would use amino acids, and it would help to repair the collagen and the skin tissue and thing, that has been damaged. So growth and tissue repair, you should know this. Immunity, you'll learn more about this in Unit 2, but when you have a foreign invader entering the body, they would typically have a compound attached to them called an antigen, and your body will detect that antigen, right? Think of the antigen like a burglar alarm in your body, and it's going to signal a security system, basically immune system, and it's going to produce B lymphocytes. B lymphocytes produce antibodies. They call them B lymphocytes because they mature in the bone marrow. But let me say that you get COVID. God forbid you get COVID, right? And you need to get rid of that COVID pathogen from your body. You can't just have 10 white blood cells to do that. So what your white blood cells have to do is that they have to undergo mitosis, and they have to multiply, so that they can have enough to get rid of the pathogen before the pathogen can proliferate enough so it doesn't destroy your whole body, because they need to produce a lot of antibodies from those B cells. Cell turnover would be tissue repair. So yeah, that would fall under that. That's a good example. You know that every day is dead a little bit. Your hair does fall out, your skin does shed. So yeah, mitosis has helped to replace those lost tissues. Right. So let me get into the stages of mitosis. Okay, so first we have interphase. Interphase is not a stage of mitosis. I just kind of fit it in here. Interphase is, as I said, 95% of the cell cycle. And interphase is where you have DNA replication. I don't think I need to go into DNA replication because that's part of nucleic acids. But DNA replication is basically you have one whole strand of DNA, and you have a number of enzymes such as DNA polymerase, yeah, different DNA polymerases, that's going to make a complementary strand to that old strand of DNA. And you're going to have an old strand and a new strand. So basically what you're going to have here is DNA doubling up. And that occurs for 95% of the cell's life. So during interphase, the nucleolus is actually still intact. The nuclear envelope is still there. And of course, you have the nuclear pores and things like that. The chromatin is still there, is not condensed into, it's not condensed into chromosomes yet. So it's just like little stringy threads. And that there would be interphase. These are the centrioles here. Prophase. Okay, so yeah, what you want to remember is IPMAT, right? This is how let me show you a video depicting it first. And let me show you how I just remember it. Well, how I used to remember it is make up all kind of stupidness in my head. The more stupid it is, the more likely I'm able to remember it. Alright, so let me show you something. So let me start with interphase. Interphase is the easy one because, as I say, that's just it had the nucleolus. There's the chromatin inside. Right? So there's the nucleus, there's the chromatin, there's the nucleolus, there's the centrioles, and these are the microtubules that are going to branch out here. Interphase is just the point of DNA replication. It's just doubling up the amount of genetic material. And there's the plasma membrane on the outside here, and of course within you have the mitochondria. Okay. So, all in all, the stages is interphase, prophase, metaphase, anaphase, telophase, right? So let me tell you how I used to remember this. And I'll come back to the video just now. Alright. And I want you to keep this in mind as we go in through it, right? I want you to keep this in mind as we go in through it. Alright. Prophase, metaphase, anaphase, telophase. I only have one stage in it. It don't have two stages like prophase, one like meiosis, right? Alright. So I'm gonna tell all you a story here. And once you remember the story, you're gonna remember what does happen in each one. So interphase is where you still have your chromatin, thread and thing. You still have your nucleolus. Now, imagine now, all you finish CAPE, all you finish CXC, what you probably going to do. I don't know if all you have in it, but typically you go to a graduation. And you know in America, there's called the graduation, the prom. So think prom, right? Basically, what you're doing is that you're sewing them chromatin threads into a nice chromosome suit or dress. So basically the chromatin threads, they would condense and they would form a chromosome, they would form chromosomes and chromatids. Basically the things that we are accustomed to seeing where they look like, you know, now, more condensed, I don't know how to describe it, but little wiggly tigglies. So they look like that. So think prom. You're sewing chromatin threads into a nice chromosome suit or a dress, right? And let me look at what else is happening in prophase. The nucleolus disappears, nuclear membrane begins to break down. So I want you to think of this. You're broken out for prom. You have no barriers anymore. This is because the nuclear envelope break down. Right? So, that's prophase. Prophase, yes. So metaphase now, prophase here will also be pairing up in some circumstances, right? You'll be pairing up in some circumstances, but we'll get more into that just now. Metaphase, you are meeting in the middle, right? This is where you go into the prom and you're meeting people now. So look at what is happening here at metaphase. In metaphase, everybody kind of meet up in the middle here. Nuclear membrane has been fully broken down, right? The chromosomes are lying at the equator of the cell. That's the middle of the cell. And the centrioles on the polar ends. And what you have now is these threads that are holding on to what you call the kinetochores of the chromatids. Don't worry about that too much. But basically, they're meeting up at this imaginary line that you call the metaphase plate. So you're meeting in the middle, and in some circumstances, it will be paired up. Centrioles on the end, on the poles. Anaphase. You know, you go to any FET, you go to any grads and thing, you might get a little fight. So now, somebody whine on somebody else, something happen, somebody mass, somebody clacks, who knows? What happening is, now you have angry, you have some anger, you have anger, and anaphase anger, right? Everybody meet up in the middle, everybody jam up together, all kind of thing could happen. One man touch a next man, girl, all kind of thing could happen, right? So now what has happened now is that the chromatids are being pulled apart. So it'd be like, we angry, hey, all they get away from each other, we're breaking up this fight. That's anaphase. So anaphase now is like, after everybody meet up in the middle, it had a little thing, a little bacchanal that happened, now they getting pulled apart. Anaphase, anger, away, yeah. Pulled apart, away, wherever, however you want to think about it. Telophase now is where everybody starts splitting up into different groups. So you know, after the grads now, everybody vexed, everybody angry, and what you do? You go on your telephone, you go on your phone, and at this point here now, we have two separate groups. And these two separate groups, what's going to happen is that those are going to form into daughter cells. And let me look at what's happening in telophase here. Telophase, you're telling everybody on the phone what happened. But telophase, this is what's happening here. So let's look again at metaphase and anaphase, right? Metaphase, nuclear membranes, this is where you're meeting your friends. Everybody meeting in the middle. Chromosomes are lying at the equator. Centrioles on the polar ends of the cell. Anaphase, this is where you're angry. You're angry. Everybody getting pulled away. Anaphase. A for away, A for angry, A for apart. So the chromosomes break at their centromeres. The sister chromatids move to the poles of the cell because somebody breaking up the fight. Everything moving apart now. So that's anaphase. You'll notice now we still don't have a nuclear envelope. We still don't have a nucleus. Everything just being pulled apart. We have these microtubules and these centrioles. Telophase is kind of like, well, everybody split up into two groups. Everybody going home. So the chromatids at the poles unwind to form chromatin threads once again. So they no longer have the chromosomal structures. They come back into threads. Spindle fibers break down. The nuclear membrane and the nucleolus reform. So now we have two cells, but they're still connected. They still have this little cleavage furrow that's connecting them. Right? So what's going to happen now is that the cytoplasm now has to divide. And the cytoplasm would divide and split the cell in two. This is what is called cytokinesis. The word cytokinesis literally means the movement of cytoplasm. Kinesis like kinetic. So two identical daughter cells are formed from one parent cell. Right? So once again, let me look at the video. I'll explain this as we go in. So this interphase into prophase. Remember, you're sewing together your chromatin threads to form a nice chromosome suit and chromosome dress for prom. There's prophase. These are your chromosomes. Okay, so let me explain something about chromosomes here that might be a little confusing at first. If we count on this particular diagram, 1, 2, 3, 4, 5, 6. We have 6 chromosomes. But each chromosome here that you're seeing has 2 chromatids. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 chromatids. So we have 6 chromosomes, but 12 chromatids. But when we count in chromosomes, we don't really count chromatids. We just count the units. So in this case here, we don't say it has 12 chromosomes. We say it just has 1, 2, 3, 4, 5, 6. You just count the full units. So it has 6 chromosomes. Don't worry about the number of chromatids really. Now remember, the only reason it has 2 chromatids is because the DNA would have replicated. Right? The DNA replicated and it doubled up. That's why. Now, so these are your sister chromatids and you have a centromere holding them together. This is a nucleolus. So a nucleolus, you notice, has broken down during prophase and the nuclear envelope has broken down because they're ready to brock out, ready to go prom. So look at what's happening here now. So remember, this is prophase and now we're moving into metaphase. So metaphase, we're going to have centrioles moving to the poles of the cell and they're going to grow microtubules. So look at what's going on during metaphase now. Remember, metaphase, this is where you're meeting your friends, everybody meeting in the middle. M for meet, M for middle. They're meeting at the equator of the cell. Right. So they're going to meet there. And the chromatids are going to attach to the, what you call the kinetics or the centromeres. And the tubules are going to start to pull apart these sister chromatids. This is anaphase when they start to pull apart. Because something happened and now they're angry with each other, so they're moving apart. Remember, anaphase, apart, away, angry. Okay. Now there's the kind of confusing thing that could happen here, right? If we count the number of chromatids, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now remember, say count each unit. So in anaphase, what the weird thing is, anaphase, this actually has 12, we say 12 chromosomes in it. But it won't stay like that for long because remember, 6 gonna go in one cell and 6 gonna go in the next one. So we end up, when we start with 6, we ended up with 6. So only for the briefest of moments, you're gonna have 12 chromosomes. Because they don't count each chromatid, they count each unit. So they're gonna move apart. And all this is still anaphase. And eventually now telophase, like telephone. The term telo or tele just means like another location, another place, like teleport, for example. Telephone, television, it comes from another place. It just means like a distance away. So telophase in this case means that both of them go into distances away. And what happens here is that the nuclear, I'll go back to just now. I'll go back to the abbreviation thing just now. So during telophase, it forms 2 complete units now. The chromosomes or the chromatids form chromatin threads. The nuclear envelope reforms and the nucleolus reforms. Cytokinesis, which just means movement of cytoplasm, is where this little cleavage furrow is split. The cytoplasm basically separates from it and you get two distinct daughter cells. In plants is a little different because plants don't have centrioles, but it's basically the same thing happens. It's just you have a different thing that forms the microtubules. We don't have to get into that. Okay, so coming back to this. Yeah, we're doing a little crunch here. So once you remember this story, the whole thing is easy to remember. Say it's a real stupid story, but as I say, the dumber the story, the easier it is to remember sometimes. So once again, prophase, prom, sewing your chromatin threads into a nice chromosome suit, nuclear envelope begins to break down, nucleolus begins to break down because they're ready to get out there. Metaphase, those chromatids meet in the middle, those chromosomes meet in the middle. Anaphase, angry, chromatids pulled apart, microtubules form, connect to the centrioles, pull them apart. Telophase. Oh! It should be back now. But don't worry, I wasn't really showing anything. I was just talking. Yeah. So easy as that. That is what it is. Take a screenshot. I don't really think you need to take a screenshot, but take a screenshot, I guess. Right. Right. So IPMAT interphase, which is not really a stage of mitosis, but it's a stage of cell growth. Now, Arif Khan, I think, is some man who does just make these videos. Who knows? Or probably just upload them. Man, make sure to have your name there, though. Alright. Oh, let's look at this diagram here. This is a electron micrograph. Not electron micrograph, there's a light micrograph of root tip squash of onion. And this is typically what it is bring here. And see if you can figure out which one is interphase, prophase, metaphase, anaphase, and telophase. And I'll just give you like a 20 seconds, 30 seconds to see if you can figure it out. If you can name it going in anti-clockwise order. Like which one is this, which one is this, which one is this in anti-clockwise order. So this is something that they tend to bring a lot. They'll bring a light micrograph and they might ask you to draw some of the cells and annotate or to label the phases. Kyle, Pierre, don't put interphase as one of these stages. But they still just group it together. Here you know what? Put it, but just indicate that it occurs before mitosis. All you see the thing, all you write CSEC already. All you know the thing, write as much as you could. You never know how the mark schemes could look. Write as much as you could, and just be clear with what you're writing and don't contradict yourself. So if they ask for it, just say Interphase is the stage before mitosis where DNA replication occurs. Simple as that. Nucleolus is still present. No chromosomes yet, only chromatin threads. Anyhow, let me go through each one here now. Honestly, if you put pro-metaphase, it's still correct, but you don't need to specify like early metaphase, early anaphase, and like if you put that, I mean, you're good to notice it, but they will mark it correct regardless if you put metaphase or pro-metaphase. They usually be a little thing with it. Anyway, so this is it here, and I'm gonna explain how you'll know each one. Let me go in order here again. So interphase, how you know is interphase? It still have a nucleolus, right? Clearly, and you can still see the nuclear envelope and thing here. Prophase, this would actually be like very early prophase because you could still kind of see the nucleolus, but you're seeing the chromosomal material here. And the chromosomal material is what's the giveaway here. You're seeing still some of the nuclear membrane here. So this is like early prophase. Metaphase, this one is actually very easy to identify. This and anaphase are easy because metaphase, you'll see the chromatids or the chromosomes are in the equator of the cell, the center. And anaphase is a next easy one to identify because this would actually be kind of early anaphase. And this one might be a later anaphase, but you don't need to specify. But anaphase, you can see them being pulled apart here because A for anaphase, A for anger, A for apart, A for away. M for metaphase, M for meeting, M for middle. And then we have telophase. So how you can actually tell the difference between telophase and anaphase is you could see the reforming, although this is very early telophase in this example. This might look like telophase here, down here. This here. But it's like a very early telophase because you could see the nuclear envelope forming here. This might be a better one because you can actually still see the cleavage furrow. Right. So a term that you might come across here is homologous chromosomes. Well, you know homo means same. So homologous just means the chromosomes are similar in structure and they're similar in terms of their gene loci. What a gene locus is basically, think of like each chromosome has like little points where you could insert the genes. Like it have position A, position B, position C, position D. And what would happen there is that gene A would be in locus A, gene B would be in locus B, and so on, right? So the locus is just like a position where you'd find a gene or allele. So homologous chromosomes have the same number of gene loci. Think of loci as location, right? And these are said to be homologous because they're similar in shape and they have the same number of loci for the same type of gene. So let me say locus 1 is for eye color. Then that would be, you might have a brown allele in one and a blue allele in that one. So position, it basically just means position or location. So humans have 22 pairs of homologous chromosomes, and we can see them here, which means that they're similar in shape and they have the same number of locations for those alleles called loci. But the 23rd pair is not homologous. So these are your sex chromosomes, which means that they can be an X chromosome or Y chromosome. So of course, females would have 2 X's and males would have an X and a Y. I feel I is the main moderator here. Me know where them butts coming from. Anyhow. We should know the difference between diploid and haploid by now. Diploid have the full set of chromosomes. In terms of humans, there are 46. These are found in somatic cells, and basically these means that non-sex cells. So they could be skin cells, it could be stomach lining cells, epithelial cells in the lungs, epithelial cells in the capillaries. Basically anywhere in the body that is not a sex cell would be a somatic cell. Haploid cells have half the number of chromosomes, so that would be 23 in humans. Of course, as I said, these are found in gametes. They would be in humans, they would be spermatozoa or secondary oocytes. Okay. I think so far a lot of this is the same as CSEC. I don't think anything has really been different, because I think you just still learn about prophase and metaphase. I don't think you have to, but I mean, you still be taught it regardless. You still should know it regardless. Meiosis here is where it starts to get a little tricky, a little different, but more or less you could think of meiosis as mitosis occurring twice, except in the first phase of meiosis is where you get a little bit of crossing over happening, where you get some exchange of genetic material. Otherwise you could think of it as mitosis occurring twice. The only difference is meiosis, it have a little jamming up in meiosis 1, where you exchange things. You know, you go to the prom, you give this one a number, them give you the number, you know, a little piece, get exchange. I don't know. So these butts and them related, boy. Okay, so what's different here now? Let me look at something for meiosis here. We're gonna explain it step by step. Right. Okay. So let me explain what's happening here. So let me look at meiosis 1. Let me start with prophase 1. Right. Prophase 1, almost the same thing. You have a chromatin threads, they condense to form chromosomes. Almost the same thing. But look at what's happening here in prophase 1. We get a little joining up. Right? We get a little joining up here. So you see these points here where the loci cross, these little sections are the chromosomes. Each one of these is called a chiasma. The plural of chiasma is called chiasmata. When they are arranged like this, we call them a tetrad of chromatids or a bivalent. So in meiosis, they arrange to form bivalents. So they literally pair up. And what's going to happen is that they're going to exchange DNA at these loci where they join up. So for example, if you can see my cursor, this first set here, this is going to exchange DNA, or they're going to cross over DNA at here and here. So what's going to happen is that they're not going to be the same structure after, because they're going to swap DNA. That is also going to happen at here and here and here, where the intertwining of the loci occurs. So this is called synapsis. And remember these parts here, where they join, these are called chiasmata. Right, so this joining up, this is called synapsis. And they're going to swap DNA at those points. So this is called recombination or crossing over. So the actual positioning of the chromatids is called synapses, but when they actually cross over, that's called recombination. And what you have here now is during prophase 1, you see this red chromatid now has a little bit of blue on it, and this blue chromatid has a little bit of red on it. To answer your question, Akisha, the nuclear envelope begins to disappear during prophase 1, yeah. And it should, by the end of prophase 1, it should be totally gone. So basically when metaphase starting, everything should be out in the open. Metaphase 1, I should say. Okay. So here we have the nucleus. Remember, this now is metaphase 1, where we have these microtubules forming, right? And they're going to do the same thing that they do before. They're going to pull them apart. So they're going to pull these chromosomes apart. But remember, each one now is going to have a little bit of exchange DNA. So, see in here? So this is what leads to genetic variation in the gametes. Right. Sorry, so this is anaphase 1, but again, pulled apart. Microtubules, centrioles pulling them apart. So you see it's almost the same thing as mitosis. You just have this, the difference is that they pair up. You have synapses, and then you have recombination. Now, meiosis 2, real easy. Meiosis 2 have no difference from mitosis. Meiosis 2 is just mitosis. So remember the main difference here is that meiosis 1, let me rewind, have this joining up at the loci here, forming of the chiasmata, and the swapping of DNA or recombination. That's the main difference. That's the notable thing. And remember, this happens during prophase 1. Everything else there is kind of the same. I guess the other difference is during anaphase 1, because they don't actually split up the chromatids. They just pull them apart after crossing over DNA. But I say the one thing you have to remember is during prophase 1, they hug up so much that little bits of each other come out on each other. Meiosis is that kind of effect. Alright, and I'll go into independent assortment just now. Right. So I say this one here, meiosis 2 is just mitosis. The difference being that each one is not identical because they would have underwent synapsis and recombination, crossing over. Same thing, metaphase 2, same thing like mitosis, anaphase 2, same thing like mitosis. And telophase 2, same thing like mitosis. Okay, and then I have cytokinesis again. And remember, in this particular illustration that we see here, remember we started off with 4 chromosomes. Now we have 2 chromosomes, but each one undergoes variation. So we end up with 2 chromosomes per daughter cell and 4 daughter cells. Now, for CAPE, you don't have to know it step by step for meiosis. You really just have to know the differences. I don't think I ever see anything where they ask for the step by step of meiosis. They might just ask like prophase 1, Right. So coming back here, let me go in the chat and answer some questions. At what stage does the cell become haploid in meiosis? Okay, so the stage that it becomes haploid, that will be, I believe that is anaphase 1. So during anaphase 1, I think that's where they will split and they will form 2. Somebody correct me if I'm wrong, but I think that in anaphase 1 is where they become haploid. And anaphase 2, they get pulled apart again. Random segregation occurs during anaphase. Okay, so I'll get into what independent assortment and thing means just now. But firstly, we continue. Alright, look at here. So meiosis 2's events are very similar in mitosis. Meiosis 1, there's really only 2 things you have to take note of. Instead of the chromosomes aligning independently in the equator, they pair up into bivalents and they form these chiasmata, and the chromosome number is halved during anaphase 1. And that's really all you need to remember for now. Do you have to know the different pattern? No, you don't have to know the different patterns. So if you're just supposed to recognize when it happened. Like if you're looking at an illustration or a diagram, then you would see that, okay, the loci cross here, they cross here. You don't have to know all the different patterns in which, because that too could be random. And because they can connect at different loci. So you don't need to memorize those things. Okay, so why does genetic variation occur with meiosis? This is what you should be concentrating on. As I say, you don't need to go through it step by step, really. You just have to be able to differentiate between mitosis and meiosis. Mostly what they might ask is like, what happens at prophase 1, which is just the crossing over. But what you should know is how it contributes to genetic variation. This is your bulk part of when you're talking about variation. So there's 3 things you want to look at. There's crossing over, of course, which we just talked about, where they cross loci at the chiasmata and they swap DNA. This is recombination. Then you have independent assortment. So when the chromosomes pair up, one comes from the mother, of course, one comes from the father, because the gametes have haploid number of chromosomes, which means that they only have one of each, right? So it's not in pairs. So when they pair up now, these pairs are independent of each other, and this results in a massive number of combinations. So let me see if I can explain this a little better here. So let me say that in patterns of inheritance, right? Let me say that we have a genotype that is capital A, common A, capital B, common B, right? Now remember, this could have, in the gametes, this could have 4 different configurations. This could have capital A, capital B, capital A, common B, common A, capital B, and common A, common B. First, outer, inner, last, basically of each. So you could have 4 different, and this is just for one. This is just for one set of DNA, right? For a dihybrid, a set of alleles, you would have numerous amounts of this. So let me say that we have a next set of alleles that is capital D, common D, capital E, common E. Let me just use that as an example, and this would be the combinations of alleles. So I know this might be a little confusing for some of you, but I'll go through it again just now. Now, remember here that this is assorted independently. So a person could inherit this and this, for example. Right? They don't have to inherit them in any particular order. So you could have this and this, for example, because each one is independent of each other. So it had different locations where they swap. You have different ways in which they are sorted. And then you have many of this. You have genes that determine eye color, genes that determine skin color. You have alleles that determine blood type, alleles that determine whether or not you're going to get hemophilia or sickle cell anemia, or alleles that determine whether or not you're going to get that cystic fibrosis transport protein, or whether or not you're going to have a widow's peak or lobe, or, detached or attached L. You have a whole set of them. So this is just like some random letter, but you have a whole set. And each of these is independent of each other. So the chances of getting two are the same, right, is astronomical. Like it astronomically low, I should say. It very, very unlikely that you're gonna get two of the same, of course, unless it's an identical twin, but they would have had the same DNA split twice. But that's a different story. So independent assortment ensures that there's variation, because just because somebody get capital A, capital B, that doesn't mean that they're going to get capital D, capital E. They could get, if they get capital A, capital B, they could get any one of these, and they could get any one of the next set, or any one of the next set. So it's kind of like, you're picking random, you're picking from a whole set of random boxes. You pull out one from here, one from here, one from here. I know probably want to say something like madness, but that's what it is. Each one is independent. Each locus is independent, basically. Each one is sorted randomly. And anyway, the last one here is random fertilization, which means that each gamete contains a different random mix of DNA, but not just that. You have to remember now that men, especially, every ejaculation, them just pelt out 200 million spermatozoa mature. So that's it depends on the man, but that's possibly per day. So, or even several times, it depends on the man. And other 200 million spermatozoa is only one that could fertilize an egg. So you have 200 million different configurations, and there's only one that could get through. I mean, that would be you, but it would just be one. So fertilization, unless you're specifically doing like in vitro fertilization, I mean, if you're just copulating, the thing going to be random. It's just whoever swimming the fastest go reach. So that too ensures genetic variation. It ensures that the set of chromosomes in each one is different, and the zygote is different every time. That was a lot to say. Independent assortment. Independent assortment would be during it'll be during metaphase. But metaphase segueing into anaphase. But basically think of them like when they pair up. Any two could pair up. So it could happen at, different points. But you don't need to pinpoint which ones. Still good to know. Anyway, so what are the advantages of genetic variation? This too is the next big one. It's easy, honestly. It falls into natural selection, and I think that they might segue natural selection into variation, because these two topics go very, very hand in hand. Anyhow, so the first thing here is slowing disease spread. Well, think of it like this. If you have every member of the species being the same, which means they have the same genetic makeup, that means that they are affected by the pathogens in the same way. I'll give you like a good example. If all you ever drink banana soft drink, like from Solo or something, right? You notice it don't really taste like banana. But that's because the bananas, the extracts that was used to form that soft drink, the flavor from it, is from a banana that's mostly long extinct. It's either a pathogen that a fungus, I think it's a fungus, that caused a disease called Panama disease, and that they wiped out a certain banana population. The type of bananas that we mostly eat now is the Cavendish banana. We have some forms of the Gros Michel banana and things like that. But the type to form the banana soft drink, that gone. Mostly gone, I think. But I think the flavor extract is still there. And people just get so accustomed to the flavor extract. It don't taste like banana, but it once had a banana that probably tastes like that. But it get wiped out because usually when you're eating bananas, the seeds are not viable. It have seeds, but they're not viable. You can't take a banana seed and plant it. We talk about domesticated bananas, by the way, not wild ones. Wild ones have seeds. So they usually reproduce by cuttings. And cuttings, as we know, is a form of asexual reproduction, which means that is the same DNA being passed from parent to offspring, parent to offspring, parent to offspring. The only thing that could really make it different is a little environmental variation and if there's a mutation. Otherwise, because of the limited gene pool, and because of the lack of variety in the immune system, that they're having something asexual reproduce and having no genetic variation accelerates the spread of a pathogen. So of course, having genetic variation slows the spread of diseases, especially communicable ones, of course. So we have that. Adaptation to environmental changes. So if we have, okay, let me give all the example, right? I think all you would know that chickens come from dinosaurs. Yeah, I think all you would know that chickens come from dinosaurs. The dinosaurs still alive, they're just chickens today. Chickens and other birds. And those would have been the dinosaurs that survived. And we know that the dinosaurs was wiped out due to extreme changes in climate. From very hot, very cold, the Ice Age. Most of them didn't survive. And what is happening here now is that we actually have a type of, let me see if I could find the video on it. We have a type of organism that's still, a type of bird that still exhibits a lot of the same dinosaur characteristics. I'm looking to see if I could find it here. It's called a Archaeopteryx. But basically, let's know. I'm looking to see if I could find it, and I'll show you. A Hoatzin Archaeopteryx, that's the name. And this thing here, they have them in some parts of Guyana. So they actually have some of the pterodactyl characteristics. They still retained it. They only talk about KFC, only eating the descendants of dinosaurs. Anyway, so this is a Hoatzin Archaeopteryx. That's the name of the bird. And you could see here, I'm not sure if you could see it. They have the little claws on their wings. They have little claws on their wings. Birds usually don't have that. So most of them was wiped out. But because you had a high variation in the bird-like dinosaurs, some of them is able to survive to today. That's the ones that you're seeing. So they have these little claws here. And there's actually kind of living proof of a kind of distinct link between the prehistoric and the modern day. You don't have much organisms, at least much large organisms, aside from some trilobites and nautiloids and microorganisms that exhibit that. But this is one of the large organisms, large avian ones that depict that. Anyway, so coming back to what we're talking about here. adaptation to environmental changes. So we have these things called selective pressures. A selective pressure could be a shift in temperature. It's something that occurs outside of the norm. It could be a shift in temperature. It could be a storm. It could be an invasive species. It could be even the introduction of a certain pathogen. Well, last year KFC gave the employees 2 aloo pie and a red soft drink. As far as I know, I eat KFC since I'm not supporting that. I am supporting KFC. Stupid business practices. Anyway, so if you have a wide gene pool between these organisms, they are able to actually overcome these environmental changes. So if you have, for example, a bunch of mutts, and you have birds that could only detect white mutts, and you have a wide variety of white mutts, gray mutts, dark mutts. The gray ones and the dark ones more likely to survive and reproduce. If you have some organisms that, like the camel, for example, the camel has the adaptation of storing water in the fatty tissue in its humps. You actually had camels that didn't have that fatty tissue, and those would have died out. And the ones that did, they was able to actually survive and reproduce and kind of carry on that characteristic. So, you know, they could adapt to environmental changes. I'll give you one more example. I know I'm kind of hanging on this a little bit. So if you look here, this is a legged snake. They're very, very rare, right? A legged snake. And because once upon a time, snakes had legs, kind of like salamanders, and eventually environmental pressures like being able to fit into smaller spaces, having speed to dodge predators, being able to burrow, some of them even being able to swim, the legs was actually a disadvantage. So there would have been a mutation which would have caused some snakes to be born without legs, and those populations are what you widely find today. But there are still some snakes that have legs. Typically is due to something called atavism, which means is the atavism is like when old DNA gets reactivated. It's weird. It's like some humans could grow tails, some humans could grow fur. You could look up this stuff yourself, but you know it's weird. It's like old ancient DNA gets reactivated. It's not like a good science fiction thing, but it's real. It does happen. Anyhow, let me come out of this before I should have give all a trigger warning for that. Anyway, the last thing here is limiting overcompetition. Great example is the Galapagos finches, where each finch had a different type of beak, or they did this in CSEC, and each finch had a different type of beak, where the ancestors, now because of genetic variation, you had some of them who had thick beaks, some of them who had longer beaks, some of them who had thinner beaks, and they was able to actually adapt to different diets. The ones with thicker beaks they was able to feast on fruits, they was able to crack nuts. The ones with longer beaks was able to feed on nectar and was able to actually put their beaks in lugs so that they could catch insects. As a result now, not everybody had to be eating the same thing. Because each bird was slightly different, that would have limited overcompetition. What is also was also interesting about this is the beak size actually changes their mating call a little bit. So thick beak birds, their mating call might attract other thick beak birds. Long beak, thinner beak birds might attract other ones of that. So that actually makes it easier for them to separate and group up into ecological niches. So yeah, those are the three advantages of genetic variation that you should note: slowing disease spread, adaptation to environmental changes, limiting overcompetition. Right. So I said this part here also goes into mutations. We have gene mutations and we have chromosomal mutations. Typically, when they ask about gene mutations, they ask about sickle cell anemia, but they asked about that last year. So you never know. I'm still going through everything just in case. But a mutation typically occurs during DNA replication, because DNA replication is where, remember, you have an old strand of DNA. DNA polymerase is like a construction worker that is building a new strand of DNA. And that's called semi-conservative replication, where you get a complementary new strand of DNA. That is not always perfect. What could happen is like little errors could end up happening. So for example, you could end up with a cytosine. Remember, you have 4 nitrogenous bases in DNA. You have A, T, C, and G: adenine, thymine, cytosine, and guanine. I am going through all of that because that's on the nucleic acids. But sometimes it could end up with one being substituted for another. Cytosine, instead of a cytosine, you have a thymine. So this is a random copying error. So what is interesting about this is eventually this will happen to you. If the older you get, there's a higher chance of getting more and more mutations in your body because it's bound to happen. It's an error, it's random, but it will happen at a point. And hopefully when it does happen, it's benign. Usually you might just get a mole, or you might get a little skin tag or something like that, but sometimes it could develop into cancer. Or it could develop into, something a little more vile, but typically most of the mutations that occur are benign, which means that they're not harmful. So you have 3 types. You have substitution mutations or point mutations, deletions, and insertions. And they're pretty self-explanatory. Yeah, how I like to remember it is, apple in tree, car in garage. Adenine to thymine, cytosine to guanine, and then RNA is adenine to uracil. Anyhow, so substitution or point mutation is where a single base is replaced by another. So for example, if we was to swap out this cytosine for this thymine. A deletion is where we could get an entire base pair or single base being deleted. And addition is where we get an entire base pair or a single base being added. Deletion and addition are 2 very dangerous types of mutations. Substitution could lead to sickle cell anemia, but it typically doesn't really amount to anything harmful, typically. So let me explain why. Deletion and addition are considered frame shift mutations. So you need a little bit of knowledge on transcription and translation to understand what this means, right? But if we just go back to this example here, if you look at this substitution one, we could see that the top here, T, we have 6 letters. This will be read in trees. So this would form 2 triplet codons, right? And the first codon is going to be TGG. And the second codon is going to be CAG. And just to explain, I might have to backtrack a little bit, just to explain this a little bit. What is happening here right, is that DNA is what determines your sequence of genes. DNA, or deoxyribonucleic acid, right, that would eventually get translated into a protein. DNA is like, think of it as like a blueprint. what you want to do is you want to write out that blueprint because DNA is not mobile. You need a single-stranded molecule called mRNA or messenger RNA to actually carry that information. So mRNA is like a messenger boy. And that's going to take the instructions to ribosomes. So the DNA is writing the instructions on messenger RNA. And then the messenger RNA, because it is mobile, could actually float out of the nucleus. It could actually leave. And it could go to the ribosomes and the cytoplasm. And that is going to take the instructions to the proteins to build... Sorry, you're going to take the instructions to the ribosomes to build a protein. Now, if the instructions wrong, you're going to end up building a whole different protein structure. Because what is happening here is that when you are reading... Yes, no, eh? When you have DNA and that builds a mRNA strand, each codon, right? Each codon translates into a different amino acid. So for example, AUG, this is the instruction for the ribosome to build a amino acid called methionine. You don't need to memorize that. GUG, this is the instruction to build an amino acid called valine. UUG, these three letters, this codon is used to build leucine, this amino acid leucine, and AGC is used to build this amino acid called serine. If one of these letters change, it is likely, not always, but it is likely that it will build an entirely different amino acid. So, for example, with sickle cell anemia, we'll come back here. With sickle cell anemia, you end up with... Who say they are coming? This is part of it though. You don't understand transcription, but you still have to understand to see how the mutation come in. Yes, that's correct, Sophia. Right. So let me say here for red blood cells, right? You have your normal red blood cells, and these have a protein in it called glutamic acid, GLU. Sickle cells, on the other hand, have valine instead of glutamic acid. And this is because the DNA has GAG for a normal cell, but this adenine gets swapped out for a thymine in the sickle cell one. Eventually now, this will build an entirely different amino acid. This one amino acid causes this biconcave red blood cell to become sickle shaped. Because remember, if it builds a different protein, the bonds are going to fold it differently. The polypeptide sequence are going to be different. They are going to fold in different places. You're going to have different locations for the hydrogen bonds. You're going to have different locations for the ionic bonds, different hydrophobic interactions. Typically, it doesn't make a difference. Typically, sometimes you just get a little extra melanin and you get a mole. But sometimes, I say, it could be very, very harmful, like sickle cell anemia. So anyhow, come in here now. So point mutations or substitution mutations could be just as harmful. Because what's going to happen here is that a certain amino acid is going to build from TGG, that's a codon. And a certain amino acid will build from CAG. But a totally different amino acid is going to build from TAG. And that there would change that one amino acid, but it wouldn't change the others along the way. Because the amount of pairs didn't change, it's just one letter swap. Okay, so deletion and addition are considered frame shift mutations. Bases are transcribed and translated in triplet codons, as 3-letter codons. So let me look at the deletion here. If we look at the normal DNA sequence, this is the codons here, TGG, and CAG. This would build a certain amino acid, let me say amino acid A. This would build amino acid B. But look at if we get this base being deleted here. What we're going to end up getting is GGC instead of TGG. And this is going to build a totally different amino acid, let me say C. And then we're going to get AG something, instead of CAG, we're going to get AG something. And that's going to build a total. So basically the whole protein going to come out raw. It's kind of like if you imagine you're baking a cake and somebody leave out instruction number 1 and you start with instruction number 2. The cake going to come out probably totally different from how you expect. So that's a deletion. It's kind of like a substitution mutation. It's kind of like, hey, you're baking a cake, add salt instead of sugar. The whole cake might come out, not right. A deletion might be like start with instruction 2 instead of this one, and all the rest of the instructions would change as a result. So the thing is right. You rip out a page from the cookbook, it's not right. The cake not gonna come out, they're not gonna come out the same. You're gonna end up with a totally different product. Same with addition. If you was to add a base pair here, remember TGG makes amino acid 1, CAG makes amino acid 2. But if we add a base pair, it's not TGG again, it's ATG. ATG is gonna make a different amino acid 6, for example. Then we have, instead of CAG, we have GCA. So this is what is called a frame shift mutation because it actually changes the reading frame. So whereas we're reading TGG here, we actually reading it from GGC here. And that's going to form a totally different protein. Anyway, so these are deletions and additions of frame shift mutations. Yeah, I don't think sickle cell anemia will come because they bring our whole question with that last year. But we're just going through them. Okay, chromosomal mutations are not changes in the genes themselves, but changes in either the cell's chromosome number or the chromosomal structure. So this happens during meiosis. It could be in either step of meiosis, but it typically occurs during anaphase, the anaphase parts, where the chromosomes are unevenly pulled apart. You could have what is called a monosomy or trisomy. A monosomy is basically where you have one less chromosome. A trisomy is when you have one extra. So Down syndrome is where it gets pulled apart and you end up with one extra chromosome in the gamete. So basically you might have a gamete with 22 chromosomes and you might have a gamete with 24. One extra and one less. So Down syndrome is an example of a non-disjunction, let's call it. Then you have Klinefelter syndrome. Again, this is a trisomy. You end up with 2 X chromosomes and a Y. And we have Turner syndrome. This is a monosomy, which means we end up with one less. So this is where you end up with just one X chromosome in women. Of course, it can't be a man because the man would have an X and a Y. Each one here, let me see if I could get let me see if I get an image. If I could remember accurately, Klinefelter, I think is the one where you have no neck. It had one of them where you have no neck, you know. Because it interferes with the chromosomes. I wonder if it's Turner. Not one of them. It could be neither. Yeah, I can't remember. But one of them does interfere with the formation of the neck. I think it's Turner. But anyway, me I go post them thing or you can look it up yourself. It are different types. As for the different ways in which this could occur, I'm going to make this a little bigger. We have inversions, right? Where we get the pieces of the chromatid like, okay, I don't know if you all could see it here, but you could see the gene loci label here, A, B, C, D, E. What happens is that piece of the chromatid breaks off and it inverts. So instead of A, B, C, D, E, we have A, B, E, D, C, right? It's inverted out. That's why it's called an inversion. We have a translocation mutation where we have an exchange of segments instead, where they're non-homologous. We don't need to give examples of this. We have a deletion where a part is removed, of course, a fragment is removed. And then we have duplications where one randomly divides or doubles up. You have others as well. You have insertion, I think. There's others, but these are the ones you might see in your textbook. We don't need to get too much into those. But let me see if I could get a example of a non-disjunction for you. Alright, that's now trying to get a simple enough picture to just show how you could end up with, you know, an extra one. Okay, I think this is simple enough. Okay, so if all you watch here, oops, that's not it. That hard to get like just a simple picture of this thing by. Alright. Forget that, yes? Alright, actually you can kinda see it here. You can't see the writing and thing, but there's a normal separation here. Shucks. There's a normal separation here, and you'll see that we start off with one pair and we end up with one chromatid in each cell, right? And with a non-disjunction, what we end up happening is two of these gametes come out normal, and here, instead of, you know, we get 1, 1, 1, 1, we end up with 1, 1, we end up with an extra one here, so we have 2. So this child might have Down syndrome, and you end up with a gamete that don't have that particular chromosome. That is why it's called a non-disjunction, when they pull apart unevenly. These really only get at multiple choice though. Right. So I believe that covers it for variation. So now we just have natural selection, and that will be a breeze. So let me go through natural selection, and then is module 3. Okay. Alright, so natural selection. Let me show you something here first. So I remember I'll show you this video here. So we had a seahorse, right? This male seahorse who pregnant. Well, the male seahorse does actually carry the offspring. And first of all, what we see in here is that we have an overproduction of these offspring. We have a lot more than you could say we need. So, same thing like with other animals, only we say leatherback turtles, which lay on average 70-75 eggs. So, why do you think that is? So this is because when the seahorse has 2,000 babies, only a very small fraction of those will survive at least until adulthood. So this is part of natural selection and is part of having what you call genetic vigor or wide gene pool. You need a lot of offspring or you want a break before module 3? Just say yes or no. Because after we finish natural selection, it might be close to 6 o'clock. We could come and finish module 3 and do some past papers. And I feel all it should be good for the night. Okay, so this is because we need a wide gene pool. We need a lot of them because we have a lot of selective pressures. We have changes in temperature, we have invasive species, we have diseases, we have even just like hazards in the terrain. You have a lot of different things that they have to overcome in order to survive. And the higher amount of offspring that we have is the better the chance of survival. And the little bit of differences that those offspring have gives you more and more chances to survive. Sometimes it could be the smallest difference. Sometimes it could just be that the tail a little shorter or the neck a little longer, or they're able to store a little more fatty tissue on a part of the body, or they're able to have a slightly bushier tail. It depends. It's very interesting to study, actually. It even comes down to plants producing toxins like caffeine to ward off insects. You have these basilisk lizards that their feet are just wide enough that they can actually skip across the water without sinking. It has enough surface tension to maintain their weight. So this is what one of Darwin's postulates would be: an overproduction of offspring, and that you would have little differences due to genetic variation and sometimes environmental variation that would have encouraged the survival of a species. And basically, these organisms that survive, they are able to pass on their advantageous traits to offspring. And those advantageous traits would be carried on from generation to generation. So a species has a better chance of survival if there's high genetic variation in its members. The best example is the Galapagos finches, because they were so varied with their beaks that they were able to adapt to many different diets and didn't have to compete with each other, and thus preventing competition with each other. They each had their own food source, their own resources. It has something that it is say about natural selection and survival of the fittest. You just have to be the most well adapted to the environment to beat out all the other species. You don't have to be the best of the best. So for example, if it have a bear chasing you and your friend, right? You don't necessarily have to be faster than the bear. You just have to be faster than your friend in order to survive. You just, so natural selection, survival of the fittest, tells you that you don't have to be the best of the best. You just have to overcome everybody else. You don't have to be the tops. You just have to overcome all the other weakest. And be in that alpha group that is able to survive. And that doesn't necessarily mean stronger and faster. It could just be having a certain fur color or being able to withstand a certain temperature. Alright, so natural selection, we know about these finches already, so I don't get too much into that. Okay, so before we get back into Darwin here, this is something that on the syllabus, they have it has its own little section in the syllabus, but I never see them bring a question on it. There's bring it for CSEC, and there's ask you to explain the biological species concept. and to point out any problems with the biological species concept. So you actually have two species concept, the biological species concept and the phylogenetic species concept. And the biological species concept is the one we are accustomed with since CSEC, which is two members of the same species are able to interbreed and produce fertile offspring. So, for example, you have two lizards of the same species are able to interbreed and produce, a little baby lizard. If you have two members of different species, they can still interbreed, but the offspring would be infertile. The example would be a donkey and a horse. When those reproduce, they form a mule, and mules are sterile. The problem with the biological species concept is that it cannot be applied to organisms that reproduce asexually. So they will say amoeba, for example, or hydra. They're still species. They still have groups of species, but this particular definition doesn't fit organisms that reproduce asexually. So we have a problem there. It also doesn't account for inbreeding. Where if you have a brother and a sister, and them is the same species, they're able to interbreed. The offspring might not be fertile, and of course it wouldn't be the best. It might not survive. So interbreeding here is also a problem. If you have a brother and a sister, or you have a mother and a son, or a father and a daughter, and they reproduce, you have incestuous relationships, they might form organisms that are mutated, that don't survive. So the biological species concept doesn't fit with that. A next example, just a little fact here, by the way. The youngest mother in the world, her name is Lina Medina, from Peru. I believe it's Peru. And she was 5 years old at the time when she had a baby. I believe she was impregnated at 5, had the baby at 6. And it's because of incest. It was her father raped her. And what's notable about that is that the baby actually grew into a full-blown adult, like nothing was wrong. It had no mutations or anything like that. Is one of those rare cases in which, incest, the product of incest actually, the offspring was unaffected. So instead of the biological species concept, we have the phylogenetic species concept. And this is where organisms will be classified according to their morphology. Morphology means their shape, their form, right? Morph means form. And this could include all organisms, because you can simply observe the form of any organism. And you can group them according to how they look. But you could already see where we're coming into problems with this. Almost every species, at least every large one, exhibits polymorphism. Polymorphism means that each member of the species may look wildly different. And this could be according to genetic variation. It could be according to their biological sex. It could be according to just their breed. So for example, if you look at these two dogs here, these two dogs are the same species. They are Canis lupus familiaris. They are domestic dogs. Both of them are different breeds, but they are the same species. If you are an alien, come down to Earth, and you'll be like, these two is the same species? They look too different. Well, the phylogenetic species concept would fail here because they look too different. So typically you have to take a mix of the two in order to define species. We don't actually have a fixed definition for a species right now. The other thing here is when there's differences due to biological sex. So for example, we have the peacock and the peahen. Of course, the male has to be more flamboyant than the female, because it's the male in the animal kingdom, is the male has to attract the female. So the females usually are smaller, they're less colorful, they're less vigorous than the male. So if we were to look at these two birds, you might, just looking at them, just observing them, just looking at their morphology, you might think of them as two different species, but that's not true. They are the same species, the only difference is their biological sex. The example here again would be a bird of paradise, where this phylogenetic species concept here would fail. And the male bird of paradise looks very different from the female. So this is the male. And the male, has to go through all these mating rituals in order to attract the female. The male had to dance, the male had to call, the male had to build nests, all kind of thing. And you can see here what the male is how to do. We're going to take a break when we finish natural selection, Anissa. You could take a break, and just rewind the video. So the male here looks very different from the female. And even after all this dancing, she had to go in a corner and try to figure out if he good enough. She had to check him out. Because the male is the one who had to be flamboyant. The male had to be bigger. The male had to have all these kinds of weird talents and things. Well, in the end, he get through. Kyle get through instead. Our next good example is this Japanese pufferfish. This Japanese pufferfish is the hardest working organism I ever see. This thing, this drag its ventral fin into the sand and form and decorate it with shells and all kind of thing. This Japanese pufferfish. And it has to do this. It has these fins that are designed to do this, which the female doesn't have. And you'll notice that it blends in. So it has to be able to do this to attract the female. It's not flamboyant. So it does all this. You tidy it up and basically make this whole geometrical pattern to attract the female. Well, if he get through that, I don't know what gonna make him get through. But yeah, each animal has these little adaptations that allow for survival. So yeah, each one is different, and is I always think it's kind of strange that we don't have a proper definition for species, but we have two different concepts, and I say these two are in the syllabus, and is worth to know. You have to know what they say and what are their limitations. So once again, the biological species concept says that two members of the same species are able to interbreed and produce fertile offspring, but you cannot apply this concept to asexual reproduction. So like amoeba that don't have two members of the same species. It also does not apply to inbreeding, which is they typically do not produce fertile offspring. Then we have the phylogenetic species concept, which you don't look at whether or not they can produce fertile offspring, but you more look at their biological characteristics, their biochemical characteristics, their morphological characteristics, their traits and so on. But again, this has problems as well, even though it can include all organisms. You have to be able to observe species through maturation, because even like if you look at a tadpole and a frog, they are very different. You have to look at their differences due to biological sex, their differences due to just genetic variation, like dogs, domesticated dogs. Right. So coming here to Darwin's deductions. So Darwin made 3 observations from which he made 3 deductions. I can't remember the last time this come, but this is something that they tend to bring as well. what were Darwin's 3 observations and his deductions? They very easy. The 3 observations he made, and remember he did this while he was on the voyage of the Beagle. The name of the ship was the Beagle, when he is going to the Galapagos Islands to observe the turtles there and the finches. And he noticed that when members of a species vary between each other, all organisms produce excess offspring, like what I'll show you with the seahorse. And population numbers remain fairly constant over long periods of time. That is when they stabilize. And yeah, you would get some increase and decrease, but they would not be drastic unless there's a drastic change in the environment, like there's a storm or a shift in temperature or a shift in water availability or the introduction of a disease or the introduction of an invasive species or urbanization or human intervention or something like that. If those things don't happen, population numbers remain fairly constant. So the deductions that he made for this is this: if traits can be inherited, organisms pass them on to their offspring. So, of course, the organisms that survive are the ones that are going to pass on. The ones that die cannot pass on their traits to the offspring, their alleles. 2, there's a struggle for, he called it a struggle for existence. In his book, The Origin of Species, he said that every organism has the struggle for existence. Kind of like all are we struggling to exist in this COVID environment and monkeypox and recession and all the food going up, we have a struggle for existence. We are Cape tomorrow, big struggles. But there's a struggle for existence amongst members of these species. Everybody had their own stress. And there's actually called it that, environmental stresses or selective pressures. So everything has something to overcome. Whether it has to find a reproductive mate, whether it needs to find shelter, whether it needs to find food, whether it needs to run from this hawk, whether it needs to burrow into the sand and escape some kind of predator, or whether it had to camouflage, or whether or not it had to bring food back for its offspring. Everything has a struggle for existence, and everything has to overcome that struggle in order to survive. It's actually a really nice thing to think about when you think about your own personal life, because everything does be a struggle. But is being able to overcome that, and being able to overcome it don't necessarily mean being smarter or being stronger or anything like that. Sometimes it's just the right place at the right time. Sometimes it's just simply giving the thing a try. It had this nice little anyway, I'll talk about that later. But members that are best adapted to their environment are the ones that are most likely to survive, reproduce, and pass on their advantageous traits. So if you have, let me give all the example. Imagine we have a deadly epidemic, not COVID, something that much worse. And it, I don't know if all they ever played The Last of Us. It's a PlayStation game, or if you ever hear about it. And you had the one girl, Ellie, who was she was immune to the fungus that was attacking everybody, and you could say that she was basically best adapted to the environment. But let me just imagine we have a case like that where we have a few people who are immune to a disease. Those are the people who are most likely to survive, to reproduce and pass on those traits. And as a result now, they could repopulate and replace, I don't want to say replace, but repopulate the environment, repopulate the cities and things, and replace everybody who has died. And as a result now, you have a even more vigorous population that's able to withstand that disease. So yeah, pretty simple. Alright, I don't think we have that much to go through with this again, and we'll take a break just now. Alright, so we have types of selection. There are 3 types of selection: directional selection, stabilizing selection, and disruptive selection. Well, I didn't mention part 2 because it looked unlikely she was actually, I don't think she's going to reproduce now, with a man at least, so that's why I kind of get away from The Last of Us example. Anyway, so coming back here now, let me explain the differences between these three. Alright, so alright, so you have three types of natural selection, as I said, right? I remember natural selection is the process by which heritable traits become either more or less common in a population because of selective pressures, as temperatures, invasive species, environmental shifts, diseases, etc. The ability to adapt to those pressures and overcome them and survive and produce offspring. So we have many different environmental pressures, and we have three types of selection: directional, stabilizing, and disruptive. So firstly, we look at directional selection. So directional selection is when individuals display a more extreme form of the trait. When those that are more extreme, those are the ones that are able to survive. So a good example here is Biston betullaria, which is the peppered mutt. And let me say in like dark, sooty areas, basically areas where you would have a lot of soot on the barks of trees, these black peppered mutts are best able to survive. In areas where it's kind of sandy, these white ones are able to survive. It really depends on the trait, but basically the curve shifts to the extreme in directional selection. Our next example, this is giraffes. You had long-necked giraffes, medium-necked giraffes, and short-necked giraffes. The long-necked ones is the ones that was able to survive because they was able to actually reach to feed on the foliage on the trees. So that's directional selection, where the extreme of a trait is favored. The extreme, not the average or not the norm, is the extreme of it is favored. So think about your long-necked giraffe, for example. So stabilizing selection is where you're going to have only the average form is selected for. So for example, with robins could lay clutches or groups of 2 eggs, 4 eggs, and 6 eggs, and sometimes even 8 eggs. The clutch that is most likely to survive is the one with 4 eggs. This is because if there's only 2 eggs, right, these two mightn't hatch, they might die, and they mightn't be able to actually grow into adulthood or continue the species. If there's way too much eggs in the clutch, what would happen is that they would all have to compete for food, and they would all have to compete for the same resources, so they're less likely to survive. But when you only have the average amount, which is 4 in this case, which is ideal, this is what works the best. This would be stabilizing selection. And remember, that is where the average form of the trait has the highest fitness. That is the one that's selected. So you get the highest population formed in the average variant of it. The last one that we have here is disruptive selection. So this is where it would favor both extremes, but not the average form. So example of this would be the Chinook salmon, and let me explain why. Right. So example, this would be the Chinook salmon. You have a small salmon, your medium-sized salmon, and a very large salmon. These 3 salmon have to reproduce. Now, the small salmon, they could, and well, if you know how fish does fertilize eggs, they don't actually mate. The female does lay the unfertilized eggs, and the male does go in and spray some gametes on it and fertilize it. So, if you ever watch Finding Nemo or Finding Dory, the female clownfish would lay the eggs in the anemones, and the male would come and spray some gametes on it and fertilize the egg. Well, sometimes it's the male is raised the offspring, by the way, because it's them who stuck with it. The female done gone. Anyhow, So the small one could actually sneak in undetected and fertilize them eggs. It could do this. The large one could kind of, well, I could say, the large one could kind of intimidate its way to fertilize the eggs because it's big, it's strong. So in this case here, where these middle-sized ones beaten up with each other to fertilize them eggs, this small man here don't reach. He bypassed them, he's small. This big one here, he inveigle, he way in, he reach. Whereas these middle ones here, they kind of just there. Short man and tall man get through. Medium sized man they get through. So that'll be disruptive selection. So this is where both extremes are favored over the average form. Yeah, and disruptive selection could lead to speciation in two different directions. So coming back here, to go through it one more time, directional selection, one variant that has an extreme form of the trait is selected over the average and the other extreme. So if we have the peppered mutt, we could see that the black mutts are able to camouflage and thus evade predation. You could barely see the black one here, but the white one is clearly more visible, so they're more easily predated. So directional selection would favor the black mutt over the gray and the white. It favors one extreme over the others. Stabilizing selection, I explained with the robins, favors the average form. The highest fitness is the average form. So where the clutch of 4 eggs have the highest chance of survival because they literally have more that are able to survive than 2, and they don't have to compete for resources like those that lay 6 and 8. So the clutches of 4 are most likely to survive. That is stabilizing selection. Not saying that these won't survive, but it would be the least likely. And with the salmon, disruptive selection, both extremes of the trait are selected over the average form, which could undergo 2 sets of speciation. So in this case here, as I said, the small ones could fertilize the eggs by being sneaky and stealthy. The big ones can intimidate their ways. They could basically brute force their way in to fertilize the eggs. The average form ones has neither advantage, so these are selected against. So disruptive selection is when both extremes of the trait are selected over the average form. And this is where, again, you could have 2 different sets of species being formed from the small and the large. It depends. So they didn't bring these 3 types of selection in a while. So I believe that they might bring it, and you should know at least one example for each. This could easily be a 6-mark to 8-mark question, especially if it involves the graphs. Right, this is the last thing we have to get into here, which is isolation mechanisms and speciation. So by 6 o'clock we could take a break. So isolation mechanisms are barriers that are going to prevent members of a species from interacting. So these barriers can be geographical, behavioral, mechanical, ecological, or temporal. We're going to go through each one now. But basically, when you have these isolation mechanisms, they prevent 2 species from interacting, right? So if the species A would not interact with species B, or members of group A would not interact with members of group B, and they could be the same species. And therefore, you're not going to have any gene flow. Gene flow means that they're not going to exchange DNA with each other. The alleles from one is not going to cross over into the population of another one. This is going to lead to splintered populations. And these populations are going to change over time, and speciation may occur. So an example of this would be a geographical barrier. Shout out to amoeba sisters again. So geographical barriers are where, well, there's a literal land mass or there's a geographical formation that's preventing 2 members of the species from meeting. So example could be a body of water, example could be a large mountain, or examples could be a canyon. So for example, it are two types of squirrels, the kaibab and the abe squirrel, that are separated on both sides of the Grand Canyon in the USA. Of course, they wouldn't be able to meet because it have a whole canyon separating them. So you could imagine now on each side, on each part here, it would have its own selective pressures. So for example here, you would have, you could have a different predator here on the left than on the right. And as a result now, both these members of the species have to find different ways to overcome this. So they might be better at hiding, they might be better at running, they might be better at camouflaging, they might be better at burrowing. It really depends on the terrain and the type of environment that they're in. But geographical barriers could also mean that there are different types of food available on both sides. Example would it be the finches. On some islands it would be more prevalent with fruits. On some islands more prevalent with insects. On some islands more prevalent with worms. So geographical barriers would prevent these organisms from meeting, but they would basically evolve in their own separate ways. Ecological barriers now. This is where it's different from a geographical barrier, where two species are going to live in the same area, but they're going to rarely or never meet. So an example could be, let me say, these two frogs in this illustration. You have one of them that prefer to be terrestrial, which means more land-based, and you have one of them that prefer to be more aquatic, water-based. So ecotone charity would be more of a transitional area between two ecosystems. So you could have ecological barriers in an ecotone, but it doesn't necessarily have to be. I'll give you a good example, like a good Trinidadian example. You have these, you have an endemic species in Trinidad called a golden tree frog. They typically found around Paramin or the El Tucush mountain that we have here in Trinidad. And these golden tree frogs are actually pretty rare to spot. See if I get a picture of one. So these golden tree frogs here, they live in, yes, now, you can't find one, huh? This are working out so hot. All right, there's a golden tree frog. They typically live in a type of plant called a bromeliad. And a bromeliad could basically hold a lot of water in it, kind of like a small little minor pond, if you can imagine. And they could live in that. They could live and die in that. And you have other types of frogs or bullfrogs in the area that would never come into contact with these frogs, even though they're living in the same ecosystem. So that would be an ecological barrier. It's not necessarily a geographical barrier because there's no big landmass separating them, but they favor different ecologies. That's an ecological barrier. They just prefer, I prefer to be here, you prefer to be there, we'll probably never meet. I prefer this hot area, you prefer this cold area. It's kind of like that, but it's within the same ecosystem. Behavioral barriers are when two species have different courtship behaviors, and as thus they will not mate. A good example is when two birds have different mating calls. One mating call might not be compatible or might not sound good to another bird, and as a result they just won't mate. There's a behavioral barrier. It prevents them from meeting, it prevents them from interacting, it prevents them from mating. Eastern and western meadowlarks, that's a type of bird, have different mating calls. Something you might not know. It has a good example on Easter. Some species of grasshoppers and crickets have different calls and they just never interact as a result. It's kind of like if you have somebody supporting some football team and the next one supporting the next football team, or you have somebody AI is a PNM, and the next one AI is a UNC, nah, we can't do it. You know what I mean? That's a bad example, but you have different types of fireflies that have different lighting signals. They light up at different periods, and as a result, if they have two different lighting signals, they would never interact, they would never reproduce. That's a behavioral. The other two that we have is mechanical barriers and temporal barriers. Mechanical barriers mean that the two species are physically incompatible in terms of size, in terms of genitalia, in terms of gametes. So for example, we say a duck, right? If you Google duck penis, you can see that the penis of a duck is like a long corkscrew. That long corkscrew cannot just penetrate any other bird vagina. And as a result now, they're mechanically incompatible. It's only that duck could mate with that certain species of duck. You can't mate with other types of ducks. If you look up echidna, if you know Sonic the Hedgehog and Knuckles from that series, Knuckles the Echidna, echidna actually has two to three different penis glands, as penis heads. And that's because the female tends to have about 2 or 3 vaginas. It depends. Of course, it needs to be very specific, and there's a mechanical barrier. It prevents other species from mating with it. So yeah. So there's a mechanical barrier. But it's not just with animals, it's also with plants. Sometimes the pollen is just incompatible with the stigma of another plant, or sometimes just the gametes. The sperm cell doesn't have the receptors to fuse with the oocyte of another. The other one, the last one that we have here, are temporal barriers. Temporal means time. So in terms of this, it just means that two species, they could live in the same area, live in the same ecosystem. They could have everything compatible with each other, but except that they mature at different times. So an example could be with two plants. You could have one plant producing pollen, and the next one, the stigma is not ready to receive that pollen. You could have some cicadas that some of them mature in 13-year lifespan, some of them mature in 17-year cycles. It is only every, whatever is the LCM at 13 and 17 is only that 200 and something years they're actually able to meet up. So temporal barriers are where they experience different times of sexual maturity. You know, you have some stink men out there, the fully mature men, but they're like small girls. You can't do nothing with it, right? Don't do that, eh? But there's a temporal barrier because it's two different, times of sexual maturity. And you have some animals that exhibit this. You have some of them that the males mature before the females, and they would have to actually go out and seek out females from another area. It depends. And I think that wraps it up for... No. It's not one more part. Allopatric speciation. Hey, don't mention that name here. Hey, you want to bring down the Kremlin down on New York? Hey, don't mention that name, Spurwin is Spurwin. Spurwin is related to nobody else. Spurwin have heating, other people have their thing. Alright. Allopatric speciation. Yes, Carissa. Temporal barriers like protandria and protogeny, that is absolutely correct. Yeah. Allopatric speciation. There's the last part here. Allopatric. Allo means different or other, and patric means fatherland, from patriarch. So allopatric means other land, other fatherland. So basically, allopatric speciation is speciation that occurs in other lands. It originates in other lands. So, is It's the same. Geographical isolation or geographical barriers lead to allopatric speciation. So we talked about this already. Species will form splinter groups due to a separation in landmasses. It could be a mountain, it could be an ocean, it could be a canyon. And the presence of this landmass means that the groups will not meet, they will not interact, you'll have no gene flow, you'll have no mating, no reproducing, nothing. They might never see each other. So we say use a mountain as an example. On either side of the mountain, there may be different selective pressures, different colored trees, different terrain, different predators, different amounts of water. And each member of these species must now adapt to these pressures. So a good example to use is the Galapagos finches, where allopatric speciation occurred because they're separated by bodies of water. And the Kaibab and the Abe squirrels are separated by the Grand Canyon, so they have to make do on each side and encounter different predators on each side and survive those predators on each side. So allopatric speciation, remember allo means other, patric means fatherland. So it means speciation that occurs on other lands, separation of lands. Survival of the fittest outcast might not be enough. You'd have to put a lot more into your definition there. So the other type of speciation would be sympatric speciation. Sim means like within or similar, kind of like symmetry. So sympatric speciation occurs when there is no geographical separation. It's the opposite of allopatric speciation. So this is basically where there's no separation, no landmass separating the organisms, yet they have undergone speciation. A good example of this is the maggot fly or the fruit fly called R. pomonella. And basically what happened is that they typically lay their eggs in berries called hawthorn berries. And when I believe it was the Pilgrims or the Quakers, when they came over in New England in the 1800s, New England is that upper eastern part of the USA, where have Vermont and New Jersey and New York and things like that. When they would have come there, they brought apples with them. And these fruit flies now started laying their eggs in apples instead of the hawthorn berries. And there's no geographical separation, but only a difference in behavior. And because of this difference in behavior, because one remained laying eggs in berries and the other one laid eggs in apples, that actually resulted in speciation. That's an example of sympatric speciation where you had no geographical separation. New Hampshire, yeah. Giovanni, yeah. If all the other isolating mechanisms did not lead to if all the other isolating mechanisms lead to speciation but it's not geographical, it typically is sympatric. Let's say the fruit fly is the good example. Yes, so we could take a break there. We'll take an hour, or they go and eat and thing, and we will reconvene at 7.
Geddes Grant Office World presents the award-winning Lanier copiers. Low in cost, high in performance, Lanier matches the copying needs of any size business. Call or visit Geddes Grant Office World for special introductory offers.
Rough play. Tough too. This shoe will leather it through. Lakers leathers, tough enough for the games people play.
Happy days when you shop at Sitaram Book Center, Tunapuna. Get 12 copybooks for $10, 16-inch flats for Toko Composite, Sanwa Junior and Senior Secondary, Eldorado Junior and Kirep Junior Secondary. Sitaram Book Center, Eastern Main Road, Tunapuna, where good education begins.
It's the once-in-a-blue-moon clearance sale at Lim Choice. Get plastic toilet seats at $55, 10% off factory prices on paints. Cabinet knobs and hinges at 50 cents. Everything is on sale at Lim Choice. Once-in-a-blue-moon clearance sale. Western Main Road Center.
Barbecue, pit and pop,
on Charlotte Street. There we'll meet. The pit is hot, the pub is new, all kinds of food, barbecued. Smells good from the outside. It's the only one of its kind. Let's go inside. Barbecue pit and pub.
Opposite the old market, 33 Charlotte Street.
So there goes another satisfied customer. You've just got 3 days to get that car you've dreamed of at SP Motors' big used car flea market. Sale starts 10 a.m. daily on June 26, 27, and 28. Hurry to SP Motors. Fly over Chaguanas now.
It's bad guys versus good guys in giant-sized puppy sticker 6-packs from Sunshine Snacks. Get them for less than half the price with any 4 wrappers from these Sunshine Snacks or Lolita, plus $2 at any Batter store.
In my profession, staying in shape is very important. Regular exercise, good diet, and of course, stay fresh. That's how I do it. Stay fresh, low fat. My choice for staying in shape.
My answer to skin protection for the whole family, new Protex Skin Health Soap. Protex helps guard my baby against diaper rash and our son against cuts and bruises. Don't talk about my teenage daughter. Yes? She swears by Protex for skin blemishes. My husband, now he likes Protex because it takes away bacteria and the odor they cause. Of course, I love Protex too. It really helps against those little skin infections, leaving my skin the way I want it. Protex protects my family every day.
Out from the dark comes a strength that goes beyond expectations. Special old cask rum, smooth, mellow, blended in the tradition of Trinidad's finest old rums. Special old cask, the dark strength you won't take lightly.
Carpenters, come, come, come. Tonight you be licensed, electricians, run, run, run. Tonight you be licensed, nails and accessories by the store. Bulbs and light fixtures evermore. Paints and brushes just for you. Paint up your house up with looking new. Tonight you be liars, working a number. Staff well, helpful, service much better. Hardware, so forth and get two. Tonight you be liars, the place for you.
For great taste in sandwiches, start with a freshly baked Linda loaf, hamburger bread, hops or even bagel. And for quality cakes and pastries, Linda's are the best. Order beautifully decorated cakes for those special occasions, weddings, birthdays or graduations. And if you want that extra special, personalized touch, their friendly staff will decorate while you wait. Visit Linda's at 55 Western Main Road, St. James, or any of their 5 locations. Linda's Bakery, coming soon to Excellent City Centre in Port of Spain.
This is the season to be happy. Open up your eyes. Get great big savings on gift items when you shop at Pennywise. Get great prices and hair and skincare. It's for every size. Save on toiletries, lotions, and Christmas items when you shop at Pennywise.
Experience the grandeur of the cinema in the comfort of your home and watch the big screen come to life with sharp 25 and 27-inch remote-controlled color TVs. These cable-ready, brand-new sharp design sets will fill your living room with stereo sound, complete with closed caption, sleep timer, child view limiter, and more. Each TV set comes with a famous CTC Electronics 3-year warranty, so look sharp.
Clear for takeoff.
Aye, aye! A few days before Christmas, Casey brought for me Christmas gift packages, delightful party bags, ooh, and candies, and cosmic gum bulbs to decorate your trees. Casey Candy, you make life sweet for me.
is in action. People like you and me share one vision. From the start, from the heart, to make a brighter day. It's the way we bring banking to life. First citizens, building a nation. People like you and me, we're first citizens.
Ready, get set, go! It's the ultimate driving machine, the KFC Bunny Buggy. Fast, fun, and only $3.99 with the purchase of a KFC Kids Pack and one Hop Hop dollar. Hurry, get yours today. Stocks are limited.
Come now, can you buy a real nice lady?
What the bullshit on Friday?
Say, senorita, I bring a cow. We can have milk for soul from now.
Who tell you I was a cow? If you know what's the fear, move back out.
But darling, I was only trying to be helpful. Think about the convenience.
Convenience? Mister, this is convenience. Nicer milk powder coming straight from Holland. And you'll believe it, straight from the cow. This is all that is used now.
These days you're really looking nicer and nicer.
Get yourself. Besides, take that cow back where you find it.
You can't please nobody now. You self couldn't tell me nicer have milk powder.
Where is that mumbling there?
I said I didn't know that nicer have milk powder.
Well, nicer have everything. This is nicer country.
Nicer is nicer.
For Mother's Day, give Mom a gift for good health and longer life from Modern Caribbean. Strong Magnetic Cup cures and prevents kidney and bladder stones, obesity, and other diseases. Slimmer Belts relieves back and menstrual pains and slims the waistline. Massage sandals and shoes relieves arthritis, tiredness, and improves health. Necklaces and bracelets relieves pain in neck, shoulders, wrists, and more. Available at Modern Caribbean, Roundabout Plaza, Gulf City Mall, and Mon Repo, San Fernando. Phone 653-0011 or 638-2682.
If you can dance, how long can you do it?
Yeah, the biggest dance-a-thon is on, so check the funky prizes to be won. Have some fun as you compete, but remember it's the clock you've gotta beat.
Dance-a-thon 93 from Radio 95.
Caribbean Sound Basin, May 30th, 2 to 10.
Puff and Stuff for the freshest bread, the tastiest cakes and pastries, and delicious desserts. Kids love the delightful lunches. Check us for children's parties, meetings, and seminars. With a friendly and knowledgeable staff, Puff and Stuff is there for you. Say Puff and Stuff.
1993 dimensional and like today ain't that conventional. So what we've got is quite intentional at Cole's Powerhouse. Our electrical range is sixth sensational. It's new, it's oh so conversational, and all our prices inspirational at Court's Powerhouse. Up to date and always fashionable, Court's top brands are international, and your choice is always rational at Court's Powerhouse.
Kids, here's great news from Royal Castle. Get a delicious Canning's Lolly absolutely free when you purchase Royal Kids Castle now. Better all over with your Canning's Lolly.
Are you serious about your investment? Let the Trinidad Cooperative Bank work for you. Your investment yields higher earnings with our fixed deposit. Rates are negotiable according to your deposit. Call us. We'll come to you to discuss and advise on your investment. The Trinidad Cooperative Bank.
Change your oil and you can be all in smiles, 'cause you can win 70,000 free miles. If you want a chance to win all this free gas, count on NP now and change your oil fast.
You know, Amanda's feel proud when he reach home and he have a nice door to open, a nice young single. solid, secure, quality wood, beautiful young designs, carved doors, flush doors, doors with glass, solid panel doors, secure doors, and at prices everybody could afford. Yes, man! With a young sing door behind you and a young sing door in front of you, you and the whole family could.
Yes, it's open house and free stickers for all kids this Sunday at Carnival Expo. Bring the kids for Fun-Filled Day. There's Magic Fantasyland and Calypso Martini at 4 o'clock. Open house, free stickers for all kids this Sunday at Carnival Expo. Admission free until 1 o'clock.
Want to know how to cross that bridge that separates you from the house of your dreams? Check with the Trinidad Cooperative Bank, now offering bridging finance for first-time builders. The Cooperative Bank at Charlotte and Duke Streets, Barataria, and Point Fortin.
The National Commercial Bank, we aim to give you quality service. Convenient locations to service your banking needs. Working specially to satisfy each customer, your affairs are handled confidentially and with empathy. NCB offers security, competitive interest rates, and added value for your money. Our wide range of banking products includes the unique Life Insured Savings Plan. The National provides state-of-the-art banking technology, including our 24-hour self-service automatic banking machines. All this and more at the National Commercial Bank, the bank to help build the country.
Go ahead and get Miss Violet, tender juicy chicken. Get Miss Violet. Each and every bite is a shared delight of the zesty chicken. Get Miss Violet.
The National Gas Company transports natural gas, a vital resource for our nation's industrial development. Trinidad and Tobago's economic turnaround is in our pipelines. See the light.
There are some things that neither time nor distance can change. With Illusion, there's no time or distance.
Because a man never forgets a woman whose fragrance he remembers.
With Illusion, there's reality. Illusion, the sensational new perfume and deodorant for today's woman. When you need to keep an important appointment, when you need to keep your cool all day long, make your day one of long-lasting confidence with Rage. With Rage, you're silently ready for any situation. Rage, the deodorant cologne for men. She'll never forget you for it.
When your job calls for a lot of brawn and plenty brain,
Go for a gold, go for a gold, go for a Nutriment. Gold!
Nutriment Gold, it nourishes all the parts of your body.
When you need to lose weight, you exercise. When you need a garden, you plant. You need your telephone, so pay your phone bill. When you pay your phone bill on time, you're doing what you have to keep your telephone. So pay it, and don't cut your lifeline.
Serving people.
Telco.
In the offices, on the streets, in the shops, and in the factories, the wheels of enterprise begin to turn as the country gears for yet another day of productive effort.
Well, you know me ain't no housewife, but I know a good product when I see one. Well, all you don't know that a Breeze box holding more detergent than some of them other detergent box. But what I really want you to know and understand is that Breeze is unsurpassed in cleaning clothes and making them smell good too. So don't get tired. With Breeze, you could see it clean and smell it clean all through. Still good economic sense. Don't doubt me.
I love the way you look in that blouse.
That's because I wore it when we met.
Really? But it looks so new.
Because I wash it in Softland.
What's Softland, Mommy?
Softland's a gentle detergent and I use it on all our delicate clothes.
Will you wash my nightie in Softland too?
I did, with my very own hands.
And it smells nice, too.
Softland. Tender loving care for clothes you care about.
Never before so much to see. Never before so much to do. Everybody will be at Caribbean Expo 88. See you at Expo 88.
Black in the raw. Gold when refined. NP Super Lube. Black Gold. A supreme quality, high-performance motor oil. Super Lube. Only NP has it, so demand it. NP Super Lube guarantees protection for your engine. NP Super Lube. Quality you can depend on at all service stations.
Now in Trinidad, an executive limousine service from Henry's Taxi Service and Car Rentals, 3 Damien Street. Just take the Roxy roundabout by Hamilton Street, telephone 23716 for Henry's executive limousine service. Chevys and Cadillacs, and over 30 years' experience of driving top businessmen and politicians in total air-conditioned comfort. Get yourself ready for your next meeting or flight in a limousine from Henry's Taxi Service. 24 hours. Telephone 23716.
You can paint? I thought we getting a painter.
Anybody could paint if they use kaleidoscope. It's tough and durable like you. And top quality like you. And long-lasting and colorful like me. With kaleidoscope, anybody's a painter. You and all. Color your home, kaleidoscope.
Think of what you can do when there's a lion in you. Royal Extra Stout. Look for the lion on the label.
Drink something solid, solid. Drink Royal Extra Stout. Drink Royal Extra. Solid, solid. Hey!
Get a fresh new deal. Now it's any Coca-Cola Premium of your choice with any marked crowns plus cash. Get basketballs, wraparound darkers, head and sweatband bands, Coca-Cola t-shirts. Get your choice now. In exchange for crowns with any premium marked inside plus cash at any Delimas, all banner stores and Almondos Tobago.
It's a Coke. Coca-Cola music.
Nestle sweetened condensed milk.
It's a taste that you never outgrow.
There's a toasted bun with a Mario's burger.
That's no secret.
There's lean beef in a Mario's burger.
That's no secret.
There's fresh salad in a Mario's burger. That's no secret, no.
The secret is the sauce.
It's made with care. That Mario's burger.
That's no secret.
It's a short wait for a Mario's burger.
That's no secret.
It's a perfect meal. It's a Mario's burger. That's no secret, no.
The secret is the sauce.
Mario's?
Brilliant.
Dixie Cola now. Dixie Cola now. Dixie Cola now. Dixie Cola. Dixie Cola brings you cola in 16-ounce bottles. Cola you can use and use again. The plastic cap keeps it from going flat. Get with it, cause this is as good as it gets. Dixie Cola, now in 16-ounce bottles.
The grand finale of On Track, the most happening television program on air, videotaped on location at Club Platinum West Mall. On Track brings a countdown of the 5 hottest songs played in clubs throughout Trinidad and Tobago. This week we feature Kindred.
It's just a Trini vibration to African rhythm. You're begging for the music, so I reach to get you some.
Sister Run.
I'm a sister who can sing into a song, get down. Like a funky James Brown, my song. There's not one you can speak to, so get a stand up and rock so you know what I can.
General Grant.
In the dance on the shot, booyaka, booyaka, they shut me down, cause they shot car. In the dance on the back, booyaka, booyaka, holy papa, body part.
And Victorian.
Look, kick it up and let me what I'ma do. I'ma take the mic and I'ma tell it to you. It's true, I'ma tell it to you. Here's a second here, we have a brand new crew. And every time it come and drop it, come on, come on, jump it down and round.
The grand finale of On Track this Saturday at 12 right here on AVM channels 4 and 16.
Come down and see for yourself just how we are beating high prices at Quartz.
It's amazing what it does to people. You've never seen anything like it.
Guaranteed low prices, easy credit, low deposit.
With the down payments cut down and low, plus the chance to be a Quartz cash accumulator winner.
So come on down to Quartz now. You'd be amazed.
It's time to shop at Quartz.
To all them Magabottle boys, come on to the play scene. You see, don't you have one original 16 in the land? Drink up, drink up, drink up, drink up. All them youths them. Drink up, drink up. All them pussy them. Drink up, drink up. All them massive them. Drink up, drink up. When you put the massive on it up, your massive knob, massive 16 in the air, you just drink up, drink up. Drink up, drink up. With your massive flavor. Drink up, drink up. Original power. Cannons rule the scene, original massive 16. Drink up, drink up.
If you walk downtown, wouldn't it be nice if you could shop downtown? Some place where you could walk around, up and down, for everything conceivable under the sun. If you walk downtown, it's easier to shop downtown. We're right here at the cool sort mall, the only place.
It have a smile out there, coming from right in here, coming from the soul of our shores, and you know that smile is truly yours. That smile come from you, and it come from me too. Keep it flying forever, because that smile is truly yours. We gon' fly that smile, with pride and style, with an engine that roars like a lion for a cause. Be we, truly yours.
The fete is grand, with products from Angostura. Get your glass in hand and be part of this fiesta. Everybody drinking,
White oak and tonic water, Angostura rum punch with hammers and appetizer.
Drink a rum and a punch a cream, drink a rum.
Old oak is the pleaser.
Drink a rum and a punch a cream, drink a rum.
Royal oak is the master.
Drink a rum and a punch a cream, drink a rum.
Well, to get all your flavors,
drink a rum and a punch a cream, drink a rum.
Angostura bitters. Merry Christmas, everybody from Angostura.
Drink a rum and a punch a cream, drink a rum.
Geddes Grant Office World presents the award-winning Lanier copiers. Low in cost, high in performance, Lanier matches the copying needs of any size business. Call or visit Geddes Grant Office World for special introductory offers.
Rough play. Tough turf. This shoe will leather through. Lakers leathers. Tough enough for the games people play.
Happy days when you shop at Cram Book Center, Tunapuna. Get 12 copybooks for $10, 16-inch plats for Toko Composite, Sanwa Junior and Senior Secondary, Eldorado Junior and Kirab Junior Secondary. Cram Book Center, Eastern Main Road, Tunapuna, where good education begins.
It's the once-in-a-blue moon clearance sale at Lim Choice. Get plastic toilet seats at $55, 10% off factory prices on paints. Cabinet knobs and hinges at 50 cents. Everything is on sale at Lim Choice. Once-in-a-blue moon clearance sale. Western Main Road, St.
Barbecue, pit and pub, on Charlotte Street. There we'll meet. The pit is hot, the pub is new. All kinds of food. Barbecued, smells good from the outside. It's the only one of its kind. Let's go inside. Barbecue pit and pub.
Opposite the old market, 33 Charlotte Street.
So there goes another satisfied customer. You've just got 3 days to get that car you've dreamed of at SP Motors' big used car flea market. Sale starts 10 a.m. daily on June 26, 27 and 28. Hurry to SP Motors, Flyover Chaguanas, now.
It's bad guys versus good guys in giant-sized puppy sticker 6-packs from Sunshine Snacks. Get them for less than half the price with any 4 wrappers from these Sunshine Snacks or Lolita, plus $2 at any Batter store.
In my profession, staying in shape is very important. Regular exercise, good diet, and of course, stay fresh. That's how I do it. Stay fresh, low fat. My choice for staying in shape.
My answer to skin protection for the whole family, new Protex Skin Health Soap. Protex helps guard my baby against diaper rash and our son against cuts and bruises. Don't talk about my teenage daughter. Yes? She swears by Protex for skin blemishes. My husband, now he likes Protex because it takes away bacteria and the odor they cause. Of course, I love Protex too. It really helps against those little skin infections, leaving my skin the way I want it. Protex protects my family every day.
Out from the dark comes a strength that goes beyond expectations. Special old cask rum, smooth, mellow, blended in the tradition of Trinidad's finest old rums. Special old cask, the dark strength you won't take lightly.
Carpenters, come, come, come. Tonight you be license, electricians, run, run, run. Tonight you be license, nails and accessories by the store. Bulbs and light fixtures evermore. Paints and brushes just for you. Paint up your house, have it looking new. Tonight you be liars, holding a long back. Staff well, helpful service, much better. Hardware, so what and get to. Tonight you be liars, the place for you.
For great taste in sandwiches, start with a freshly baked Linda loaf, hamburger bread, hops or even bagel. And for quality cakes and pastries, Linda's are the best. Order beautifully decorated cakes for those special occasions, weddings, birthdays or graduations. And if you want that extra special, personalized touch, their friendly staff will decorate while you wait. Visit Linda's at 55 Western Main Road, St. James, or any of their 5 locations. Linda's Bakery, coming soon to Excellent City Centre in Port of Spain.
This is the season to be happy. Open up your eyes. Get great big savings on gift items when you shop at Pennywise. Get fragrances and hair and skincare. It's for every size. Save on toiletries, lotions, and Christmas items when you shop at Pennywise.
Experience the grandeur of the cinema in the comfort of your home and watch the big screen come to life with Sharp 25 and 27-inch remote control color TVs. These cable-ready brand new Sharp design sets will fill your living room with stereo sound, complete with closed caption, sleep timer, child view limiter, and more. Each TV set comes with a famous CTC Electronics 3-year warranty, so look sharp.
Clear for takeoff.
Aye, aye! A few days before Christmas, Casey brought for me Christmas gift packages, delightful party bags, ooh, and candies, and cosmic gumbulbs to decorate your trees. Casey Candy, you make life sweet for me.
First citizens, bankers in action, people like you and me, share one vision, from the start, from the heart, to make a brighter day. It's the way we bring banking to life. First citizens, building a nation, people like you and me, we're first citizens.
Ready, get set, go! It's the ultimate driving machine, the KFC Bunny Buggy. Fast, fun, and only $3.99 with the purchase of a KFC Kids Pack and one Hop Hop dollar. Hurry, get yours today. Stocks are limited.
Come now, can you buy a real nice lady? What can you say on Friday? Say, senorita, I bring a cow. We can have milk for so from now. Who tell you I was a cow? If you know what's the fear, move back out. But darling, I was only trying to be helpful. Think about the convenience. Convenience? Mister, this is convenience. Nicer milk powder coming straight from Holland. And you'll believe it straight from the cow. This is all that is used now. These days you're really looking nicer and nicer. Catch yourself. Besides, take that cow back where you find it. You can't please nobody now. You self couldn't tell me nicer have milk powder. Where is that mumbling there? I said I didn't know how nicer have milk powder. Well, nicer have everything. This is nicer country.
Nicer is nicer.
For Mother's Day, give Mom a gift for good health and longer life from Modern Caribbean. Strong Magnetic Cup cures and prevents kidney and bladder stones, obesity, and other diseases. Slimmer Belts relieves back and menstrual pains and slims the waistline. Massage sandals and shoes relieves arthritis, tiredness, and improves health. Necklaces and bracelets relieves pain in neck, shoulders, wrists, and more. Available at Modern Caribbean, Roundabout Plaza, Gulf City Mall, and Mon Repos, San Fernando. Phone 653-0011 or 638-2682.
If you can dance, how long can you do it?
Yeah, the biggest dance-a-thon is on, so check the funky prizes to be won. Have some fun as you compete, but remember it's the clock you've gotta be.
Dance-a-thon 93 from Radio 95.
Caribbean Sound Basin, May 30th, 2 to 10.
Puff and Stuff for the freshest bread, the tastiest cakes and pastries, and delicious desserts. Kids love the delightful lunches. Check us for children's parties, meetings, and seminars. With a friendly and knowledgeable staff, Puff and Stuff is there for you. Say Puff and Stuff.
1993 dimensional, and like today ain't that conventional. So what we've got is quite intentional at Cork's Powerhouse. Our electrical range is sixth sensational. It's new, it's oh so conversational, and all our prices inspirational at Cork's Powerhouse. Up to date and always fashionable, Cork's top brands are international, and your choice is always rational at Cork's Powerhouse.
Kids, here's great news from Royal Castle. Get a delicious Canning's lolly absolutely free when you purchase Royal Kids Castle now. Better all over with your Canning's lolly.
Are you serious about your investment? Let the Trinidad Cooperative Bank work for you. Your investment yields higher earnings with our fixed deposit. Rates are negotiable according to your deposit. Call us. We'll come to you to discuss and advise on your investment. The Trinidad Cooperative Bank.
Change your oil and you can be all in smiles, 'cause you can win 70,000 free miles. If you want a chance to win all this free gas, count on NB now and change your oil fast.
You know, Amanda's feel proud when he reach home and he have a nice door to open, a nice young sing door. Solid, secure, quality good, beautiful young sing designs, carved doors, flush doors, doors with glass, solid panel doors, secure doors, and at prices everybody could afford. Yes, man, with a young sing door behind you and a young sing door in front of you, you and the whole family could.
Yes, it's open house and free stickers for all kids this Sunday at Carnival Expo. Bring the kids for Fun Filled Day. There's Magic Fantasyland and Calypso Matinee at 4 o'clock. Open house, free stickers for all kids this Sunday at Carnival Expo. Admission free until 1 o'clock.
Want to know how to cross that bridge that separates you from the house of your dreams? Check with the Trinidad Cooperative Bank, now offering bridging finance for first-time builders. The Cooperative Bank at Charlotte and Duke Streets, Barataria, and Point Fortin.
The National Commercial Bank, we aim to give you quality service. Convenient locations to service your banking needs. Working specially to satisfy each customer, your affairs are handled confidentially and with empathy. NCB offers security, competitive interest rates, and added value for your money. Our wide range of banking products includes the unique life-insured savings plan. The National provides state-of-the-art banking technology, including our 24-hour self-service automatic banking machines. All this and more at the National Commercial Bank, the bank to help build the country.
Go ahead and get Miss Violet, tender juicy chicken. Miss Violet. Each and every bite is a shared delight of the zesty chicken. Miss Violet.
The National Gas Company transports natural gas, a vital resource for our nation's industrial development. Trinidad and Tobago's economic turnaround is in our pipelines. See the light.
There are some things that neither time nor distance can change. With illusion, there's no time or distance.
Because a man never forgets a woman whose fragrance he remembers.
With illusion, there's reality. Illusion, the sensational new perfume and deodorant for today's woman. When you need to keep an important appointment, when you need to keep your cool all day long, make your day one of long-lasting confidence with Rage. With Rage, you're silently ready for any situation. Rage, the deodorant cologne for men. She'll never forget you for it.
when your job calls for a lot of brawn and plenty brain.
Go for a gold, go for a gold, go for a Nutriment Gold!
Nutriment Gold. It nourishes all the parts of your body.
When you need to lose weight, you exercise. When you need a garden, you plant. You need your telephone, so pay your phone bill. When you pay your phone bill on time, you're doing what you have to keep your telephone. So pay it, and don't cut your lifeline.
Serving people.
Telco.
In the offices, on the streets, in the shops, and in the factories, the wheels of enterprise begin to turn as the country gears for yet another day of productive effort.
Well, you know me, I no housewife, but I know a good product when I see one. Well, all you don't know that a breeze box holding more detergent than some of them other detergent box. But what I really want you to know and understand is that Breeze is unsurpassed in cleaning clothes and making them smell good too. So don't get tired. With Breeze, you could see it clean and smell it clean all through. Still good economic sense. Don't doubt me.
I love the way you look in that blouse.
That's because I wore it when we met.
Really? But it looks so new.
Because I wash it in Softland.
What's Softland, Mommy?
Softland's a gentle detergent and I use it on all our delicate clothes.
Will you wash my nightie in Softland too?
I did, with my very own hands.
And it smells nice, too.
Softland. Tender loving care for clothes you care about.
Never before so much to see. Never before so much to do. Everybody will be at Caribbean Expo 88. See you at Expo 88.
Black in the raw. Gold when refined. NP Super Lube. Black Gold. A supreme quality, high-performance motor oil. Super Lube. Only NP has it, so demand it. NP Super Lube guarantees protection for your engine. NP Super Lube. Quality you can depend on at all service stations.
Now in Trinidad, an executive limousine service from Henry's Taxi Service and Car Rentals, 3 Damien Street. Just take the Roxy roundabout by Hamilton Street, telephone 23716 for Henry's executive limousine service. Chevys and Cadillacs, and over 30 years' experience of driving top businessmen and politicians in total air-conditioned comfort. Get yourself ready for your next meeting or flight in a limousine from Henry's Taxi Service. 24 hours. Telephone 23716.
painter. Anybody could paint if they use kaleidoscope. It's tough and durable like you, and top quality like you, and long-lasting and colorful like me. With kaleidoscope, anybody's a painter. You and all.
Color your home, kaleidoscope.
Think of what you can do when there's a lion in you. Royal Extra Stout. Look for the lion on the label.
Drink something solid, solid. Drink Royal Extra Stout. Drink Royal Extra. Solid, solid. Hey!
Get a fresh new deal. Now it's any Coca-Cola Premium of your choice with any marked crowns plus cash. Get basketballs, wraparound darkers, head and sweatband bands, Coca-Cola t-shirts. Get your choice now. In exchange for crowns with any premium marked inside plus cash at any Delimas, all Batter stores and Almondos Tobacco.
It's a Coca-Cola music.
Well, yeah. I think we back. Start back in a minute. Right. 7 o'clock. I feel it's time to start back. All you going, I feel, hope all you running good to go. Well, these last 3 hours and so, what we're gonna do, we're gonna finish module 3. We're going straight, no breaks. No gas. All gas, no breaks. All gas, no breaks. Yeah, man. Okay. The playlist. I gotta link them after. I just well, this one called, I just download a whole set. This one called what all them things be called 1 a.m. study session, low file chill beats, and the other one from a is the soundtrack from a visual novel called Valhalla. It is be a bartender in a game, virtual bartender, whatever. Anyway, so all gas, no breaks. We finish in module 3, and then we'll go through a few past papers, because remember, we don't have to go through the whole past paper. We just have to go through the broad topics, so that shouldn't take too long. And whatever we don't finish, or whatever you feel you miss, I have a good bit of past paper here has been done on other videos. Those of you who are new to the channel, you could just look through the videos, and you're bound to see a playlist with keep Unit 1 past papers. And next year, I'll have Unit 2 past papers there as well as a speed run of the syllabus. Anyhow, there we go. So, Module 3 involves plant reproduction and animal reproduction. Plant reproduction, in my opinion, is a breeze. Animal reproduction could be a little more difficult. There used to be a third part, asexual reproduction, but that has gotten cut down to just having asexual reproduction being included in plants, which is just the different ways in which it could be done, like cuttings and tissue culture and things like that. And just for you to note the differences between sexual and asexual reproduction in terms of production of offspring, the difference in gene pool, passing down advantageous traits or deleterious alleles, things like that. So we'll go through all of that, but the stuff with like conidophores and conidospores and hyphae and mycelium and all kind of thing, which usually is involved with asexual reproduction of fungi, is no longer there. And yeah, that's right, Jonathan. Cyberpunk bartending action Valhalla. It is a pretty good game for what it is if you enjoy visual novel type games. Right. So let me get started here. So first, reproduction in flowering plants, basics. We have the male part of the plant, the female part. Sometimes the male and the female is on the same plant. Sometimes you have male plants and female plants separate. Regardless, the male parts of the plant produce pollen, so that the pollen are found with contain the male gametes. The pollen is not the gamete, right? The pollen is kind of like, think of it as the follicle for the gamete. It contains it. So the male gametes are formed within the pollen, and these are found in anthers, and anthers are attached to the ends of stamens. Easy to remember, stamen, men, male, right? So that's the male part. And from what we could see here, we could see that this is the anther. And typically the anther has 4 pollen sacs in it. Like if you was to cut it in a transverse manner. The stamen is also called the androecium. Andro typically means is a prefix for male. That's actually the term android. It's kind of like a male robot, whatever. But andro typically means male, and gyne or gyne means female. So the gynoecium is the female part of the plant, the carpels. And I don't know if you all know the word androgyny. That's when a man just kind of look like a woman, or a woman look like a man. It basically blur the lines, But andro is the prefix for male, gyno is the prefix for female. Anyway, female gametes are found within embryo sacs, and these are located within the ovules. Of course, the ovules are located inside the ovaries. This is the ovary here. This is the style, which leads down to the ovary, and atop the gynoecium, we have the stigma, right? So pollination occurs when pollen from an anther is deposited onto the stigma. So this could be within the same plant or the same flower, and this is called self-pollination, or it could be between 2 different plants, and this is called cross-pollination. Fertilization occurs when the male gametes from the pollen fuse with the female gametes of the ovule. So same thing with animals, is basically the fusion of those 2 gametes to form a zygote. Easy. So let's get a little more cape level into this, as more C-sec level. Let's go 1 level below. So here we have the male parts of the flower. And here we see a section. It actually supposed to have 1, 2, 3, 4, but this diagram is cut just to show the 2. But this is a micrograph of the anther, and I have a feeling like this is what they might ask it to draw. They might bring a micrograph of it and tell it to draw a plan diagram of it. If it's not the stem, if it's not the roots, it will most likely be the anther. It will be one of those if they bring in a plan diagram, because none of the other topics involves anything with a micrograph, because the other topics is enzymes, natural selection. Well, I guess with the animal reproductive system, you have the seminiferous tubules and the ovary, but it usually is the anther, the stem, or the root. So one of those. Anyway, so in the male parts of the flower, look at what we have here. We have 4 pollen sacs. Within those pollen sacs, we have pollen grains, and the pollen grains are formed from microsporangial cells. You can think of those as the precursor to the pollen grains. And these are formed, remember, this is within 1 anther. You'd have 1, 2, 3, 4. Now on the outer part here, we have the fibrous layer, and just as it says, it's just a thick on cellulose wall. It's mostly used for mechanical support and protection. On the inner layer of the pollen sac here, I'll run through the plan drawing when we go in through the past papers, because one of the past papers have it. The tapetum, weirdly, is Latin for carpet, right? So I don't know if they name it that because in a microscope it does look like a carpet. I don't know, but the tapetum provides nutrition to developing grains in the pollen sac, and this is kind of like the inner layer inside here. So these cells here, and I'll just make this a little bigger so you can see it. That's correct, Nairobi, yes. So it might include cell structure there too, so that could also come. So this is the tapetum here. These basically provide nutrition for these developing pollen mother cells. The butt start back, boy. Right. So you see here between two pollen sacs here, we have a stomium. So the stomium is the point of dehiscence. You might be like, well, what is dehiscence? Dehiscence is basically the point, this is the splitting of the anther to release the pollen. So this will basically break apart. Here the membrane is a little thinner, and this will break apart, and the pollen grains will get released into the atmosphere as a result when the stomium splits. You don't have to really know that much about it. And inside here we have the pollen mother cells. So the pollen mother cells are actually diploid, and these here would then undergo meiosis, and these would form gamete nuclei. So these pollen mother cells would eventually form what would be the male gametes within here. Simple. We're going to get into that now. So what happens during the formation of pollen? Well, the formation of pollen grains occur when a pollen mother cell undergoes meiosis. So here we could see this is a pollen mother cell. Another name for it is a microsporocyte if you want to be fancy. And this is a diploid cell. So you remember from mitosis and meiosis, when a cell undergoes meiosis, we get 4 daughter cells. And these 4 daughter cells here, they are genetically varied. So these 4 cells would be genetically varied. Each one of these haploid cells is called a microspore, right? So each of these is called a microspore. So this pollen mother cell gives quote-unquote birth to 4 microspores. Eventually, they will separate when cytokinesis happens, and they will undergo mitosis. So the strange thing about mitosis in these is that cytokinesis doesn't occur, right? So I know there's a word for that when cytokinesis, but it evades my mind right now. But what happens is that the nucleic material splits, right? So you get a kind of nucleic fission, but we don't actually get fission of the cells itself. So it's still 4 cells, but each one now contains 2 nuclei, right? You could think of it as 2 of them contain 2 different sets of cell material, but it's still 1 unit that contains these, and these are mature pollen grains. So to go through that one more time, we have a pollen mother cell is going to undergo meiosis, form 4 haploid varied daughter cells. They're going to separate cytokinesis, and then they're going to undergo mitosis. When they undergo mitosis, cytokinesis doesn't happen here. Instead, you just get the genetic material, the nuclei and so on being split, but they don't separate into 8 cells, let me say. You still have 4, but each one has 2 nuclei. Let's take a closer look into that. So this is one of those, these are one of those inside those microspores that would have undergone mitosis. And this would be what we would call a pollen grain. And there are 4 different things that we have to take into account here, right? Now the thing is pretty much 2 cells, but the one is kind of like a Venn diagram where one is within the other one, if you want to understand what I mean. One cell is actually within the other cell, right? So it's strange. It's not like they separate. It's like one of them is within the other one, kind of like endocytosis. But anyway, so the outer layer of this is called the exine, or of course we know the prefix exo means outside, and then we have the intine, which is the layer right below that. The exine is a waterproof wall, again is mainly used for mechanical protection, and the intine is an inner wall with enzymes that will help to break down any material, basically to help with general cell activity and regulation and things, right? You don't have to know much about it. You really just have to know how to label it. Now, the parts that we have to take note of now are these 2 nuclei. We have the generative nucleus, which is within this generative cell here, and this is actually within another cell called the pollen tube cell. So how you could tell the difference, by the way, between them, and I don't have a micrograph here, but we'll get into one later. The generative nucleus is a lot more elongated, and the tube nucleus tends to be rounder. That's actually how you could tell just by observation. So the generative cell, these are going to form the male gamete nuclei. So these are actually going to undergo mitosis again. And remember, 2 of these are haploid, by the way, right? 2 of these only have 1 set of chromosomes. So this is going to undergo mitosis, and that's going to form 2 haploid nuclei, right? And then you have the tube nucleus. So the tube nucleus, or the pollen tube nucleus, this is going to grow the pollen tube to deliver the gamete nuclei to the embryo sac. The embryo sac, if you remember, is within the ovule, within the ovary, right? So the ovule, the embryo sac contains the female gamete, which is the egg. So if I had to put it in terms of, let me say, a human analogy, this would be, let me say for example, a sperm cell. This would be the sperm cell. The pollen tube would represent the part that the sperm cell has to swim, and the ovule and the ovary would be kind of like the oviducts and the uterus within a human. So in the case, the difference with sperm cells and these these pollen nuclei, these pollen gametes, is that these don't have flagella, they can't swim. They require chemicals, they require this pollen tube to grow down the stigma and the style, and then into the ovary, into the ovule, and deposit the male nuclei there. If I had to use a human analogy to do that, it would be like, imagine like the man, while he's copulating, his penis would have to grow past the cervix, past the uterus, into the fallopian tubes and deposit the sperm cells there. Of course, that's not what happened, right? Because the sperm cells could swim. There's no need for that. So the pollen tube is kind of like a pollen penis, so to speak. But it could grow and it could reach the female gamete. So that is the male parts. The female parts of the flower tend to look a little more complicated. And we have a lot of things here to look at. Thankfully, there's only really a few that we really have to remember. So first, we have, and let me make this a little bigger for now. Alright, let's see how this. I feel I can make that bigger without blocking all the words. Alright, I feel like I gotta stay so. Okay, so this here, this whole thing is the ovule. This entire thing is the ovule, right? And this part here, this dark green part, this is the embryo sac. This dark green part here is the embryo sac. The stuff around it makes up the ovule, right? Of course, the whole thing is the ovule. But only this dark green part here is the embryo sac. First, let's look at stuff that is not in the embryo sac, right? So we have a placenta. The size matters as it pertains to reproduction in humans. That depends on the individual. I can't speak for everybody. Anyway, so the placenta is just the point of attachment for this embryo sac, and this is actually where it will get some nutrients and things like that from the parent plant. Not that important right now. It's just there to help to maintain the seed structure and function. The funicle is a stalk-like connection. Remember, the placenta would be attached to the ovary. The funicle is kind of like the umbilical cord. If you want to think of it like that, it connects the ovule to the ovary. Now you have to remember now that this ovule, when it fertilizes, this becomes a seed. And that seed now will would eventually be distributed by wind or water, animal, or self-dispersed or something like that. So think of the funicle as the placenta, as the umbilical cord. Here we have the integuments. The integuments would eventually form what you call the testa or the seed coat. This here is just really for protection of the embryo sac. These just wrap around and they're just used for support really. Now within the embryo sac we have real things here. So we have two ends of the embryo sac. We have the chalaza and we have the micropyle. So this side we call it the chalaza end, and this side we call it the micropyle end. The chalaza, by the way, chalaza is something that is actually found in multiple things. I don't know if you ever crack an egg, and the egg have these little stringy things inside that attach to the yolk. You exactly call that a chalaza. But in this case here, chalaza just kind of refers to, it just refers to the to the opposite end. Chalaza actually is Latin for on the opposite. Not it actually is Latin for not on the opposite foot. I think it's Latin for opposite. I'm not sure, but it just refers to the opposite end, to the micropyle. Nothing that you really need to remember. The micropyle is, you could think of this as if the pollen tube was a penis, the micropyle is like the vagina. The pollen tube is going to grow into the micropyle, and it's going to deposit the male nuclei into the embryo sac from there. So the micropyle is just like an entryway to get into the embryo sac. So I say we have plenty of things here. Now let me look inside the embryo sac, because inside this embryo sac, there are plenty of things inside here. So we have 8 cells in here. Well, I shouldn't say 8 cells, 8 nuclei, right? 8 nuclei. There should be 8 nuclei. Right, so it has 8 nuclei inside the embryo sac. We have 3 antipodals. The term antipodal means on the opposite foot, because anti means opposite, podal comes from pedal, which means foot. So antipodals just means on the opposite foot. So you have 3 of these, and all of these are haploid. We have an egg cell, which is in the middle of 2 synergids. So these synergids here, how I like to describe them is like, imagine like if you're a fella and you're like a girl, sometimes it's kind of hard to just approach the girl. So you need a wingman now, you need 2 wingmen to help you out. Synergids kind of like that. Synergids does help this sperm nuclei reach the egg cell. So it's like the female version of wingwoman, I guess. So the synergids is kind of help out the egg cell. So they would kind of guide the sperm nuclei not to break. They need guidance. So what happens is that the sperm nuclei get deposited. So they will get deposited around here. And the egg cell right there. But sometimes they would make it here and they can't really reach the nucleus. What would happen here is that the synergids secrete chemicals to help to guide the male nuclei away from the central cell and towards the egg cell nucleus. So that's what the synergids do. And synergids work in synergy with the egg cell. Of course, synergy means it means like to work together. So the part that could get a little confusing here is that this middle part here is actually a huge cell called the central cell. And we have 2 nuclei in here. Each of these is haploid, and they are called polar nuclei. Sometimes they count them as a diploid, but they are actually 2 separate nuclei. So these are called polar nuclei. So if we count here inside the embryo sac, how much things we have? We have 1, 2, 3, 4, 5, 6, 7, 8. And that accounts for 8 haploid nuclei within the embryo sac. 3 antipodals, 2 synergids, that's 5. 1 egg cell, that's 6. And 2 polar nuclei, that is 8. And those polar nuclei within the central cell. There are other parts here that I didn't get into, like the nucellus. We don't really need to go into those, honestly. They just kind of there for just extra, that's just extra knowledge. So if you went through that, that's fine. You wouldn't get that. Alright, so coming back here now. So just to recap, the antipodal cells, they nourish the embryo sac. So these here absorb nutrients from the chalaza, and the chalaza is kind of like a nutrient dense portion of the ovule, and they would absorb nutrients from that, and thus they would actually provide nutrition for the rest of the embryo sac. The synergids direct pollen tube growth to the egg cell, and the polar nuclei. Now remember, both of these put together are diploid, right? Now eventually, remember I said it are 2 male nuclei. So one of them is going to fertilize the egg cell, and that there is going to form a zygote, and one of them is going to fuse with these 2 polar nuclei. Now, remember, say the polar nuclei is 2 haploid cells. So when you get a next haploid cell fusing with that, you end up with 3N. Right? You end up with a triploid nucleus, and that's actually what forms the endosperm. We'll get into that just now. But that's basically what happens post-fertilization. So we haven't reached there yet. I kind of getting ahead of myself. Right now, all you have to just remember, this is the embryo sac, which is contained within the ovule. And there are eight haploid nuclei in here. 1 egg cell, 3 antipodals that nourish the embryo sac, 2 synergids to guide the pollen tube towards the egg cell, 2 polar nuclei which will get fertilized and become an endosperm. Dominique, the chalaza is just the end opposite to the micropyle. The chalaza is just a nutrient-dense part of the ovule that sends nutrition to the antipodal cells. I have no idea whose face, and I'm not coasting on you. Honestly, I don't even know who he is. But what's going on? Hope you're good. Try to be less annoying on the chat. Alright, so formation of the embryo sac. This is what happens here. So this is something that could be very confusing. But I'm going to explain it step by step. And you'll see that it's not very hard, honestly. So, all remember, let me come back to the male part again, right? So the male part, remember that this has a pollen mother cell. And this undergoes meiosis and splits into 4 haploid cells. These then separate and so on. But you end up using all 4 because remember, male gametes, you have to produce plenty of them, kind of like men have to produce plenty sperm cells, right? They expend energy to produce a lot because we don't know which ones are actually going to pollinate what, which ones are going to fertilize what, and so on. So, for the female parts is a little different. We're going to start off with 1 diploid megaspore mother cell. Just like how we had a diploid pollen mother cell, right? The megaspore mother cell is going to undergo meiosis. And you're going to produce 4 haploid cells. Now, this is what happens now. This is where it gets a little different. It forms 4 separate haploid cells, right? It forms what you call a tetrad, which is 4. But 3 of these will degenerate. And only 1 will survive. So maybe like, well, why expend energy to produce 4 if, you're just going to help grow 1? Well, basically to explain this, you have to think about it like this. In most nature, whether it be plant or animal, the female is the one that has to think of it like they have to nourish the young. And if you have 4 young, 5 young, 6 offspring within, you have to nourish all of them. That's a lot. So having 4, imagine 4 of them get fertilized per ovule, that's too much. It makes them less likely to survive because you would have to split the nutrition between the 4. So what it does instead is that it ends up depositing most of its nutrients to the top one. Sorry, not the top one, the lowest one. And that there grows. And as it grows, it sort of pushes the other 3 out. And as a result now, you end up with 1 fully developed haploid cell. And the rest of them just die off. Is kind of like that meme. I don't know if all you know the meme. Let me see if I could find it. It have this child in a pool and the mother holding up the child. Let me see if I could find it. Drowning. The mother holding up one child and the next child drowning. So let me see. right? So it's kind of like this. Imagine like this is the megaspore mother cell. These are the 3 other haploid cells. You just kind of leave them to dead. And this is the one surviving haploid cell. You kind of give this one all your attention, and you kind of give this one all your love and thing. And these die, and well, yeah, they get forgotten. That's the meme. You can think of it like that. So only one of them actually becomes viable or functional. So this one haploid cell now, it takes in nutrition, it takes in amino acids, it takes in sugars, and what happens there, it begins to grow and develop cytoplasmic material and all kinds of different protoplasm. And remember this one nucleus here, right? This has to eventually form 8 nuclei here. Remember, in the embryo sac we have 8. So think of how much times they need to divide by 2, or sorry, multiply by 2 to get 8. Well, first we have one round of mitosis that's going to produce 2 nuclei. Our next round that's going to produce 4, and our next round that's going to produce 8. And eventually now this is going to specialize, and they're going to move apart and differentiate into the antipodal cells, the egg cell, the synogens, and the polar nuclei. And within this, you're going to have a central cell in the middle here. And the polar nuclei are going to float within the cytoplasm of that central cell. So, this is, and you have to know this process, right? So, we have meiosis first to make it haploid. And remember, all of these are haploid cells. Then you have 3 rounds of mitosis that's going to, remember, it will stay haploid after with mitosis because they will be identical at first. So we have 1 to 2, 2 to 4, and 4 to 8. And then we have, you have some differentiation and cell specialization. Basically, you have a bunch of hormones and things that are going to interact to allow these to drift to the poles of the cell, Eventually, you're going to have maturation and a mature embryo sac. And that there is the formation of the embryo sac. The key thing to remember here is that even though it undergoes meiosis, 3 of them degenerate and only 1 of them really survives. That one is the one that becomes the 8 nuclei in the embryo sac. Easy. Okay, so let's look at pollination. So pollination is the transfer of pollen from anther to stigma. And this could occur within the same flower. It could occur within 2 flowers of the same plant, or it could occur between 2 flowers of 2 different plants. Usually this is done by insects such as bees or butterflies, or it could also be done by wind. You want to do that in CSEC. It depends on the type of flower. So self-pollination occurs within the same flower or between 2 flowers of the same plant, cross-pollination between 2 flowers of 2 different plants. Okay, so this is where it does have a little bit of confusion. Even people going in to write the exam just get us confused. Self-pollination is not the same thing as asexual reproduction, right? Because we still have fusion of gametes, we still have fertilization, we still have post-fertilization structures like zygotes and fruits and seeds and things like that. So it's not asexual reproduction. We still have 2 sets of male and female gametes and male and female reproductive organs being involved. So it's still sexual reproduction, even though it's within the same organism, because it still have fusion of gametes. So you still have meiosis taking place, and you still have synapsis and recombination and all kind of thing. Self-pollination limits the gene pool, but it doesn't limit it as much as, let me say, cuttings or fragmentation or budding. So it's not really considered asexual reproduction. I know it sounds weird, but yeah, it's not really considered that. And that's purely because you have meiosis and you have gametes involved. So you could count it as 2 different processes. Now, what happens during pollination? So rewinding a little bit, we talk about the pollen grain, right? And the pollen grain, I said, has an outer layer called an exine, an inner layer called an intine, which has digestive enzymes. There's cytoplasm. And remember I said that it undergoes mitosis, when the microspores undergo mitosis, they form two nuclei. They have, this is the pollen tube nucleus, and this is the generative nucleus. Sorry, this is the pollen tube cell with a pollen tube nucleus. This is the generative cell with the generative nucleus. And if you remember here, was say, if you're just looking at them, how can you tell the difference? The generative nucleus is more elongated, the tube nucleus is more round. So the generative nucleus divides by mitosis. So this thing here divides by mitosis and forms two male gametes. Of course, that would be, these are both haploid, so they would form two haploid male gametes, and these are what you call this, the pollen sperm nuclei. They still call them sperm. But you could just call them the male gametes if you want, either one. If you don't want to confuse human sperm cells with pollen sperm cells, you could just call them the pollen male gametes. So the tube nucleus uses digestive enzymes to elongate and grow down the style to the ovary. So remember I said, if I had to use a human analogy, if the humans could do this, this is like the man, if the man was copulating with a woman, if the man penis could have grown through the cervix, through the uterus, and into the fallopian tubes, that's kind of like what a pollen tube does do. Of course, men don't do that because sperm cells have flagella, right? So it never had a need. So how does it do this? Well, the tube nucleus uses digestive enzymes to elongate. It basically takes like I believe is think of it like a cocktail of sucrose and glucose and auxins, and that basically encourages growth, and it grows down the style to the ovary. So this will grow in a downward manner, and the two male gametes are going to follow the pollen tube into the embryo sac. So this is the tube nucleus still here, and these are the two male gametes that are going to be flowing through this pollen tube, and the chemicals are going to guide this for it to move along. The name of that process is chemotaxis. I don't think you need to know that, but taxi means to move, so chemotaxis means movement via chemicals. So, double fertilization. This is something that you would have done in CSEC level, I believe. And this is basically what's happening here. And I'm actually gonna show you a video. I think I have something on it here. Let me see if I could find. It might be a little easier to picture. Okay. So here, so this is inside the ovary, right? And inside one of the ovules, this is the embryo sac, where we're going to have 8 haploid nuclei. Alright, so again, mitosis, and this is where we have our 8 haploid nuclei that's going to form the embryo sac. Now remember, on the calyzal end, that'll be on top here, we have the 3 antipodal cells. In the center here, the central cell, we have the 2 polar nuclei. And down here, we have the egg cell, which is a lot larger than the 2 by its side, flanking its side, called the synogens. Cell growth is going to occur, and each one is kind of going to get encased in its own little cell body there. So, the one in red is the egg cell. This is your female gamete, and this is, remember, all of these are haploid. The exception being these 2 polar nuclei. If we was to take them together, they would be diploid, but they're still made up of 2 haploid nuclei. So, what is happening here is when a pollen grain gets deposited from an anther. So this is the anther here. And the pollen grains get released. So remember I said in the center here we have the tube nucleus and the generative nucleus. The tube nucleus is what helps grow the pollen tube. The generative nucleus will undergo mitosis and release 2 male gamete nuclei. Think of them as 2 sperm cells. So we have a tube nucleus leading the way, and these are 2 male nuclei here, and they're flowing down, right? So we have 2 haploid cells, and this is why it's called double fertilization, because each one of those is gonna fuse with something inside the embryo sac. Mitosis completes along the way, so now we have 2 haploid male nuclei. One of them is going to fuse with the egg cell, and this is going to become a zygote. And the second one is going to fuse with the polar nuclei. So here we have a diploid cell, the zygote, and this would eventually grow to become a new plant, right? This is like a baby plant, the little embryonic plant it will grow into. This will form a triploid nucleus, which means there's 3 sets of chromosomes. And this here, remember, this here is the pollen tube. The pollen tube grows into the micropylar end, and one forms the zygote, the next one forms the endosperm. So the endosperm is kind of like the placenta of the seed, if you want to think of it like that. Is the food storage. I'll give you an example. If well, my favorite example is this. If you ever drink coconut water, the coconut water is the endosperm material. So you're drinking plenty endosperm, you drink plenty coconut water. And those nutrients in the coconut water, that was meant to nourish the coconut seed. So we end up with 2 things getting quote-unquote fertilized, with proper fertilization of the egg into the zygote, and the polar nuclei, the diploid central cell, turns into the endosperm. Easy. Right, and the endosperm together with what would eventually form the cotyledons. The cotyledons would be, it depends on the plant, but the cotyledons would also be a food storage. Right, so once again, one haploid male nucleus fuses with the haploid egg cell to form the diploid zygote, and this would eventually develop into an embryo. That embryonic plant would eventually grow like a radicle and a plumule, and eventually grow into a seed, from a seed into a seedling, and eventually into a mature plant. So one male nucleus fuses with the polar nuclei to form the endosperm nucleus. And there's a triploid because it has 3 sets of chromosomes. This forms the endosperm, and that will provide nutrition. As I said, it's like the placenta for the seed. The reason why you need that endosperm is because the plant, remember, the seed is not connected to the parent plant, so it has to be self-sufficient. When you put that into the ground, it must have its own food source. Okay, so that's called double fertilization. Right, so coming back here now, really, post-fertilization, this is something that they like to bring as well. What happens after fertilization? But I think this came last year. After fertilization occurs, many changes occur in the flower. Usually you will be asked to name between 2 and 5 changes, and this is what happens here. The egg cell becomes a zygote, and eventually that will undergo mitosis until it becomes an embryo or an embryonic plant. The ovary becomes the fruit. The ovary wall becomes the fruit pericarp. So if you come back to the coconut here, the pericarp of the coconut is the coconut jelly. That would have been the ovary wall. It would be basically, the nice parts of the fruit, the nice sugary, fleshy parts of the mango, for example. The ovule itself, remember that contains the embryo sac that becomes the seed, still attached to the parent plant until it's ready for dispersal. The integuments which would have protected the ovule that becomes the seed coat, that's the tester, usually as it does there for mechanical support and protection. The endosperm nucleus, which would have been the polar nuclei, becomes the endosperm, and the petals wither and fall off. Typically, you're left with a little seed scar where you have the where the micropyle and the hilum used to be, but you need to take too much note of that. This is something it's on the syllabus, but as I was going through all the past papers, I never see anything come on it, so you never know. But this is what occurs during embryonic development of a seed. And here we could see, here's the zygote, and this is the fertilized egg cell. And this is eventually going to undergo mitosis, but it's not, the cytoplasm is not going to be, what's the word I want to say? It's not going to be evenly distributed. So it's not going to undergo like true well, it undergoes true nucleic division, which means that the nucleus has the same material. But in terms of cytokinesis, it only breaks off a little bit of the cytoplasm. So this part here that branches off, so imagine like the zygote here, and it divides and pushes out a little part here. This is called a terminal cell. So a terminal cell, these terminal cells keep forming, right? And on top here, this is called a basal cell. So this is the terminal cell and this is the basal cell. So eventually now these terminal cells are going to continue forming, and you're going to get a belt of them called a suspensor. And what would have been the initial terminal cell, this one here, eventually this will divide to form like a globular embryo. So if I had to put an analogy to this, I say a human analogy is usually the easiest one. Think of the suspensor as like an umbilical cord. The basal cell is kind of like a placenta, I guess. And the globular embryo is like, well, the embryo, right? It's like the fetus. So eventually now you'll have like a transfer of nutrients from this basal cell until, all the material runs out. So the first terminal cell keeps dividing to form a globular embryo. An early root or radicle and a shoot or plumule develops. This happens after differentiation, as well as some early leaves and storages called cotyledons. Not terribly important, but basically it's just saying that you start off with a zygote, it divides to form a basal cell and a terminal cell. Eventually that terminal cell forms a belt, and on the end of it you have an embryo that this keeps dividing by mitosis. And eventually this will differentiate into an early root or radicle, an early shoot or plumule, and cotyledons, basically food storages. Go back to the former slide. Take a screenshot, because we already discussed all this. 5, 4, 3, 2, 1. Let me continue. Okay. The last part of plant reproduction, not counting asexual reproduction, is outbreeding mechanisms. And I don't think this came last year. They love to bring this. They love to bring this stuff, outbreeding mechanisms, because they like to link it to the whole gene pool, making sure that the gene pool is big enough, making sure that natural selection could occur, speciation could occur. But basically outbreeding is the opposite of inbreeding. Inbreeding is, when you have, inbreeding is like self-pollination, where you have the same flower fertilizing itself, right? That'll be like inbreeding. Outbreeding is kind of like this. It's kind of like making sure that one flower, the pollen from one flower, could reach the stigma of another. Basically, outbreeding ensures that cross-pollination occurs. right? So think of it like that. Outbreeding mechanisms is to ensure that the pollen don't just stay home. The pollen have to go out and reach a next flower. That is outbreeding. So what it does is that it ensures genetic variation and diversity because you have the mixing of alleles, you have genetic recombination, you have different traits being met, you have a whole set of different characteristics coming together, and you also have the chance of speciation. This maintains genetic vigor, keeps the gene pool wide enough in the species to ensure survival, and say again, it aids in speciation. So there are 4 mechanisms that, well, I should say 5 mechanisms that we're going to look at, right? Self-incompatibility, male sterility, dioecious plants, protandry, protogeny, and heterostyly. Typically, these first 3 are the ones you're going to see pop up more often. Well, I should say these first 4. Now, keep in mind, when I am discussing these, outbreeding means outbreeding means cross-pollination and cross-fertilization. It means it has to be 2 different plants. Not just 2 different flowers, 2 different plants. Whereas inbreeding means self-pollination and self-fertilization, whereas typically the same flower or 2 flowers of the same plant. So, are there any benefits to I'm gonna assume now you say inbreeding. There could be, if you want to preserve certain advantageous characteristics. You have certain types of flowers called cleistogamous flowers, like tulips. They're kind of closed in, and those kind of help to keep pollen inside so it could promote self-pollination. Some tulips are bred like that, but out in the wild there's no advantage for inbreeding. There's no genetic advantages to it in the wild. So, let's start with self-incompatibility. What are you looking for? A song? It's supposed to have something playing. Okay. Self-incompatibility is this. Self-incompatibility ensures that pollen of the same alleles of a plant do not germinate. Right? So these are typically denoted by these letters or numbers here. S1, S2, S3, and S4. There are different types, of course, but using these as examples here. Let's say that the plant is an S1, S2 plant, right? That means that it can either release pollen that is of a S1 variant or an S2 variant. Let me repeat that. Let's say that our plant has a genotype of S1, S2. It means that it can release pollen grains that either contain S1 alleles or S2 alleles, right? Basically, the plant has a mechanism built into its stigma that prevents S1 pollen grains and S2 pollen grains from growing a pollen tube. So let me look at this example here. Here we could see an S1, S2 plant. Neither a S1 pollen grain or an S2 pollen grain is going to be able to grow a pollen tube here. Now let's look at this plant here. This is a S1, S2 plant. S1 pollen grains are not compatible. This is why it's called self-incompatibility. Its own gametes cannot fertilize itself, but it can fertilize something that is not an allele that is in its genotype. So S3 pollen grains would grow pollen tubes. My throat, a little bit, but I good. S3 and S4, right, are fully compatible because they are not of the same genotype as the plant. So this S1, S2 plant could accept S3 pollen grains, S4 pollen grains, S5, S6, S7, S8, S9, just once it's not S1 and S2. Once it contains the same alleles, it basically shuts off the hormones that encourage pollen tube growth, so the thing cannot, grow pollen tubes to fertilize the egg cells. Male sterility ensures pollen grains are not produced or are not viable. So basically the plant shooting blanks. So if male sterility occurs, pollen grains would not be fully formed, or the gametes would not be fully formed. It's kind of the same as a man if he can't produce sperm cells. That's male sterility. Simple as that. Of course, this is usually found in plants with male and female parts. It means that the female parts are operational, but the male parts are not, which means that it can only be pollinated by another male plant, not itself. Right, so here we have some other terms here. We have dioecious plants and monoecious plants. Let me explain what these mean. The term monoecious means one house, or monoece means one house. Dioecious means two houses. So how does that relate to the mechanism that we're talking about? Dioecious plants have male flowers on a single plant and female flowers on the next plant. So for example, one plant would only have stamens. It wouldn't have any gynosium. It wouldn't have any stigma and ovaries. The next plant wouldn't have any stamens. It would only have stigma, style, and ovary. They would only have female parts. So in this case here, it is impossible for the plant to pollinate and fertilize itself because it only has either only male parts or only female parts. An example of this would be cannabis. Cannabis plants have, some of them are just male, these are male parts, some of them are just female. Monoecious plants is not an outbreeding mechanism, but this is when the plant has both male and female flowers, and as a result, self-pollination is possible. How I could put it like this, boy? Monoecious plants is like your Shogunus senior secondary, or your Woodbrook, your Pleasantville, your Couvasec. They are the male and the female in the same house. They have them in the same place. Your dioecious plants is like your Ashja. You have your Ashja boys and your Ashja girls. You have your Naps boys and your Naps girls. That's dioecious. You have males to one, females to the next one. Protandry and protogeny. So let me break down the terms. Pro or prot or proto means before. Remember, I say andro or andry means male. Gyno or gyne means female. So protandry just means the male parts mature before the female parts. In the case here, the stamens may mature before the stigmas become receptive to pollen. So it just means male parts mature before. So the males and them could produce pollen, but the stigmas would not be able to receive that pollen. As a result, self-pollination and self-fertilization becomes impossible. Protogeny is the reverse. Protogeny is where the female parts mature before the male parts. So the stigmas become receptive to pollen before the stamens could even produce pollen. So if you think of it like this, if we just had to think of it simply, protandry is like the man wake up, but the woman still sleeping. Protogeny is like the woman wake up, but the man still sleeping. And all in all, they can't do nothing in sleep. That's rape, right? So that's basically what it is. It's simple terms, but more complex terms, if you had to write it. Protandry is when the stamens mature and are able to produce pollen before the stigmas are receptive to that pollen. And protogeny means that the stigmas are receptive, they mature and are receptive to the pollen before the stamens can even produce pollen. And these are the outbreeding mechanisms. They love to bring this. Now, the last one here. Right, so we get into pin and thrum now. So this is the last one. We had this last one. This is heterostyle. Hetero, as you know, means different. Style refers to the style, like as in stigma, style, and ovary. But it could also refer just to the carpels in general. So heterostyle means that the shapes of them are incompatible with each other. So you have various forms of flowers that makes it difficult for self-pollination to occur due to the stigma and the anther position. It is difficult for pin flowers to self-pollinate. So if you look at the pin ones here, right? Look at where the stigma is. Do you see how high it is? And look at where the anther is. So it is very difficult for the pollen from the anthers to get deposited onto the stigmas here. Typically, the anthers and the stigma would be out, and the anthers would, let out release pollen, which would deposit on the stigma. Because the anthers are so low down here, it's unlikely that they would come into contact with the stigma. Whereas for the thrum flower now, self-pollination is a little more likely because the pollen could just fall on the stigma here. Right? So the pin flowers are what really demonstrate this outbreeding mechanism. So that there is the outbreeding mechanisms. So to recap, just the basics of what you should know, because I believe that this would come. I just have a feeling. It's like this or that plant diagram for the anther. Who knows? Learn the definitions of all of these and learn why outbreeding mechanisms are useful. That's what we're going to get into now. So, what are the advantages and disadvantages of asexual reproduction? And basically you could think of these as the disadvantages for asexual reproduction would also be advantages for sexual reproduction. So the opposites of this I mean. So let me start here with the advantages. But I don't think this common because last year a 15-mark question come on this, so I feel this coming, but I'll still go through it. The advantages for asexual reproduction: the offspring can remain well adapted to a non-changing environment, as all traits are inherited from the parent organism. Basically, this means that once there's no big selective pressures, no big temperature changes, no invasive species, no pathogens, no urbanization, human intervention, anything like that, these organisms will do just fine once it's a non-changing environment. Raj put here, I have one. I'll link it a little after. It's kind of late for you to be reading through that book. This man really took my man not reading book for this exam. Anyway, where you might come across something. Anyway, the next advantage of asexual reproduction is that it encourages rapid growth of the populations. And let me say, for example, that it had a disease that wiped out most of the plants in an area, and those plants could reproduce asexually. Well, as a result now, all you need is one parent, and you don't need any gametes, so you don't have any gestation period or anything like that. These gametes, this one parent could bud and fragment and all kind of thing and produce a whole set of offspring very, very quickly. So you could replenish lost populations very quickly, and you could also colonize new habitats very quickly. The offspring may be able to utilize the parent as a nutrient source. So example would be like Bryophyllum, which forms little leaflets attached to the parent leaf. And because of that now, it could still be attached to it and use it as a nutrient source. It's not like a seed that has to be dispersed. The disadvantages of asexual reproduction. Well, due to the lack of genetic variation, pathogens and diseases may be able to spread through populations very quickly. Overcompetition may occur because all of these offspring and their parents would be very close to each other. There's no real big method of dispersal with asexual reproduction unless it's like fungi and things like that. But typically they just bud or they branch off, or leaflets fall, or you have little cuttings, which is human intervention, but typically these plants will be very close together and they will be sapping all the same nutrients. You also have very low genetic diversity, and as such you have reduced genetic vigor. And this can lead to less likely to undergo evolutionary change in these species. So these are typically 3 advantages and 3 disadvantages. Of course, the advantages of sexual reproduction or cross-fertilization is the opposite of these, where you would have high genetic variation, more protection against pathogens and diseases and environmental changes and selective pressures, less over-competition, and the plants would be more likely to undergo evolutionary changes and speciation and have an increase in vigor. How come I didn't go through the bacteria? I don't know what's the bacteria thing. May know that. But that wraps up plant reproduction. Now we go into animal reproduction. So animal reproduction, I know this is, I mean, I never really find it was hard, even when I do it long time ago. I know it's a big step up from what you do in C-Sec. Everything here is honestly a big step up. But I know a lot of people, they catch the tail with it. Spores not on the syllabus again. Your teacher might still go through it with you, but the whole formation of conadophores and conadospores and all kind of thing, it out of the syllabus. Alright, let me put on back the Valhalla music. Alright, that's now it. Plant tissue culture could come, but not so much the process of doing it. Just the mention of how it could produce plants very quickly, but those plants are highly susceptible to disease. Oh, natural selection with the back. Okay. Well, we'll come across we'll come across a past paper with that. Okay. Over here, Tolia. Strap in. Ready to take a wild ride with this animal reproduction. Ooh, here we go. Alright. Spermatogenesis literally means the formation of sperm cells. So, where does this occur? This occurs inside the lumens of the seminiferous tubules, and this is in the testes. So the lumens is like the passageways. Like, you know how your arteries have lumens for the blood to pass? The lumens is like a little gap in the tubule, it's like the middle part. And spermatogenesis occurs from the wall of the seminiferous tubules and branches off going towards the lumen. So, let me show you a picture here first. This here is the outer part of the lumen, the seminiferous tubule. This here contains what we call the germinal epithelium. Why are we doing animal? And animals is what? And that's one of the things. Animal and human reproduction. Tell me if I'm wrong. So the germinal epithelium is basically where these germ cells gonna form. Germ as in germinate. And this is where the sperm cells, you can think of them as this is where they originate, right? This is where they generate from. So the germinal epithelium is a set of diploid cells. And these eventually now grow and undergo mitosis to form spermatocytes. These are spermatocytes here. These are called secondary spermatocytes. Wait, a little loud. Okay, so we have primary spermatocytes, and these are diploid cells, right? And these would eventually undergo meiosis, and they would form secondary spermatocytes. These are haploid. Spermatocytes are just the precursors to sperm cells, spermatozoa. So these secondary spermatocytes are eventually going to differentiate, and they're going to grow a little acrosome, and eventually they will branch off and grow a flagellum. The centrioles will help to grow the flagellum, and these would form spermatids. And when they form a fully grown flagellum, basically when they reduce in size a little bit, they form spermatozoa. So this is how this works here. We start with the germinal epithelium, and those eventually grow spermatogonia cells. And these cells here, think of them as these are like the primordial cells. These would eventually branch off and become all these hundreds of millions of sperm cells. So these undergo mitosis and form primary spermatocytes. They undergo meiosis and become secondary spermatocytes and then spermatids. So meiosis 1 turns them into secondary spermatocytes. Meiosis 2 turns them into spermatids. Eventually, they are going to undergo differentiation, and they're going to grow a flagella, they're going to grow an acrosome, they're going to grow a middle piece, all these different things. They're going to get that nice little sperm shape you're accustomed with, right? And they're going to grow into spermatozoa. But basically, you need 2 rounds of meiosis to turn a primary spermatocyte into a spermatid, and then that differentiates into spermatozoa. So let me see here. Let me see if I could put that a little more simply here, like in a little flow chart. Okay, so think of it like this. We have spermatogonia. This is going to undergo mitosis, produce primary spermatocytes. Meiosis 1 forms second. So this is when it's reached secondary. This is haploid. Meiosis 2 finishes, then it forms spermatids. And then these will now differentiate and specialize from sperm cells with flagella and acrosomes and things like that. These sperm cells will then get released via ejaculation. And that is spermatogenesis. Now there's one part here that there's two things here that I left out. There are these cells that nourish all of these cells here, and these are called Sertoli cells or nurse cells, right? So these nurse cells help to provide nutrition for all these spermatocyte cells, all these spermatids and things. And if you really look at them, I don't know if you could see it, but it's this pink part here. If you really look at them, it looks like a large pink blob, and you can see it looking like it's the sperm cells suckling on the nurse. It's literally called nurse cells. It's like the nursing on it. So this is the nucleus of the Sertoli cell, and these help to nourish all these other cells here. Let me see if I could get a little visual to show you. Honestly, I don't find it so hard, but I know people just freak out. Alright, me and none. Oh, okay, I think I have something here. Right, so if you watch here, this is inside the testes. This is the male gonad, the testes. We have seminiferous tubules located all throughout this. These are subdivided into little lobules, aren't they? Not that important. And let me look at what it looks like inside. So this is the seminiferous tubule. This is the lumen. And this is the tubule wall that you're seeing here. The germinal epithelium is located right around the lumen here. These here are going to eventually give rise to spermatozoa, or sperm cells as we know it. So it's the main site of spermatogenesis. So if we used to look at a section through it, it would look like this. This would be the germinal epithelium. And these would be spermatozoa on top here. These here would be spermatogonia cells. Then you would form primary spermatocytes, secondary spermatocytes, spermatids, and then spermatozoa. And remember, these spermatozoa are haploid. So eventually along this point you undergo meiosis. Right. So this is what is important to remember here. One germinal epithelial cell, basically one spermatogonium, undergoes meiosis. After 2 rounds of meiosis, meiosis 1 and 2, you form 4 spermatids. All these spermatids are haploid. Forget all this other stuff here. Eventually, those will grow a flagellum, lose a little bit of mass, and become spermatozoa. That's all you have to remember. Now, what helps with that these are the spermatids. What helps this whole process are these cells called Sertoli cells. That's what you're seeing here. These provide nourishment. They provide nutrition. They're also called nurse cells for this entire process to occur. Right? Forget all this other. It is actually go deeper than this, but we need to go deeper than that. That's what it's for. That's all you have to know. Read the thing before the exam. I'll go tell all you. Maybe come back there. Alright. So, well, if all you're balling for spermatogenesis, we go ball a little more for oogenesis, because all you know the female parts of this thing does be real complex compared to the male. So let me go with the female reproductive system. So the female reproductive system, pretty easy in terms of the layout. We have the vagina, the cervix, which separates the vagina from the uterus. The uterus lining is also called the endometrium. We also have the muscle lining called the myometrium. The uterus branches into 2 fallopian tubes or oviducts, and this is typically where fertilization would occur. And we have on the ends of these, we have the ovaries, and the ovaries produce oocytes, which are the female gametes, and hormones such as estrogen and progesterone. These are also called gonadal hormones. So a secondary oocyte, remember not an ovum, is released during ovulation from the ovaries. So if fertilization occurs, the zygote is implanted on the endometrium. So when an egg is released, and if that egg gets fertilized, it implants on the endometrium, and that would eventually grow some extra embryonic structures such as placenta, umbilical cord, amnion, chorion, and would eventually form a fetus. So the endometrium allows the growth of structures that exchange materials between the fetal and the maternal bloodstream. More on that later. All right, Ollie. So oogenesis. This I come in a while now, so I had to be a little frightened. Oogenesis is one of the most complicated things you will probably encounter in this syllabus and beyond. It is because it is not a continuous process. It starts and stops at different parts in the life cycle. So first, let me start with inside the ovary. These things called primary oocytes are formed, right? These primary oocytes are produced when the girl, before the girl even born, right? So before the girl even born, while she's still a fetus, and now growing ovaries, these primary oocytes gonna start to develop inside this little fetus girl ovaries. And typically you will have all of them, right? So if here's a girl, right now, all them eggs that you have in the ovaries, that was there since before you're born, is only one of them does mature every 28 days, basically once a month, every menstrual cycle. So, before you're even born, you have a bunch of these primary oocytes inside the uterus. Not inside the uterus, inside the ovaries. These are diploid cells. There are 46 chromosomes, right? Talking about humans. Now, there's the thing. Before you even born, right? So if you look here, before birth, meiosis gonna start. Form these primary oocytes. And as you're born, that meiosis is gonna stop. Right? We don't need to memorize which phase it is stop as. It is stop at prophase 1. So check the timeline so far. Check the timeline. Before you're born, you produce all these primary oocytes. Meiosis does start, right? But it is stop. It just start, but stop. When you're born. When does it continue? Well, think about when you get your first period. When you start to ovulate. Puberty. So there's the confusing thing. The things start before you're born, then it suddenly stops, then you reach 12, 13 years old and it start back again. So that's the human body. Yes, the human body could be a little weird. It have all kind of things that does go off with the human body. So, before birth, the things start, as soon as you're born, it stop. When does it start back? Puberty. Puberty now is kind of like your body remember, hey, as a female, I need to make eggs. It's kind of like your body now remember that. As a girl, I need to make eggs. So upon puberty now, meiosis is going to continue. And this is where you're getting here. It's going to continue and it's going to form a secondary oocyte. The secondary oocyte is your gamete. This is what we call the egg cell, right? The egg cell gets, encased on a little follicle, it gets released during ovulation, and if it fuses with a sperm cell, it becomes a zygote and then an embryo and a fetus. This is your female gamete. So, you might be like, what the hell is this thing? Well, this is called a polar body. Now remember, I said this primary oocyte has 46 chromosomes. The secondary oocyte is a gamete. So this would have 23 chromosomes, half the amount. So where the other 23 gone? The other 23 went into this polar body. The polar body is a chromosomal dustbin. This is just a place to throw away the other 23 chromosomes. So once again, let me use this meme to explain. There's your ovary. This is a secondary oocyte. This is a polar body. Nobody cares about the polar body. It's just there to get rid of the extra 23 chromosomes. So this is the ovary, giving all of its loving, tender care to the secondary oocyte because this is the gamete. This is what will eventually or could eventually get fertilized. This is the polar body, which is there to throw away 23 chromosomes. It don't have any use. It's not viable. It cannot get fertilized. It don't have enough cytoplasm. It don't have enough food storage, anything. Is literally a garbage can for chromosomes. So, what will happen now? This polar body now could eventually finish up meiosis and become two polar bodies. Basically, remember you have 1, 2, 3, 4 daughter cells here. But who cares? Nobody cares. This could happen, it don't happen, it really don't matter because this is just a chromosome trash can. The secondary oocyte. So this is where this is get confusing now. Let me look back at this diagram here. The secondary oocyte, as I said, is your female gamete. Now I said in C-sec biology, they lie to you and tell you that the female gamete is the ovum, but the ovum is actually something totally different. So this is where this gets confusing again. The secondary oocyte do actually finish meiosis until a sperm cell fuses with it. That's strange because we know when a sperm cell fuses with an egg cell, right, it becomes a zygote. But actually what happens is that when that sperm cell fuses with that egg cell, it becomes an ovum and then a zygote. So this is what happens here. Starting from the top. A diploid primary oocyte. These are formed before you're born. After puberty, meiosis finishes, forms a secondary oocyte. This is your gamete, and a polar body. This is just there to get rid of the extra 23 chromosomes. So whether or not this finishes meiosis or not doesn't matter. All of these will degenerate. This doesn't matter. When fertilization occurs, basically when the sperm cell fuses with the oocyte, the secondary oocyte, meiosis completes, and then it becomes an ovum, and then it becomes a zygote. And what it does, it throws off a second polar body because meiosis is now complete. And as a result now, you end up with eventually a zygote and 4 daughter cells, 3 of them totally not viable. 1 of them would eventually become a zygote. There's plenty all here. There's plenty. And it really have no simple way to explain it. So I could tell all you, look up some things, read up some things before you come. If we sacrificing, honestly, it might come, it mightn't come. We go see. Okay. So some of these things here, I'm kind of just gonna skim past. You could read the stuff yourself. I'll just go through briefly because I wanna do the past papers. Learn the difference between spermatogenesis and oogenesis. Learn the sperm cell structure. this does come a lot of the time too, so it'll be nice if this comes. You should note the different parts, such as the head, the neck, which contains the centrioles, the middle piece, which contains the mitochondria, and the flagellum, which of course allows motility, allows it to move and swim. The head has the acrosome, which contains hydrolytic enzymes to digest a part into the female gamete. It also contains the haploid set of paternal chromosomes. Here we have the oocyte, and the main difference between the secondary oocyte and the sperm cell is, yeah, but I feel like I'm getting tired with this thing. Wait. I feel into to skip all of this. I feel all I could read them thing all yourself. Let me tell all a story. It had a Japanese poet, a Japanese calligrapher, right? And he was taking a walk in the rain. He found he wasn't successful at anything. And I think his name was Omikaze. I can't remember his name. And he was taking a walk in the rain. Nice, light, rainy afternoon. Contemplating himself, see, like, me I really good at anything, you know. The last few things I try, me I really get through. So, he see this frog, and this frog was jumping from this lily pad trying to reach this weeping willow. Trying to reach this branch. And he was like, that stupid frog, boy. That frog would never reach. The frog keep trying for you know, probably an hour. And eventually now a breeze blew. And the branch dipped just low enough. At the same time the frog jumped. And the frog actually get onto the branch. May I know what the frog do on the branch after that. But the Japanese poet was like, hey, sometimes it just had to be the right time, yes? But you had to be trying all the time. You had to be trying. If you try, you're gonna never get through. You mightn't get through if you try. But you had to try. And you had to keep trying, because I think at one point the right time will come. I tell you, with me, I used to kind of try. I was always kind of stupid in school. I'm kind of bright now. But you had to keep trying. Let me do some past papers. Sometimes it just had to be the right time. Alright, let me look at some past papers there. Alright, so this is 2019. I'm gonna put that here. Alright, so let me watch here. We're only going through them broad topics ones, eh? Alright, so watch here. Figure 1 shows a light micrograph of the transverse section of the primary stem of a dahlia plant, a typical die cut. And here we have 5 parts, A, B, C, D, E. We went through this diagram already. When you're drawing a plant diagram, I didn't draw this, I get it from somewhere, but apparently this is an acceptable diagram, so I find it look off. But if you want to get your 3 marks for this, it had to at least look like the thing. You had to make sure, so you see how this one have like a certain number of vascular bundles, try and make sure to have the same amount of vascular bundles. Try for it to be a faithful representation of the thing. And you have to label it, and below here you're supposed to say what the labels are. Of course, for any drawing that you do, it don't have a title here, but you title it and you put a magnification. Magnification is pretty easy to calculate if you get trouble calculating magnification. Magnification is basically the image size divided by the object size. So what you do is that you choose a certain measurement in your image. So for example, you could go from what you could do the diameter, and this would be your image. You take this length here and divide it by the length of this. And if there's any conversions to do, you do it. But in this case here, they don't have a scale or anything, but typically you could use a scale bar as well. If they have a scale bar, just use that. Hey, if all I can do magnification up to now, me I know what to tell all you know. Anyway, provide annotations for each label structure A to E. So A is the epidermis, B is the cortex, C is the pit or the medulla, D and E both make up the vascular bundle, and the more inner part here is the xylem, and this part here, E, is the phloem. But D to me is also kind of pointing to the middle of the xylem and the phloem, so it could also be cambium, so they would accept either one. But I think what I was looking for was more xylem and phloem. So the epidermis is the outermost layer of the dicot stem that is covered by thick cuticle. So it's just 5 marks, so one little annotation should be okay. It ought to be as long as this. Is the outermost layer of the stem that is covered by thick cuticle. It has multiple root hairs, protects the under. Anything like this would work, any one of these. The cortex is below the epidermis, and this is mostly used for mechanical support. The pit or the medulla, this occupies the central portion. This has like a lot of air spaces that allow the movement of water, nutrients, materials, also storage of those materials. The xylem, as we know, is used to transport water, and the phloem is used to transport sucrose, right? Food. So this is just all the different things I could put. I will upload these files after, right? YouTube might take a few hours, honestly, to upload the stream, because it's YouTube. It's supposed to be set to automatically upload, but YouTube just take a few hours to process it and upload it, so you might get in the morning or very early in the morning. I will upload all these files afterwards though, like all these past paper things, because I have a bunch of them done already. Alright, so all this is membranes. It didn't really look like it had anything. Will what be in color? Yeah, it will be in color. Me, you know. Okay. Okay, so let me look here. I have a Google Drive. I'll upload everything to our Google Drive and I'll link it. I'll probably post it on community posts. Alright, so figure 3 below illustrates how mutation brings about genetic variation. Complete the missing codons in the boxes labeled A and B. Well, I think this is more nucleic acid, so I can really go through these because this is like, I must skip this one. Alright, look at natural selection question here. Natural selection was proposed by Darwin to be the mechanism of evolutionary change. Discuss the process of natural selection. 2A. Oh, yeah. Let me do this one first. Table 1 shows the relative growth rate data for sensitive and resistant strains of S. typhimurium bacteria exposed to varying concentrations of tetracycline antibiotic. Well, here now, these graph questions, I am bound to go through these here. They'll take kind of long. So I will just upload it and you will go through it yourself, because the graphs here and, all the answers here, for me to go through all of this. Actually, let me go through this one. Let me go through it, yeah? These graph questions, they take too long. Watch now. I have a better idea. Me going through these past papers is going to take a long time because there's plenty of them. So how about we stop the stream here? Because I feel tired and my mouth, my throat hurting. And let me upload everything for you. And all I could burn out the whole night going through the answers here. That's why I feel gonna happen. I know it's an anticlimactic end, oh God, boy. This Cape biology is real work. So I feel like I just upload it and done. And all it gonna be real good. It's not a secret who I am. Some people here know me from before. At least from last year, or some former people who I teach. So it had a certain man who tried to dox me last year. But you can't dox me, I mean, it's not a secret. Yeah, I gotta upload the PowerPoint, I gotta upload everything. So I feel we could done the stream. Well, here stay on the chat, and I gotta just upload the link here. Let me upload everything to the Google Drive. Stay on the chat, I gotta just link it in the chat. Shouldn't take long to upload. It have paper ones in there, paper one answers as well. Alright, I'm gonna upload all this thing there. Only do past papers down to 2013, because before that it had a syllabus shift. So me and bother with the others there. All you do in biology and all you know TS stands for, boy. what going on in all this school? Somebody click that link and tell me if it's working. Everything there, stop asking. Every single resource that you need is in there. So let me close it off. I feeling sleepy. It was nice and rainy today. All you good luck tomorrow. All you go do good. I believe in all you. Me and all you, but I believe in all you. I believe in the nation's youth. Nah, I never get scared. Even when I'm going to fail exam and thing, I don't get scared. It's just an exam. You can do other things in your life. You all take care. Hope all you good. We might run one for paper one, but I don't think we need to. Later as all you might see all you next year for unit 2.